Good morning. Just checking if you can hear me. Are you able to hear me? Thank you.
Thanks. Speakers online, yeah. Our first speaker, Shingai.
Um, can I just talk to
Yeah, you can.
Morning, Shingai. Can you
Good
hear us?
morning. Yes, I can.
Hi, how are you doing?
I'm well, thanks. Is this Greg?
Yes.
I still remember your voice.
You remember my voice. That's good. So we don't even have a quorum yet.
Oh, okay, no problem. I just
Ah,
wanted to check if everything was ready. So once you're
when
ready to get going, you can let me know.
where in the world are you at the moment?
I'm in Tokyo.
In Tokyo. Golly gosh, what's the time there?
It's 7 hours ahead, so it's 3 in the afternoon.
Oh, okay. So it's mid-afternoon.
Yeah,
Well, enjoy the sushi.
I have been.
We'll just give it another 5 minutes if you don't mind. All right.
Okay, thank you.
So while we're waiting, instead of talking about it when we get going, we've got three presentations. You've seen the program, eh? So
Yes.
so you're talking, and then Pia from Afrigen, and then Morena from BioVac, and each one's got 40 minutes, and then we have 30 minutes for discussion. So if you finish early, we can take, any questions for you. We'll hopefully give you your 40 minutes. But if you can make it a bit shorter, then you could have some questions immediately after your presentation.
Yeah,
Because
I don't
by
think
the,
I'll
by the
take more than 30 minutes, so
yeah,
we should have time
because by
for
the time
questions.
Oriana speaks, they would have forgotten about you.
All right, that's fine. I'll keep it short so that we can have a good discussion.
So we can have a few questions for you directly.
Okay.
And will you be able to stay on till the other speakers finish?
Yeah, I plan to, but if anything
Okay.
changes,
All
I'll
right.
drop an
No, that's
email, but I'll
excellent.
I'll try and stay online.
Thanks. So just give us another 5, 6 minutes. All right.
All right, thanks.
Thanks, Shingai. Right, it's 8:02 now. So we'll just wait another 3, 4 minutes and then we'll start.
Yeah.
Is there anybody from Uganda here? I thought there is. Ah, there's a Ugandan. Tanzania? Was there Tanzanian here? So you know we now have an African in the mayoral office in New York. So Mamdani won the election. Mamdani is a Ugandan, brought up also in Tanzania and South Africa, and is now the mayor of New York, and is a left-wing mayor, and hopefully he'll give Trump a clap, as they say. You know what a clap is? A clap is a smack. Okay. So so we look forward to some interesting times in the near future. So Shingai, we do have a quorum, and we're going to start now. So this morning's session is about manufacturing of vaccines and the development of new vaccines, especially in relation to the African context like TB and HIV. And Shingai, and as well as for Pia's benefit, Morena's not here yet. There's been quite a lot of discussion in the last 2 days about the need for Africa to develop indigenous vaccine capacity, given the fact that we having all these outbreaks and that we have the highest levels of zero-dose children. So we look forward to your presentation, Shingai. And I'll give you 2 minutes to introduce yourself, and you can tell them about the fantastic time you had when you were still at VACFA.
Thanks so much, Greg, and hi to everyone. I hope you've been having a fantastic course. My name is Shingai Machingaidze, and I currently work with CEPI, that's the Coalition for Epidemic Preparedness Innovations, and I'm the head of strategy and engagement. So Greg has already told you, I worked with VACFA many years ago, actually, very early days in my career. Initially with SATV, the South African TB Vaccine Initiative. I'm sure you've had speakers from there, with Greg, and then also with the Vaccines for Africa Initiative, which he established. And I worked alongside Prof. Wisonge. So very nice to see this course still running, growing bigger and getting better, and continuing to make much needed impact across the continent. And for CEPI, we are very happy to be sponsoring the vaccinology course from this year and for the next 3 years, because we believe that this is a course that is having real impact on the continent. One of the biggest challenges is not so much building the individual streams of capacity on the continent. We've done this relatively well, exceeding, many expectations over the years. And in some areas, we have advanced to world-class levels. But one of the gaps continues to be how all these connect and how we can use these to respond to an outbreak or, for CEPI particularly, we're interested in epidemics and pandemics. But how do all these different capacities that we fund connect and help us to be, better with stronger health systems? So I hope my presentation is going to tell you a little bit more about the work that CEPI is doing and what we are contributing to this ecosystem. I'll just share my presentation deck. Please confirm if you can see it. Are you able to see the?
We can see it, Shingai. It's fine. Thanks.
Okay, good. Okay. So I'm going to tell you a little bit about the 100 Days Mission. And just as I get started, Greg, if you can help me in the room, could you let me know with a show of hands, how many of you have heard about the 100 Days Mission and know what it is?
So we have about half a dozen to a dozen hands, Shingai.
Okay. In a total of how many in the room?
How many in the room altogether?
Yeah.
Sorry, so I missed your question.
No, I'm
Shingai.
saying roughly it's 12 who indicated they know the 100 Days
About
Mission.
12 people indicated that they know about the 100-day mission.
Okay. Hopefully, by the time I finish, I
And
will have a greater show of hands and understanding in the room. So I'll
it's
start
12
by
out of 12 out of 60, plus
okay,
minus. And then we
all
have
right.
quite a few people online as well.
Okay, great. All right, so I will spend time and go into the details to make sure that everyone then has a good understanding of the 100 Days Mission. So to start, what is CEPI? So CEPI is a global partnership which was launched in 2017. It was launched as a partnership between the governments of Norway and India, between the Bill and Melinda Gates Foundation, now the Gates Foundation, Wellcome Trust, and the World Economic Forum. And at the time, if you remember 2017, this was just after the West Africa Ebola outbreak from 2014 to 2016, which really shook the world, the R&D ecosystem, and our ability to respond to outbreaks. And so CEPI emerged from a consensus that a coordinated international and intergovernmental plan was needed to develop and deploy new vaccines to prevent future epidemics and pandemics. The vision is a world in which epidemics and pandemics are no longer a threat to humanity. The mission to accelerate and develop vaccines and other biologic countermeasures against epidemic and pandemic threats so they can be accessible to all people. So what's unique to CEPI, or what's the value addition that CEPI brings to the ecosystem? We work with governments, with regulators, with philanthropists, with civil society, with the pharmaceutical industry, with academia, such as teams like VACFA. And our unique role is to connect um, uh, different stakeholders and use this with our extensive networks to allow us to pool and deploy resources in ways that countries could not often do so. And this also includes, investments, particularly in R&D, with a specific role to try and de-risk the early stages of R&D, which is usually the bottleneck in progressing candidates in a pipeline. So CEPI is focused on vaccines and it's viral vaccines. A lot of people ask, well, why is your mandate so narrow? This was the thinking at the time that CEPI should focus on a specific portfolio due to the complexity of the types of investments and also the cost of the investments that were required. So to date, we have over 60 investments in vaccine candidates or platform technologies. I'll tell you also about the platform technologies as I progress in the presentation. We have priority pathogens that we focus on, which are chikungunya, and you can see the number of investments for each of these. Lassa fever, MERS, Nipah, COVID-19, broadly protective cough vaccines, Rift Valley fever, and Disease X platforms. Disease X is the unknown. So I'll also spend a bit of time explaining to you some of the work we're doing towards the Disease X portfolio. So since CEPI started in 2017, there have been many programmatic achievements, and I'll take you through a few of them here. So there are what we call enabling projects, and how these were established was by recognizing that in order to progress vaccine candidates for these priority pathogens that had been identified for CEPI, there was need to look at what the enabling environment is in the different regions that would facilitate for these candidates to progress well. And so through that, the centralized lab network was established, which has 17 labs. The preclinical models network, now called the animal models network, also established. The manufacturing network, also established. And then a Lassa fever study, an epidemiological study, the first ever. I'll also tell you a little bit about that, was established. And there's also the research preparedness network as well. So these are the different enabling networks that CEPI has invested in. In terms of investments that CEPI realized, which had not yet been achieved, we advanced the first RVF vaccine candidate into phase 1 trials. the first ever Nipah vaccine into phase 1 trials, the first ever MERS vaccine into phase 2 trials, the first ever Lassa vaccine into phase 2 trials, the first licensed chikungunya vaccine, as well as the first medical product designed using AI to be approved for any indication anywhere in the world. Coronaviruses, I think you've probably heard a lot about COVID and the consequences of COVID and what that means for the R&D ecosystem and vaccinology in general. But CEPI had a very strong investment portfolio for coronaviruses, supported 14 COVID-19 vaccine candidates, 4 of which were granted emergency use licensure, 3 approved for domestic use. CEPI was the leading funder for vaccine R&D, investing in 13 candidates. And of course, you are all familiar with COVAX, which was co-led by CEPI, allowing nearly 2 billion vaccines to be shipped to 146 countries and averting 2.7 million deaths. Of course, there are many lessons learned from these initiatives, but definitely a success for CEPI, but also for the global health community in general. And then with filoviruses, we supported the generation of data to support expanded access and licensed vaccines. And then with Marburg, we were able to support the Ministry of Health in Rwanda to respond to the Marburg vaccine, Marburg virus last year. And you know, this is a very good example of what the 100-day mission means when practically applied at a country level. And within 9 days, the Rwanda Ministry of Health, together with partners, was able to lead a response which deployed a vaccine into clinical trials in 9 days. The trial was focused on healthcare workers, and the outbreak was contained within 42 days. So the 100 days mission itself, we can define it by saying the aim is vaccines should be ready for initial authorization and manufacturing at scale within 100 days of recognition of a pandemic pathogen when appropriate. Currently, the trigger is the WHO declaring a public health emergency of international concern, a FAIC. And so this 100 days mission is at the center of CEPI's work. The way that we build our partnerships, the way that we make our investments is all centered around this 100 days mission. So CEPI has been working together with many partners for the world to understand and embrace the 100 days mission. It's a shift to develop safe and effective vaccines faster, and it requires a number of prerequisites to be in place. So CEPI's part in the 100 days mission is focused on one part of a continuum, but we cannot do what we do without other players in the ecosystem and certain capacities being functional and being efficient. And so some of these prerequisites include having strong disease surveillance and global early warning systems, speeding up the identification of immune response markers, creating a vaccine library and establishing vaccine platforms against representative prototype pathogens across multiple virus families, getting clinical trial and laboratory networks at the ready, establishing global manufacturing capacity, and this includes the regional component, which I think you've discussed quite a lot, to make top quality, safe and effective new vaccines quickly, as well as mitigating biosecurity and biosafety risks. And of course, these prerequisites are supported by an enabling regulatory policy and financing architecture. So there is no use doing, all the science right, getting all the money, if you don't have the regulators on board who are then able to approve and allow for these emergency vaccines to be utilized in the case of an emergency. So you can see that there is a continuum and a connection between many different stakeholders. And so CEPI funds directly certain aspects of this work and then works with other partners towards building these capacities to ensure that the 100-day mission ambition can become a reality. So if you look at the traditional vaccine development timelines, I think what you're familiar with is that vaccine manufacturing takes a long time. This slide said plus 5 years, but in reality, it's more plus 10, plus 20 years from the bench to the shelf or to the arms when it reaches patients. And in our case in Africa, sometimes there's an additional lag where the vaccine is already available. It's licensed, but it takes some time for it to be available to our populations, largely because of financing limitations and the ability to then purchase these vaccines. So if we look at this timeline, which is your research from the lab, progressing to clinical, your phase 1, phase 2, phase 3, and then your manufacturing, your filling, review, etc. In the COVID-19 case, we were able to compress this down to 350 days using the mRNA platform technology. And that demonstrated that if you are strategic in how you make investments, it is possible to compress these timelines through building platform technologies that are adaptable, but also preparing well in advance, allowing for a much quicker process for developing a vaccine. So when we then talk about the 100 days mission, we are looking at multiple phases. There is the readiness, that is pre-outbreak. So these are all the investments that CEPI is making now, together with many partners that are looking at different components or aspects that require investments, that require capabilities to be built, that require collaboration between stakeholders, etc., preparing the ecosystem. Then there is the reaction time. That is between the outbreak and the initial vaccine availability for use. That period is what we define as the 100 days. So preceding the 100 days, there may be years of investment, as in the case of mRNA. There were many years of investment prior to COVID-19, which many people didn't quite understand and mistook that the entire vaccine development process took place within 350 days, which it did not. And then, of course, post the 100 days, there is then the rollout and post-initial availability for use. So the scale up and ability to produce the emergency vaccine means the vaccine is available within the emergency setting. And just to point out that when you have emergency use licensure, this doesn't mean you have final approval. As you know, if you look at COVID-19 vaccines, there's still the process post the emergency to finalize the approval with the regulators. Okay. So I'm going to tell you a little bit more about some of the investments that CEPI is making, and then also take you through some of the enabling networks that we have been supporting. So CEPI invests in new vaccine delivery technology, recognizing that in an epidemic or in a pandemic, you're not necessarily going to be in a city. You might be in remote areas where supply chain, etc., is difficult. And so you really need to think about alternative vaccine delivery technologies such as vaccine patches, pills, under the tongue films, nasal vaccines. And there's a particular focus or consideration of Africa, of South America, of Southeast Asia, where these kinds of new technologies would make a very big difference in terms of how quickly we're able to reach our populations with vaccines that do not require a cold chain. CEPI also recognizes that you have to build the capacity to track the disease outbreaks, right? So epidemiology and modeling becomes extremely important. And this ability needs to be built and embedded within our health systems and our outbreak response mechanisms in countries, but in between countries, and then in between regions as well, right? So your global response is only functional if you have a coordinated regional response. And a coordinated regional response is only functional if individual countries have the ability to respond and manage a response in their country and the ability to connect with other countries within their region. So you can see that it is a continuum. I mentioned already that we have the ENABLE study, which is the largest ever Lassa fever research study currently taking place in West Africa. And Lassa is, for those who of course know about Lassa, it's a seasonal outbreak, and it is devastating in the areas where it does occur. But of course, this is not a disease that is widespread going beyond West Africa. And so this study was set out to give researchers a better picture and a true disease burden, which of course is important when you're trying to develop a vaccine. And of course, ultimately, when you then decide, once you have a vaccine, to design a program that will then respond to this seasonal outbreak. So since 2019, 23,000 participants have taken place. Yeah, I think it's really been a learning curve for CEPI, but also for the member states in West Africa who have really been leading from the front in this work. CEPI also invests in standardizing vaccine development. So there's a working group that was established together with WHO that looks at standardizations and assays. As you know, a big part of vaccine development is the lab, standardizations and assays, and the ability to be able to do these together in the same way in different places is what will allow you to be able to respond quickly and together. So there are standards, international standards that have been developed for Lassa fever, for MERS, and for COVID-19 with the support of CEPI. And the team works very closely with FIND and with PATH to identify gaps in the creation or validation of diagnostics for priority pathogens. So there is no way for you to develop a vaccine or a therapeutic without a diagnostic. And your ability to have that diagnostic, and in our setting in Africa, where most of you are working, a point of care diagnostic becomes even more important to be able to respond quickly and within the field. So that was about the different types of investment that CEPI makes. And now a little bit more about the enabling networks. So, as I go through these, it should start to help you show how CEPI is trying to connect the dots to help Africa and, of course, other regions around the world be better prepared. So the preclinical or animal models network has labs in Africa, in Kenya, and in South Africa that are part of the network. And sorry, I think I've gone a slide. Okay. There we go, sorry. So the CEPI network of labs performs network of biocontainment labs for performance of model development studies and preclinical vaccine testing. It has BSL-3 and BSL-4 partners which can handle the CEPI priority pathogens. There is compliance with high ethical standards, very high quality research coming out of this network. And the objectives are to develop and characterize preclinical models of viral disease, the use of preclinical models for vaccine testing from proof of concept to licensure and enabling studies, as well as preparedness and capacity building. For the CEPI centralized lab network, there are African labs, they're part of the CLN, which are in South Africa, which are in Uganda, Kenya, and Senegal. The CLN supports CEPI-funded and non-funded vaccine developers to evaluate CEPI priority pathogen vaccines against common protocols to ensure alignment and information sharing when identifying the most promising candidates. The objectives are to test subsets of samples to ensure comparability across programs, testing complete sets of samples from preclinical and, of course, in clinical trials, to facilitate rapid evaluation, approval, and dissemination of the most effective vaccine candidates, as well as preparedness and capacity building. And some of the available assays are listed at the bottom. Then the Clinical Research Preparedness Network. So this is, I think, a very interesting approach and a new approach, which is thinking about if you want to be able to respond during an outbreak, you need to have built the capacity when there isn't an outbreak. And so you need to leverage the routine vaccine development capacity, clinical trials capacity, and be able to pivot that in an emergency response. But when there is no emergency, you need to make sure that you're able to use those capabilities for routine work. And I think that is a challenge or a gap where intentional investments are needed to build up these routine research facilities such that they remain viable and able to continue working, and then be able to respond if and when there is an outbreak. So the pictogram you can see there shows you track A and track B. And track A would be routine, clinical vaccine development, clinical trials. So the goal is to strengthen clinical trial capacity for GCP compliant late-stage clinical development. For us, of course, focusing on CEPI's priorities, supporting all vaccines on a portfolio level, and targeting key geographies in the Global South. But important to note that when you build capacity for clinical trials, while there may be a focus with CEPI on its priority diseases, the general routine capacity, if built in a manner to promote sort of a disease agnostic approach, it means it would apply for CEPI priority pathogens, but also for others. So that's a very important component about the approach being taken in supporting the clinical research preparedness capability function. If you look at track B, this is now the emergency response during future outbreaks. And this is where the 100 days mission ambition comes in. If you think of that timeline that I showed you a few slides ago, the goal for this is to leverage the routine investments that have been made in track A to support long-term evidence generation readiness across regions. And that's embedding all infrastructure strengthening in national and supra-regional preparedness plans, close collaboration with Africa CDC and now the Africa Medicines Agency, supporting the hub and spoke model for clinical trials, which I'll tell you a little bit about now, as well as working towards a self-sustained infrastructure. The hub and spokes model is an approach that working in partnership with Africa CDC and many partners in the ecosystem is recognizing that we have a cycle where we build up capacity and lose capacity in Africa, largely because most of our clinical trials are grant funded, which means you build up the capacity for your grant, and unless you secure another grant when your current grant ends, you then lose that capacity, sometimes lose staff, and then you get a new grant, and then you have to start building up the capacity again. And so that continuous cycle is actually quite disruptive, but most importantly, it's expensive. And an outbreak can take place when you're in between grants, which means you're not functioning at your full capacity, meaning you're not able to adequately respond to the outbreak. So that's the connection now with the hub and spokes model, which is then now saying, how do we work together in the ecosystem to identify key hubs who can be capacitated to be able to have sort of a coordinating, supportive function to other clinical trial centers around them. This obviously means these hubs would be distributed across the continent, in Africa in particular, and there would be shared standards between funders to understand sort of what are key or minimum standards required for clinical trials, whether it is about data infrastructure, whether it is about how we're building the trial infrastructure, the laboratory, etc. And that way it means then you are not only dependent on one stream of funding, or designing your entire clinical trial site just according to what one funder wants. So that's quite important, and that's ongoing work with Africa CDC, with the AMA, with many other partners. And then CEPI is working on this through what we call technical coordinating partners. And these have been identified and are working alongside African partners to build this capacity. The technical coordinating partners, who are supported by CEPI, are an interim measure that's helping build up capacity for this coordination function, with the idea that they would be phased out, and this coordination would then continue within the regions. So the network you can see on the right-hand side, it started with the West Africa Network, which was launched in 2003. And we have now since begun the work on the East and Central Africa Network. So the West Africa Network started and particularly focused on Lassa vaccine simply because of the region, and also because of where the vaccine candidates were in the pipeline. And East and Central Africa is obviously going to focus on Ebola, Mpox. We have some trials on Rift Valley ongoing as well. There was a bit of a chikungunya scare that was coming in from Madagascar. So that's the work of the East and Central Africa Network, but it's only getting started. And then you can see that there's plans for a Southeast Asia network as well. And then looking at how these different networks also then speak to each other. So the main program characteristics are to support regional coordination of evidence generation in future epidemics and pandemics, to facilitate growth of clinical research entities for competitive institutions which can survive, independent from project-related funding, the TCPs that support the work, aligning with existing regional programs put in place by agencies such as Africa CDC, as well as leveraging existing infrastructure and experience rather than duplicating what's already in place. And the CEPI Vaccine Manufacturing Network. This empowers manufacturers in low and middle income countries with limited experience in rapid tech transfer, scale, ramp up, etc. And it also identifies high performing entities that has demonstrated competencies in rapid scale up for manufacturing to deliver the 100 days mission in the short term. So in South Africa, we have Aspen. You will hear from Afrigen and BioVac. They're not part of this network, but CEPI definitely recognizes them as key to building Africa's manufacturing capabilities and is working with them. And we have Institute Pasteur in Dakar. We also then have the GS Learn, which is the Global South Leaders in Epidemic Analytics and Response Network. This is the newest network, and this is really to the, established to respond to, the gap to build the epidemiological and modeling capacity that's required for rapid epidemic modeling. And we're at the final stages, and I think the selected institutions should be announced soon, and we should be able to share that information. So I think this is a summary of, this is a summary of the different networks, and you can see the spread of CEPI around the world. And a big part then of what we do is, in order to be able to respond to this 100-day mission ambition, how do we connect all these different functions in a way such that you are able to mount a response, as was demonstrated by Rwanda with a Marburg response. So I'll stop there, and I hope I've been able to give you a little bit of an understanding of some of the work CEPI is doing to support Africa to be better prepared for the next epidemics or pandemics or current epidemics or outbreaks that are taking place. Thanks so much, Greg.
Thanks very much, Shingai, for a wonderful presentation. And hopefully we are in safe hands with CEPI. And you've done very well in time. We actually have 10 minutes of Shingai's time for questions. We're going to have questions at the end of the session after the other two speakers, but maybe we can take some questions for Shingai directly. So who would like to start with questions to Shingai? So, oh, okay, we have one at the back. Just give your name and your affiliation so Shingai knows who is talking.
Stelo from Africa Health Research Institute. The issue of the focus, I see it's on the research, particularly clinical trials. The issue of funding in terms of clinical trials and mostly the investigator-led studies. Sometimes, especially African investigator-led studies, they normally don't get so much of the funding. And we've seen several cuts on ACTG, NIH, and all those studies that were funded by US. How is CEPI trying to intervene and prioritize research in that regard?
Thanks so much, Greg. Should I answer and then we take the next question?
Answer while people are thinking about additional questions.
Okay. Yeah, so I think that's a very relevant question. The current R&D funding landscape is tough. There definitely was a very big reliance on the US funding. About 60% of R&D was funded by the US, which of course has had a ripple effect across the entire ecosystem. CEPI continues to make its investments in R&D as per the different investment areas and approaches that I've told you. But again, CEPI is only one player in the field. CEPI is a sort of a niche stakeholder within the R&D ecosystem. So they focus on specific types of technology, they focus on priority pathogens. And so we may not directly respond to a funding gap in TB research, but because CEPI is continuing to build capabilities and capacities across different functions within the vaccine ecosystem, we hope that the investments that CEPI is making will still indirectly be able to bridge some of these gaps. But we continue to have a focus on our priority pathogens and the types of technologies that we focus on. And of course, with other stakeholders, then advocating for a reassessment and thinking about how investments are being made. And of course, encouraging countries to invest in the local R&D ecosystem as well. Thanks.
Thanks, Shingai. Next question.
Hi, thank you so much for a lovely talk. Sarah from Afrigen. I was wondering about, a lot of the hesitancy around the COVID vaccine was to do with how quickly it was produced. And so do you have any insights into how to tackle this if you're trying to shorten that time period, if people will have confidence in that, and how you would deal with that?
Thanks so much. I think vaccine hesitancy is something you've covered or are still going to cover in this course, and it's incredibly important. We have such high rates of vaccine hesitancy, even for approved vaccines that have existed and been available for decades. And so when you look at it in terms of an emergency response, and then you also apply the further compressed timelines for these new technologies and the 100 days mission, you do have to critically think about how to tackle a vaccine hesitancy. But one of the points I made when I was explaining where and how the 100 days mission fits in on the timeline is that preparatory phase exists for different lengths of time. It may be several years prior to when you actually have an outbreak. So I think a big part, particularly with the 100 days mission, is being able to communicate the research that is being done for these different technologies and these different pathogens, so that when you do then have an emergency and you are able to produce a vaccine with a compressed timeline, it's with the assurance that adequate investments and time have gone into building the technologies. With a platform, you work towards getting an approval of the platform technology, which means that a lot of work has gone into the quality and safety approvals of the platform. Right? So then when you do have an outbreak and it's the change of the specific antigen that's causing the outbreak, you are then able to respond faster. But again, it's a process. You must make sure the regulators understand the science, believe it, and support it. Scientists and researchers such as yourself have to be included in the process and understand it. And then, of course, this has to translate to what we collectively then tell, the ordinary person who needs to take the vaccine. And again, I think emergencies are different. When we had COVID and there was the initial response, people's willingness to take vaccines was very high at the start and then reduced with time. And it's always about that personal risk calculation that people are going to make. So there isn't an easy answer, but it definitely requires progressive investment in making sure that you're taking stakeholders along the journey with you in the development of new technologies. So there's an understanding that being smarter in how you invest and how you build the technology does not compromise on the quality and the safety of the vaccine. That's the message that we need people to understand.
Okay, we have got 3 minutes left for you, Shingai, and we have 3 quick questions, and Shingai, 3 quick answers.
Okay.
Good morning, Shingai. My name is Tepi, but in Cape Town, they all called me Sepi. So, so I just have a question online from Mubarak. He's asking, how many African countries are you currently implementing in or have partnerships with?
That's a very quick one. So far, we have 27 that have benefited from CEPI's support in one way or the other.
Thank you. Quick question.
Thank you for the presentation. I'm Sherifat from the Clinton Health Access Initiative. So my question is quite similar. I want to know how many countries on the continent are actually eligible for CP.
I guess all countries would be eligible. There is no restriction for funding. It's just you need to be able to respond to our calls for proposal, but there is no restriction.
Okay, and then one last question from our right wingers. Who had a hand up? You.
Thank you for the presentation. This is Valerie from Desmond Tutu Health Foundation. I just am wondering, with your model of expediting, like doing the clinical trials in 100 days, I understand that the preclinical happens before this, but do the clinical trials, the phase 1 to 3, happen concurrently? And to be able to evaluate safety and efficacy, are you increasing your number of participants to achieve this? Or?
Yeah,
Very good question.
Very good question. And I think it depends on the candidate. A big part of what we're doing depends on a lot of prior investments that would have taken place. And for example, your platform technologies that would have been built. In order for you to build a platform technology such as mRNA and have this platform technology approved, you have to do so through taking a candidate through the platform to licensure. So you will have already done this work in order for you to approve the technology. What then changes is what changes in an outbreak, for example, would be a new antigen. So you have a lot of the preliminary data that has already been done within that particular viral family and with that particular technology. And so you are not trying to do phase 1 to 3 entirely within the 100 days. So I think that's the best way for me to explain it. But it all depends on the different types of technologies. And then also an added element, which is perhaps a little bit more unnerving to some people, is also there's a lot of exploration into alternative pathways to licensure, recognizing that it's very expensive to continue with the existing model. You know, the chikungunya vaccine that was licensed followed an alternative pathway to licensure, which didn't follow the traditional model. So again, these are all the different elements that we're trying to think about and say, how do we, take these forward? Not easy, but I think very necessary. Thanks.
Shingai, thanks a million. Can we give a round of applause for a wonderful presentation and setting the scene for the two subsequent speakers? Shingai, you'll hopefully be able to stay with us till later on when we have some time for additional discussion. So we've been talking about mRNA, and we have the pleasure of inviting Pia Singler, who is from Afrigen, and she's going to tell you what they're getting up to with respect to mRNA hubs in Africa. Thanks, Pia. And you can give them an additional 1-minute introduction about yourself.
Me. So I'm the principal investigator at Afrigen. So I'm leading the TB vaccine development there. And I'm also a subject matter expert in preclinical studies at Afrigen. So I would like to give you now an overview about the current state of the manufacturing space in Africa and an update of where Afrigen is. So currently the global vaccine manufacturing space is centralized in just a few regions. Over 70% of global vaccine production by value, meaning by revenue, is just from 4 companies that are located in high-income countries, particularly located in North America and Europe. And these manufacturers make use of advanced technologies such as subunit, viral vector, or mRNA technology. In contrast to that, over 70% of global vaccine production by volume is produced in developing countries, but also mainly in 3 countries, which is India, China, and Brazil. And these companies in the developing countries make use of well-established platforms such as live attenuated vaccines. So they are making vaccines which usually have a much lower profit margin. And this lack of geodiversification in the vaccine manufacturing space was also reflected in the unequal distribution of vaccines during the recent COVID-19 pandemic. Whereas many people in high-income countries already received their second dose, there were many people in low-middle-income countries, particularly here on the African continent, that had yet to receive their first dose. And there is the growing consensus that we need the vaccine manufacturing space to be geodiversified to have global health security. And this is what the vaccine manufacturing space looked like in 2021, shortly after the onset of the COVID-19 pandemic. We had a total of 7 vaccine manufacturing sites. What's very important to mention here is that these sites were focusing mostly on downstream processes. And downstream processes means they're filling and packaging vaccines that are actually made overseas and just imported into Africa. So they're not producing vaccines themselves. And the reason for that is that in general, vaccine manufacturing is quite a high-risk undertaking. And particularly the upstream process, which means the development and production of the active ingredient, is really high risk. So it's much lower risk to be involved in the downstream processes, which includes just the packaging and filling of the vaccines. So in general, we heard already, making a vaccine is very complex. Only about 1% of all the vaccine candidates make it from discovery onto the market. And it usually can take 10 years. And that means for a manufacturer, this is a really high risk investment, and the failure rate is extremely high. So, and this is one of the reasons why Africa has only little vaccine manufacturing capacity. And when we speak in numbers, Africa actually only produces 1% of its vaccine requirement on the continent, although it is consuming 25% of global vaccines by volume. But we want to change that. Together, the African Union and the African CDC established a vaccine manufacturing framework with the goal to produce 60% of local vaccine needs by 2040. And that's a very ambitious goal, but it's a goal that we can achieve. The World Health Organization established the mRNA Technology Transfer Program in 2021. And this was done together with the Medicine Patent Pool. And right at the center of this program is Afrigen Biologics, which is here in Cape Town. So Afrigen Biologics is establishing an mRNA manufacturing platform and is transferring all the knowledge and technology to 15 other low and middle income partners so that they don't have to build up their facilities from scratch, which means they save time and cost. And in total, these partners constitute a population of about 3 billion people, meaning, change the slide automatically, meaning that about 40% of the population can be supplied with vaccines. So why mRNA technology? So mRNA technology has many advantages. From one perspective, from the manufacturing. It allows rapid development of new vaccine, which makes it ideal to respond to outbreaks and to pandemic. It is easy, scalable in production. It works according to a plug-and-play principle, which means that once the manufacturing platform is set up, you basically only have to change the antigen to adapt to a new disease and produce a new product. It is relatively low in cost, and I have to say relatively because it's still very expensive, but it also allows small footprint facility, which of course also overall reduce the cost. Then from a vaccine perspective, the vaccine is non-infectious, meaning it is good to be given to immunocompromised individuals. For example, with live attenuated vaccine, you always have the risk of reversion that the pathogen is actually causing a disease. That's a risk we don't have with mRNA vaccines. And during the COVID-19 pandemic, we also could demonstrate that mRNA vaccines have a high clinical safety profile. mRNA vaccines also induce humoral and cellular immune responses, which makes them useful to be potentially used for a wide variety of diseases. So we also heard before that the traditional way of bringing a vaccine from discovery to the market takes very long. But we also have showed that it can be different. So in the COVID-19 pandemic, the sequence was publicly posted in January 2020. It took only 1 week for the first labs to initiate the development of vaccines. And about 2 months later, we had the first vaccines in clinical trial. So we actually managed to produce a vaccine from beginning to the end in about 10 months' time, which also shows the potential of mRNA vaccines. So to now foster the vaccine independence in Africa, we in Afrigen established us to be in a very unique position. We are the first end-to-end mRNA vaccine manufacturer and developer, which means that we can do everything from start to finish in-house. So we can do own in-house research and development in our R&D facilities. Then we can manufacture the vaccine in our GMP facilities. We can do in-house quality control of the products that we are producing. Then we can fill and finish them and then release them and ship them off to their destination. Here you can see an overview of our R&D facilities where we, for example, work on formulations, on adjuvants, doing analytic testing, including in vitro testing, purifications, and we also produce the pDNA. For R&D, we work closely together with local academia because we, for example, at Afrigen don't have the time to invest years into finding the right antigen. For example, for our TB vaccine, we're working closely together with ZV, who has discovered antigens that we are now using to develop a TB vaccine. And also Afrigen doesn't have any animal housing, which means we are dependent on local university partners to perform our preclinical studies. So these are our GMP facilities. Our GMP facilities are over 2,000 square meters big, and we have different clean rooms, as well as we have QC laboratories and a stability room. And in the stability room, we then have vaccine material, which is stored under different conditions and different humidity to know the stability over time at different climate zone, at different storage condition, and that we can ensure that the vaccine remains of high quality during its entire shelf life. So how to make an mRNA vaccine? It is basically a three-step process. The first step is always to produce pDNA. The pDNA is the template of the mRNA. The mRNA is produced in a cell-free in vitro transcription reaction, which is also known as the drug substance. Then it's extensively purified via tangential flow filtration, and we also have our expert from Afrigen here, and they can tell you more about it. And then we formulate the purified mRNA into a lipid nanoparticle, and now we're talking about a drug product. And the drug product, again, then gets buffer exchanged into an isotonic solution suitable for injection into humans, and we also add sucrose. And sucrose is really important because it's a cryoprotectant, and that allows us to store the vaccine at really low temperatures, such as minus 80 degrees. And then we can fill and finish our vaccine. So at Afrigen, we have two processes that we can use to make the vaccine. One that you saw before is the conventional process. The other one is a semi-automated process. The difference between the two processes is only the drug substance. So the drug product as well as the pDNA is made similarly. So as you can see here, for the drug substance at the semi-automated process, we have a machine, and that machine is provided by Quantum in Belgium. It's about 2 meters big, and it within 1 day can produce about 4 grams of mRNA. And that is much more than we can do in a conventional process, which takes us much longer. So the semi-automated process comes with many advantages. It saves us time and it saves us cost. But it also comes with a very big disadvantage. We in Africa have long delivery times. We are still dependent on sourcing materials from Europe, from US. So we have a lot of backlogs often. We have to wait for reagents. And specifically during pandemics, there is always the risk that we might not receive our reagents that we need and have to halt the production process. So that's why it's very important to have the conventional process, because that one is allowing us to source from different suppliers and to always have a continuous production of vaccines. So looking at the outcome of these processes, we see there is quite a big difference. So the conventional process means that in about 4 weeks, we can produce 25,000 doses per run. And with the semi-automated process, we can produce 45,000 doses in about 3.5 weeks. So, and then we have to quality test our vaccine. We have to make sure that our vaccine is of high quality, and therefore we had to develop a lot of tests. Here you can see the list of tests that we do for the drug substance on the left and for the drug product on the right. And some of the assays are highlighted in blue, and these are the assays that we actually have to always newly develop for every new vaccine we have, because they're either antigen specific or plasmid or construct specific. So I've talked a lot about how our facilities look like and what we're doing, and I would like to give you a visual tour. I don't know how to start it. There's no starting button. Is it working?
Sounds like we're into rapping now.
We'll just click, we'll just click next. Sure.
Afrigen Pharma is a proud African biotech company nestled in the beautiful city of Cape Town, South Africa. We have built world-class Afrigen Biologics is a proudly African biotech company nestled in the beautiful city of Cape Town, South Africa. We have built world-class capacity and capabilities to support Afrigen Biologics is a proudly African biotech company nestled in the beautiful city of Cape Town, South Africa. We have built world-class capacity and capabilities to support vaccine research, development, and manufacturing. Our facilities have been designed on a modular basis, with distinct sections dedicated to the manufacture of drug substance and bulk drug product, fill and finish into ready-to-use final products, and quality control laboratories. These manufacturing modules provide capacity for end-to-end production, involving the synthesis of plasmid DNA, in vitro transcription into mRNA drug substance, encapsulation of this drug substance into lipid nanoparticles, and final filling into vials. The universal nature of mRNA technology means that this single facility can be used for multiple disease targets. The pDNA manufacturing suite is a Class C biosafety level 1/2 cleanroom in which E. coli is used to amplify plasmid DNA, which is the starting material for mRNA synthesis. Purification via chromatography and tangential flow filtration is also performed here to generate purified circular plasmid DNA. Adjacent to the pDNA suite is the mRNA synthesis room, where mRNA is manufactured through a cell-free in vitro transcription process, followed by column chromatography and tangential flow filtration to generate purified mRNA drug substance. Here in the formulation room, the purified drug substance is encapsulated in lipid nanoparticles using impingement jet mixing. Tangential flow filtration is then performed to remove ethanol via buffer exchange. Finally, a cryoprotectant is added to generate the bulk drug product. A prime example of Afrigen's innovative technologies and early adoption strategy is the Intensify MIDI machine developed by Quantum Biosciences. This next-generation manufacturing technology facilitates the automated manufacture of mRNA drug substance at a significantly reduced price. The sterile filling line offers a Grade A environment built into a Grade B area. This state-of-the-art semi-automated filling line allows for the production of multi-dose vials varying in size from 2R to 20R vials. A unique feature of our manufacturing line is the flexibility to produce various batch sizes, from just a few hundred vials, appropriate for clinical trial material and rare diseases, up to a commercial scale of 50 million doses per annum. Testing of incoming materials and products at various stages of manufacture is at the core of our efforts to produce safe and effective products. This function is the responsibility of our quality control team, with analytical, tissue culture, biochemistry, microbiology, and PCR laboratories. These are well equipped with the required instruments and staffed with trained analysts to ensure that testing is robust and comprehensive. The analytical laboratory has capabilities which include FTIR, TOC, and a zetasizer for nanoparticle size analysis, amongst others. The biochemistry laboratory augments these tests and permits us to utilize HPLC with different detectors for various analyses, a nanodrop to determine mRNA concentration, and plate readers to perform endotoxin testing. In the PCR laboratory, we can perform quantitative measurement of targeted genes. We rely on our microbiology laboratory to conduct routine testing on water samples, perform environmental monitoring, and test samples for sterility. The sterility testing isolator, a key component of the microbiology lab, is a completely controlled system, providing a controlled environment for reliable sterility testing of vaccine products. These in-house capabilities mean that Afrigen Biologics is self-reliant when it comes to ensuring the quality and sterility of our current products, while also preparing our site for future innovation. Stability studies are integral when developing new pharmaceutical products and vaccines, ensuring quality, safety, and efficacy. A variety of studies can be conducted, including real-time stability, accelerated stability, forced degradation, in-use stability, and shelf life. The stability chambers offer a vast range of temperature and humidity conditions, varying from -80 to +40 degrees Celsius, to simulate different climatic zones. Stay tuned to see what Afrigen Biologics has to offer, not only the continent of Africa, but the world.
I'm not done. So yeah, we're currently developing several vaccines in our facilities. So they're all at the developmental stage. We work on a TB vaccine, RSV, influenza, gonorrhea, HIV, Rift Valley fever, and Mpox. And all of these diseases were identified by the WHO as priority diseases for Africa. And all of these diseases we are working on in collaboration with a lot of different partnerships. So we have partnerships for improving mRNA construct development, partnerships to identify additional disease targets, specifically with academia, who helps us to identify which antigens to use in our vaccines. Then we do formulation research, which is really important because the formulation we are using currently is built up on Moderna's vaccines, which means it's IP protected, as well as it has the disadvantage that it's not thermostable at higher temperature. So we want to find a formulation that can be used, for example, for storage at fridge temperature. Then we also work at different or improving, optimizing manufacturing technologies to make our production more cost effective. And we are also working on alternative delivery methods. We have heard before that is an issue to apply, for example, vaccines intramuscularly, specifically in resource-limited setting where healthcare workers are scared. So we are working on a needle patch which can be easily applied onto the arm without needing skilled trained healthcare worker. So in all of our collaborators are sitting in different areas of the world. We basically have collaborators all around the globe. And we need to build up this network. So this slide I showed you at the very beginning. So in 2021, we had 7 manufacturing sites. And that is where we are now. In a really short amount of time, we expanded to having now 25 manufacturing sites in Africa. And that brings us closer to the goal to being able to produce 60% of our vaccines by 2040. I do have to mention that the majority of these manufacturing sites are not operational yet, so they don't produce vaccines, similar to Afrigen, but it is only a question of time until we do. And I also want to highlight that just having vaccine manufacturing facilities alone is not enough. We need to build a whole vaccine ecosystem. We have to have a strong research and development foundation where we where we have collaboration between industry and academia for antigen discovery, for this, development of next generation vaccines, particularly improving the formulation for thermostability, freedom to operate. Then we have to have preclinical and clinical testing sites in the country or in the continent. We have to optimize our manufacturing processes to make our vaccine more cost effective. And we need human capital. So we need to train young scientists, engineers, and pharmacists. And we also need to be able to retain these talents, because brain drain is a serious issue in Africa. So then we also have to work on the regulatory strengthening, which means we have to have regulators in every country that are able to approve the vaccines that come into the market. And we have to have a harmonization between the regulators around Africa. Then to the market, we do have to have a reliable supply chain, meaning we need to have GMP certified supplier. So at the moment, we are getting the majority of our reagents consumable from Europe and America. And the issue is long lead times, import permits, and this leads to delays. We need to have local service support. We often have equipment, or the majority of our equipment is actually from Europe, meaning if anything goes wrong with our equipment, we have to have online calls to figure out what the problem is. And if we can't figure that out in-house, we need to have someone flying in, which again is very cost intensive for us, and it also delays the process. And we also have to remember that in most cases, we have one single equipment, meaning if this equipment breaks down, we have to halt our production. Then we need to be cost effective and competitive in the global market, which at the moment we are not. If we are producing vaccines, our prices are higher than, for example, vaccines produced at big pharmacies. Then when we have the vaccine, we of course also need to have the infrastructure for distribution and administration. Thank you very much.
Thanks very much for that presentation. We not on. It is on.
No.
Must I speak louder? So Pia, thanks, Melanie. We have some time for specific questions to Pia. Some people have complained about the lighting here, and being able to see the slides, but I think we'll sort it out during tea time. All right. Okay. Questions. We have one on the right-hand side here.
Thank you, Pia, for that lovely presentation. My name is Nicholas Alexander, and I'm very interested in one of your last slides was talking about helping or what the pillars of a sustainable vaccine ecosystem. Can you maybe share with us a little bit the role of Afrigen in supporting the Africa CDC's regional capability and capacity building networks? Thank you.
So, Afrigen is actually working closely with our local authority together with ZAPRA. So Afrigen is the first mRNA facility. So, for example, the authorization of a GMP facility that produces mRNA is quite new. So basically, Afrigen is learning together with ZAPRA on how to establish these facilities. And Afrigen has also been authorized to be a training facilities for other African countries to come and learn how to build a GMP facilities for mRNA production.
Thanks, Pia. So if I may ask, I mean, the understanding is that Afrigen facilities are in development. So when is the first vaccine going to roll off the?
That is the big question. No, I know that's on everyone's mind. So Afrigen's facility is actually set up. So we recently had our GMP inspection. So we're waiting, of course, for a good outcome. So once we have our GMP license, we are basically ready. And the first vaccine that we will produce is the Rift Valley fever vaccine, which is done under the 100-day mission with CEPI. So we will be ready to produce the engineering batch, which is the last stage in the production process next year. And then we will still have to proceed with GLP studies. So we presumably start with clinical studies at the end of next year.
So it takes quite a long time to get a vaccine into, if I could say, the market from the time you started your facility.
Yeah, I think what is often underestimated is also the amount of time it takes to finish the preclinical studies. A GLP toxicology study can easily take several months, like 4 months if we are fast. And from our experience, it took actually 6 months to complete.
Okay, we have a question on the left-hand side there.
Hi, thank you for the presentation. My name is Julia. I want to ask you about the intellectual property sharing. So as we know, that's quite important part of producing and especially for strengthening the local manufacturing. How are you sharing your intellectual property with other manufacturers?
So we are sharing IPs freely with all the partners that are part of the mRNA technology transfer program. So this collaboration allows everyone who is developing or establishing a new IP to share this IP with the partners. So it is basically a collaboration around different low and middle income countries.
Mike?
Yeah, Pia, thanks very much for that excellent talk. Could you just clarify whether you're adapting the mRNA to those multiple vaccine preventable conditions? I think you mentioned yellow fever as the one vaccine in development. Could you give us an idea of how you've prioritized the others and what the kind of pipeline is showing?
Yeah, so yellow fever wasn't in our pipeline. We have Rift fever. And yeah, priority basically at the moment, we set Rift as a priority because it's a part of the 100-day mission of CEPI. So this is one of where we have to demonstrate that we can really quickly produce the vaccine. Our next priority target is based on the priority on the African continent. So the next priority is TB, which is very relevant for Africa. So this is our second priority.
Sorry, what was the priority?
Rift Valley fever.
Rift Valley fever. Okay. And the next one on the list?
TB.
TB. Okay. Well, we're going to hear more about TB later on from Tom Scriver, so we won't talk more about that. Tom, we had a question of you?
Yeah, I wanted to know where are you at when it comes to the TB vaccine? You said you're working on that. If you can elaborate a little bit more about that.
Yeah. So the stage where we are with the TB vaccine, and I don't know if you're talking more about it, Tom. So we are currently doing the preclinical studies. So we have produced a vaccine in our R&D facilities. We have upscaled the vaccine. We have produced 100 milliliters of the vaccine material, which is then now tested in animals. And we have already proof of concept data where we showed our vaccine is immunogenic and protective and comparable to BCG.
I saw on your list that you had RSV as well.
Yeah.
Because that's, I mean, if you're talking about, I mean, Rift Valley fever is important, but I would assume it is a localized disease in certain regions. But RSV is a major disease causing major problems in the early neonatal period, and I mean it's a global problem, big killer, and currently we only have 1 or 2 manufacturers of RSV vaccine, and they're very expensive vaccines.
Exactly.
And I mean, Gavi is pushing to get it onto the global program with the Gavi 6.0 strategy coming into place next year. So I mean, in terms of priorities, I would think you should be going for RSV.
Oh, really?
So where in the pipeline are you with that, for example?
So the RSV is in a much earlier state in development. So we are starting animal studies likely in February next year. So yeah, that will be the first animal studies we do with our vaccine.
We have some questions from online.
Thanks, Pia. We have a question from Abdul. So he's saying, given that most vaccines currently used in Africa are produced using conventional platforms, such as live attenuated or inactivated approaches, while mRNA technology is gaining attention for its streamlined production process, does Afrigen intend to contribute to the advancement of vaccines using these traditional platforms as well? Or is the strategic is their strategic focus on mRNA vaccine development?
Yeah. So at the moment, the plan is to focus on mRNA development and use the platform MC, but it's suitable for many different diseases, and as well as in the future, also cancer therapy.
Thank you. I just wanted to ask in terms of market shaping, can you give us like an update on what's going on? Because it's great to know that vaccines are being produced in Africa, but if some African countries can still procure at lower prices from Europe, for example, they might still go for those vaccines. So just wanted to know if there's any work going on market shaping.
Say again, if we can procure more vaccines?
Market shaping in terms of prices.
Market share. Oh,
yeah.
yeah. Yeah. Okay. Yes. I think, yeah, definitely. I mean, price is definitely a big factor that we have to consider. So we have to be able to produce a vaccine that is affordable for low and middle income countries, which at the very beginning, I think might be challenging, but we do have a cost calculation that we will fall, for example, for the TB vaccine within the $10 range. And we, in the future, once we start to optimize manufacturing processes, will also be able to lower this price.
Any more questions? If not, just save your, oh, sorry, Mike.
Yeah, I notice it's biologics and vaccines. So are there any moves to, for example, manufacture a low-priced monoclonal? Because to come back to Greg's question, you know, in terms of RSV, that can also be addressed through monoclonal antibodies, but cost clearly is the impediment.
Yeah, yes, that's a good point. So we don't at the moment work on monoclonal antibodies, but for example, we also work on adjuvant formulations. So we're not solely concentrated on mRNA vaccines, although that is our main focus at the time. We're also working on formulation, adjuvants for different vaccines. In the future, potentially, we will also work on monoclonal antibodies, but for now, we focus on mRNA vaccines, yeah.
All right, Pia, thanks very much. Can we have a round of applause for Pia? So the last but not least person in this session is our good friend from down the road, Morena Makwana, who is the CEO of BioVac. And Morena, we look forward to your presentation. And if you have a chance, you should actually go and visit Morena. He's just down the road. Thanks, Morena.
Thanks, Greg. Just like to acknowledge VACFA for not only calling me back, and fortunately I just happened to be in town because there's always so much travel. So I always try and if I can make it, try to be here myself because I just value the importance of vaccine courses. The little bit of that I know, and I think with each year I know less about vaccines, but the little bit that I know of vaccines is really through these courses. So I do value it a lot. But yeah, on behalf of BioVac, just thank you for allowing BioVac to come back and to update you on, we'll update you at the end in terms of where we are. But also, I was very happy that Pia from Afrigen was here because at least she could take the hard questions. So please, I'm done now. I don't, all the hard questions went to her. Also, a disclaimer that I think for those of you that weren't listening to Pia, I'm going to repeat some of the things in a very different way, but hopefully that can be good. For those that were listening, that will also hope to go to reinforce what she was saying. Okay, so my presentation is really just to take out a zoomed out view in terms of where we are with African vaccine manufacturing from our lens. So I'm not claiming that it's really a true representation because there are other people that are doing many work, from Morocco, Egypt, Algeria, Kenya, Nigeria, Rwanda. And so I obviously know a lot more about what's happening in South Africa, but I'll just try to give you a little bit of a sense in terms of where we are on the continent, but also some of the challenges that we are having, but equally some of the opportunities. Okay, so as we go in, I think many of you are very aware of this simplified value chain. Now, to people that are not in the space, it may be complex. If I have to explain it to my family members, I have to discuss each one of these pillars. But to many of you guys, you probably follow it and you can be able to explain it to the next person. What I think becomes a bit difficult, maybe for many of us that are, I think, in the room that are more on maybe the clinical side or more on the downstream side of the value chain, is that you have this slide that you can't even read. Okay? You can't even read because there's so many words that are there. And this does not even begin to reflect the complexity that actually goes. So in actual fact, if you ask many people, if you ask Afrigen, if you ask many manufacturers, Sanofi, whoever that you pick and say, okay, is this a good reflection of the vaccine development and manufacturing landscape? They'll say yes, but it's a simplified view. So if you thought that the previous one was a simplified view, within the industry, what you're seeing here, which I'm sure you can't even read, is in industry a simplified view. So what I'm trying to demonstrate is that, and many of us hear these headlines, vaccine manufacturing is complex and all of that, and we normally leave it at that, okay? But I think it's important that maybe as an industry, as we go through and we make this presentation year in, year out, is that we try and bring that complexity, not to intimidate, but to really understand when people are saying, but BioVac, what have you been doing for the past 3 years? Whoever, Afrigen, what have you been doing? some of these things I think we need to reflect back, particularly to audiences such as yourself, to say it's tough, it's difficult, it takes a while. Okay, so and I think it's a responsibility because a lot of what, as we know, happens is more on what's happening clinically and downstream, and the impact that those would have. But I think as the industry, as the vaccine manufacturing industry in Africa starts to get a little bit more mature, I think we also need to understand what happens upstream. So let me start firstly with some of the depressing things, and then we'll go more to more interesting things. And it's really not to, the aim is not to depress you, but just to bring a bit of reality to the conversation. So the complexity that you have, and again, my presentation is really with an African lens. Okay, so it's not with a Boston lens, it's not with a San Francisco lens, not with a Japanese lens. It's really with an African lens, which is what I think is quite important, because it's quite easy to say, okay, why don't we have this vaccine? Why don't you manufacture? Why are you taking so long? But I think it's really important that we go and do a deep dive. And we often are, there's this circle that a lot of us then get trapped in, where we do not have big investors, that can really throw money at venture capital and just really say, okay, I'm going to throw money at this and hope it will do well. Now, we know, the US is one of the biggest venture capital markets out there, where people put up money and, they put money into 50 companies. And if one makes it, the investor is happy, which means that 49, that they're actually willing to take 49. Now, for us, we don't even have a venture capital. Now you can imagine just asking for $50 million from somebody. It's just, it's quite challenging, which is why sometimes we then lean on to institutions like governments, which is also how our company got formed, to really try and take some of that risk. So we have low investor appetite, and also to get partners also becomes quite difficult. Now, and as you start, if you look at our facilities, so, some people come to our facilities and they say, oh, you have large facilities. Well, they are large in the African context, but they're actually quite very small in the global context. Because, we had to set up something. Now, again, with those limited facilities in terms of size, because this is about a high fixed scale game, your costs end up going high. Now, uptake is an important part, because who are you actually selling to? Are you selling to your own country? South Africa is a very interesting space, because 60 million population, about 1 million birth cohort. You would think maybe it's enough to sustain a vaccine manufacturer. Well, it's not, and I can write a book about it. So you really need to make sure that even your uptake and your that whoever's going to take your vaccine is quite diversified. And again, the visibility in terms of where you're going, is it low demand, is it high demand? So anyway, this is just an intro, but also just to reflect from a manufacturing perspective some of the dynamics that are there in terms of, and this is just, again, as a very simplified view, and I think Pierre alluded to some of the aspects that are here. Now, depending on the vaccine that you're going to do. Now, again, let's take an African lens. Now, we're not talking, we're not a Pfizer and a GSK, and I'm sure they're also in the room, who obviously have multiple facilities. They are probably in all platforms that they can be in. As African companies, we have to choose which platform. You've just heard from Afrigen. They are an mRNA player, which obviously they were hoping to go beyond that, because you can't be anything and everything on day one, And so are you going to go for live vaccines? Are you going to go viral vector, mRNA? What are you actually, and you must choose your lane. And your lane is not only about CAPEX, but it's also about human capability. Okay, because we can say, okay, I'm going to get a scientist from Africa, but those scientists, what is it that they can do? And some are better placed, let's say in the mRNA space, some of them are better in maybe the viral vector space. So there's a lot more in terms of just not only picking because you say, okay, I want to target this disease, but do you have the capability and the infrastructure? And capability, I'm talking about human capability and the infrastructure that will support that technology. Okay, because how you set up a viral vector facility may be different to how you set up an mRNA. So which lane do you actually choose as you want to set up vaccine manufacture? Just going on to my maybe a good manufacturing practice, I can talk for months on end about regulatory and good manufacturing practice, which is important because ultimately you want to make sure that products are safe, efficacious, and so that we don't have to worry about what's happening on the downstream side. A lot of what I think many of us in the room and virtually on the call would not be able to appreciate, and that I've learned to appreciate, is things that are, I often say that when you look at a car, I mean, we all know that there's an engine, but the engine is closed up. So when cars drive around, we often look at the tires and the windows and everything else that you can see on the optics. But we know that beneath that there's an engine. Now, in vaccine manufacturing, there's an engine that's really running these things. You have your validation systems, your quality systems, environmental monitoring, all the things that have to be kept alive, whether you're manufacturing one vaccine, whether you're manufacturing zero. Okay, your systems have to be on. It's not like you can switch off, unlike a car where, okay, I can switch it off and maybe I can start it up in the next 2 years, and I'm sure it will be able to start up, and I just need to inflate the tires and then I'm gone. No. In this process, whether you're manufacturing or not, those systems have to be alive because according to regulation, you can't, well, firstly, you can't switch off, it's just completely inefficient. But also the only way that you can monitor your system and show regulators that you have your system under control is when all of those things are all switched on. Your boilers are always switched on. You're always doing the maintenance, whether you're manufacturing 1 dose or whether you're manufacturing 100 million doses. And that then goes to the cost. Costs are quite high, and against that, this is what we need to say, what am I going to set up versus what am I going to sell? Okay, so what is the market demand? So when I cost and I'm saying I'm going to do $1 a dose, or I'm going to do this, and we often see all of these numbers. I mean, just picking on Greg's example about RSV, which in the middle income country they are suggesting is going to be $49, and I'm just picking a Pfizer vaccine, and that price is out in the open domain. In middle income country markets, it's $49. I mean, it's way north of that in high income countries. And then Gavi wants it at, I don't know what the price is, but normally Gavi likes, the $2 or $1 per dose. So if you look at that, I'm sure however Pfizer looks at in terms of market demand versus the dose and who's able to pay, they're obviously thinking about all the costs that they've also incurred in the development of RSV vaccine, which I think many of you know, and I'm not sure if it's part of the agenda, has come a long way. There have been others that have failed, like Novavax and all of that. And fortunately, GSK, Pfizer, and Sanofi on the MABs have been able to do it, but it was after many, many, many costs. Again, I won't go into infrastructure. This is just on the slide, but this is a very important part in terms of what you need to maintain. And then you have all the things around even the system quality control labs, drug substance, if you want to go into drug substance, which there are few and far between on the continent. And we have also full finish operation that also have to be maintained. So I won't belabor the point, and I'm very happy to take questions later on about some of this. And as I say, this is also a very simplified view of the engine, what's happening behind the scenes that we in this audience are not typically aware of. So again, going back to our industry on the continent in terms of prior to COVID-19, I think Pierre mentioned this also, that we are limited players on the continent, and drug substance, which is, you can also use API in that regard, is limited and still limited. Platforms now, I mean, we're very fortunate, and I'll talk to you about why we as BioVac welcome other players, because at least others can focus on mRNA like Afrigen is, because, as I say, you have to pick your lane. A long time ago, BioVac picked its lane, and that was the bacterial side, because we didn't, and then there's a whole host of reasons about why we picked that lane. But at least now that we have others like Afrigen that are picking, let's say, mRNA, and others are doing that. So in that way, platform diversity, hopefully it's going to come through. But at that time, there was limited platform diversity. Infrastructure was old. Even our facility now, we feel that it's quite old. And product development capability was extremely limited. Now, our continent, as we know, is a very reliant on donors, UNICEF, Gavi, and others. And we know that the likes of Gavi the principle is to get from the lowest price. And at the moment, India is one of those players that are the dominant players. For many of you that have been in the vaccine field for a long time, it was interesting. It was interesting reflection by Chanel from Africa CDC, who's been making this point in many forums, and I didn't think of it this way, but it's very on point. So there's doubt and skepticism about African vaccine manufacture. Whether are we able to do it? Do we have the capability? Do we have all of this? All the typical skeptics that have been there and will be there, and have been there in the past. And she made a very important point, because if you recall, in the late 90s, for many of you that recall, or you can go and have a look, there was a crisis in the UNICEF system in that a lot of players were withdrawing from supplying UNICEF because the market wasn't favorable. They had one-year tenders. So, other guys were saying, look, I'm going to supply directly. I don't need to supply UNICEF. Now, what that happened is that there was a crisis in terms of supply, because a lot of the countries that were dependent on UNICEF were not getting vaccines. That was at the same time that now Gavi came around the year 2000, that helped fix the market, and had certainty, and guys invested in it. Now, around the year 2000, and thereabout, I joined vaccines about 4 years later. So, but I still recall vividly in terms of that at the time, Indian vaccine manufacturers supplying the world was new. Okay, there was only one vaccine manufacturer, I think, that was getting into the game, and at that time it was multinationals. And whilst India was looked at in that way to say, okay, now that there's one player, maybe they're plugging the gap, but there was still people wouldn't imagine a world where in 2025 you can't think of vaccine supply without thinking of India. It's at the moment, it's completely unimaginable that you can have vaccine supply in the Gavi UNICEF system without India, because India are now the largest supplier. Okay, now this is 25 years later. And so what's the point I'm making as we go through this is that as Africans, there's skepticism about whether we are going to get there. Okay. And my wish and my dream, and what BioVac would also want to prove in its limited means, is that by 2040 and 2050, a world can also get to a point where we can't think of vaccine manufacturing on the continent without African vaccine suppliers. So I know India is in a different space, but I just want to make sure, that the supply was not there from 1960. It was only 25 years ago when the guys came, and now we can't imagine a world where there's Gavi supply without India. So just moving on, and I'm going to go into a little bit more detail, but Pia has touched on this, that I think many of us, and I think it's come through many of the forums, that we know that there's a target of 60% of vaccines manufactured locally by 2040. And this is where a lot of the skepticism come in that are just related to, oh no, this is just a dream of, AU, are you dreaming? Jean Cassia from CDC, are you dreaming around this? And again, I think it's very important that we aim towards this. Now, I often say, when people say, but how realistic is it, Morana? I mean, you guys know how difficult it is. How realistic is the 60%? I say, quite frankly, I don't even care whether it's 60%. Even if it's 30%, okay? I really don't care. Can we just move from the current 1% or less than 1%? Okay, can we move into something that's double digit? Now, whether it's 60, guys, we're not going to statistical analysis, about this, okay? Can we just at least get into double digit? Because you know what that will do with a double digit? It's like compound interest. When you start, it's difficult, okay? You don't see the needle moving, okay? But as the momentum comes through, as more players come through, as the market also embraces, you're going to get that momentum that at some point when you get to, let's say, be 10 to 15%, the acceleration towards 60 will be much higher. There'll be skills, there'll be infrastructure, money will be flowing within that system. It's going to be very hard for many of us to go from the current 1% to 10%. That's hard because nobody believes in it. We're trying to raise funding, and I can talk to you guys for 4 years. Okay, hopefully it's going to come through. For 4 years, we can write a book, BioVac can write a book about just about everything, about skills and we're just one player. So I'm saying that this ambition, I think we need to embrace it. Whatever there is that we're going to get to double digit, guys, it would have moved the nil so much. Anyway, that's just my point of view to whenever I talk to the skeptics. Again, there's a lot of initiatives I think that I've spoken about, and the mRNA Tech Transfer Hub has been highlighted. And why I think we need to be obsessed about it, and I think it needs to be an obsession, is that ultimately, on the continent, we do have the numbers. We really do have the numbers, okay? And in this game, you do need the numbers in terms of making vaccine manufacturing. Childhood vaccines are not going. I know we have an issue with zero-dose children. I know we have a lot of issues with coverage and all of that, okay? But this is the population where the market is growing. I'm not sure where adult vaccines are going to go, but the fact that there's momentum, there's talk on the continent. I mean, obviously in other markets, it's gaining a lot more momentum. But I'm also foreseeing a future where adult vaccines become a norm. Where they become acceptable. Now, I know it's very difficult. I mean, if you just look at flu vaccine coverage in South Africa, some people will say, oh, we'll never get there because adults don't believe in vaccines. But I think as momentum grows, I think there will be an opportunity. I think we've just heard about outbreak responses. And unfortunately, our continent obviously has a lot of outbreaks that you guys probably know a lot more than we do. And there is a potential to even export beyond the continent. And I'll give you just one example of how that is possible. And again, I think it's important, and maybe it may be reflected in many other forums, that we do need to reduce our dependencies. I think COVID-19 showed us, but also in general, just in terms of some of the supplies, in the supply chain, if you're manufacturing vaccines and let's say just one tube is not, or one ingredient or one excipient, the whole production stops. You can have everything, but so the dependence on imports is quite huge. And again, pandemic readiness, as we've heard, not only from CEPI but also from Afrigen and others, and workforce development, which is something I'm extremely passionate about African scientists, our pharmacists, our masters, our PhDs, all of those that are sitting in other continents, who have the potential that, let's firstly get them back, but those that are still in universities, let's not lose them. If they don't want to be academics or lose them to other industries. And ultimately, I think we know that our economies need to grow. So again, I won't go through this slide, which I think is similar to what I was saying about the potential that I think Africa has. If you look at the current state, you'll be very depressed about GDPs, about geopolitics and all of that. But I think if you take a 2050 lens and you think about what needs to happen, I'm bullish, and that's why we're doing what we're still doing, or else we would have closed shop a long time ago. Okay, so now let's move on to what's happening on the continent, and let's get into some of the specifics. I always say that when vaccine manufacturing or post-COVID, anybody and everybody wanted to manufacture vaccines on the continent. And many countries obviously reflected what their need is. And similar to Pia's slide, and that Africa CDC, CHI, and everybody else went physically to everybody that made announcements. So if I go back one slide, this was the view that people had. Okay, who wants to manage? It was just countries that were there. Now, what is important between this slide and the one that's coming is that remember that because of what happened in terms of the blockages that we had during COVID-19 pandemic, a lot of the donors, particularly sitting in high-income countries, felt bad, and yes, they ought to have felt bad. And now they said, okay, and together with our presidents that on the AU that says, you know what, we need your help as the EU and others, to help us set up vaccine manufacturers. Now you can imagine. This is a world that still today people look at Africa as one market. They can't, really Nigeria, South Africa, and Kenya. For them, it's all one thing. It's like, it's like in the US, you have states, okay? They can't pinpoint now what is the difference between a BioVac and a Vaxera and an Afrigen. So, and these are donors and countries that have to deploy funds, whether it's for political purpose or not, okay? Now, how do they deploy funding to all these announcements? Okay. Now, they only know Africa from safari and from, that's, and disease and poverty. But they then have to deploy funding that say, okay, guys, there's a fund that has come up. Where do we deploy it in Africa? Now, all these announcements that have come through post-2021, how is it going to happen? So this slide that actually that Africa CDC, Chai, and others did was to really try and understand, are there real announcements? Okay, and there are some that are not on the, and I'll make reference maybe to one which is in Botswana. There was an announcement in Botswana which didn't make it to the list because there were so many announcements that were made. Donors were very confused, and they couldn't make the wood for the trees. So this was an exercise to really understand who is really doing something, and obviously they segmented us, as Pia said. So yeah, so and this is, I think, is on the Africa CDC website or on one of the studies that are, but it's in the public domain. They also took a view and say by 2030, so as we were looking towards 2040 and the others, but who is likely to be supplying Gavi? Who will be entering the Gavi markets from all these announcements by 2030? And this is about a year or so old. And these are the countries that have been identified and the players. So within there, BioVac has its products. We have Aspen, and we also have IPD from Senegal, who is also listed there. I think this is probably going to get updated soon because I think other players that will make that will come to the fore. But this was as at about a year ago. So essentially, what it is, it's saying that these dreams and these plans have been validated, and there is hope and dreams that in the next 5 years, African manufacturers will be starting to supply Gavi, much like India is. This is what this slide aims to show. I'll go through this very quickly because I think other presentations probably have been made that all of this is not possible without coordination. Okay. Now, we are not going to be sitting here in Cape Town, and trying to do this on ourselves. Our friends in Ghana, Atlantic Biosciences and DAK, are not going to sit on their own. Vaccera in Egypt is not going to do. Bio in Morocco is not going to. We all need coordination, particularly when the industry is so young. So the Africa CDC and what was called the PAVM was a very important instrument to coordinate all of these functions so that even these donors can know that there's a coordinated way of going about it. I'm going to talk a little bit about Gavi. I think you guys may have probably received a presentation about Gavi. I'm just going to go into some detail in the next slide about why this instrument is so key to get us to a particular stage, but that is an instrument that has come through. And lastly, what we are now aiming for that, we're really pressing on Africa CDC to get going is pool procurement on the continent for our countries to also buy from us as African manufacturers. That pool procurement is important because these donors are getting tired and fatigued, as we all know. Okay, I mean, look at what's happened to USAID and all of these other things. Being dependent on donors, just as we've seen just this year, has had its calamity. So for the likes of Gavi and others that are supporting our industry now with some of the instruments, that's not going to go on forever. And, we can't also be dependent on UNICEF forever. So how about African countries buy from the continent so that is far more sustainable? And that's the part that we're hoping Africa CDC can coordinate in terms of the procurement mechanism. Colleagues, I won't go through the details of this slide because there's so much into it. I'm not sure if Gavi may have presented this, but I'm talking, maybe I'll present it. This is their slide, but I'm presenting it from a manufacturer's lens. So when it pertains to African vaccine manufacturers, a fund that's now $1.2 billion strong was put together by the donors to ignite and help African vaccine manufacturers. It's called AVMA, African Vaccine Manufacturers Accelerator. It was launched last year for a 10-year period. So we're now a year in and we have 9 years to go. Panic, and I'll tell you why that's it. So it was launched last year and they said, guys, you have 10 years to seize, to download this $1 billion. Now, can you imagine? Can you imagine $1 billion sitting there? And we as African manufacturers are looking at it and you're trying to download it. They say, no, there's a criteria to download this $1 billion. The criteria is you as African manufacturers need to have vaccines that are on the UNICEF list that Gavi prioritizes, and you must get pre-qualification. okay? And you must compete. Then you will be able to download it. Now it's tricky because when you see this, you say, oh my word, and it's only for 10 years, and the clock has already started. So it's called AVMA. They have priority vaccines that are listed there, and there are certain milestones that you can get if you get pre-qualification or if you supply, and I won't go through that mechanism. It's a very important instrument, this, colleagues, because, and I can speak from a BioVac perspective. Yeah, I said earlier that we have, our infrastructure is fairly modest and it's aging slowly. We need to expand and we need larger facilities, and because of scale, and we need to be modern and to be able to compete with the world. We've been at 4 years finding difficulty in raising funding. But now that there's an instrument, okay, now BioVac can't yet touch it, okay, but it's still there, okay? There's an instrument. The lenders that are coming to, that we are talking to now say, oh, okay, BioVac, what products are you doing? Where are you going to sell them? How are you going to pay me back? This instrument is very key for us because it has allowed the lenders to say, okay, now I can talk to you. As I said to you, there's been 4 years trying to raise funding, okay? 3 billion Rand or $150 million that we've been trying to raise for the past 4 years. We are quietly confident that we are almost there. But for some of these instruments from an African player, it would not have been possible. $150 million everywhere else in the world is small change. It is small change. It's actually ridiculous when people, oh, you're only trying to raise $150 million. You know, when you talk to our colleagues elsewhere in Europe, only $150 million, why can't you raise it? You know, these guys raise it on a concept. You know, we are trying for 4 years to try and raise it. So it's these instruments that will help us try to, I'm just talking about the difficulty in terms of being on our continent, but how we are going to, or how we are hoping to do things. I'm going to rush through the African slides quickly, but again, this slide just shows that we are in an ecosystem and that we cannot do it alone. And we need all of the support that's needed there. And these are just, the logos that are here are not even, a full representation of the various parties and the organizations that are having an interest in this topic of vaccine manufacturing on the continent. Some are helping with skills, some are helping with technology, some are helping with grant funding, some are helping with lending, and yeah, there's so many of them. My last section is just going to hone in quickly into South Africa. Again, I'll remind you just of this very complex slide that I said is complex. So I'm using these slides quite often these days because it reminds me of when I joined the industry in terms of what has been happening in the country. Well, there's been some basic research, and universities have been either trying to do this. At the time that I joined, in actual fact, there was what was it called, SAVI, South African AIDS Vaccine Initiative, I think. That was the buzz in terms of doing some research in HIV vaccine. So there was some basic research. Development was not there. And now I hope we understand there's a difference between research and development, okay? Development is really now taking it into reality. There's no theory and all of that. We're now trying to hone into something that is really applicable to take it forward. Now, clinical research, South Africa, I think you guys know better. We've been quite good at clinical research. There's been zero drug substance manufacture historically. And formulation and filling, it's been there. Some of our companies have been doing that. But a lot of where we've been supplying products has been in South Africa, okay? The export has not been, historically, hasn't been there. And this is sort of like the picture that we've had. So maybe I'll just open these quickly just for the sake of time. And I hope I'm not doing an injustice to any of the companies in South Africa. So let me just, you saw the previous slide was a little bit empty, but you can see that what I'm hopeful and very excited about is that we're starting to slowly populate the empty gaps, okay? We're very happy that we're having our colleagues from Afrigen who are 20 kilometers from us, where we are part of the ecosystem, that we have UCT, Stellenbosch, UWC, CPUT within a particular geography. And that helps, firstly, in terms of getting some of the skills, the type of work that Afrigen is doing with some of the universities, some of the things that we are doing, because that ecosystem is quite important. And I'm only talking about in one city. in the country. And as you can see now, this is only representative of companies that are there. And maybe Afrigen, I should put you into the research. Maybe I've done you guys a bit of an injustice. But in terms of plugging the gap, I think both us and Afrigen are plugging this gap that was historically not there in terms of development of products. We are also getting into drug substance, okay? They are far further with mRNA. We are early in mRNA, but we are quite strong in the bacterial drug substance, and we have a candidate, an oral cholera vaccine candidate that I'm very excited, and I'll tell you a little bit about the updates on that one. On formulation and filling, we also have Aspen colleagues that are also have entered the fray. And now on imported products, I think many of you know there's MSD, Cipla, GSK, and others that are importing products and selling in the private market. And soon, I think South Africa will be starting to talk about exports. And again, I'm using this the South African picture, but I'm hoping that, you know, for colleagues that are not based in South Africa, that when you go to your own countries, if you go to Ghana and you look at the activities that the guys are doing there, that you can also start populating your slide and say, 5 years ago, it was like this, and now it's good. So this is the hope that I'm hoping to give you, that out of all the doom and gloom, there is activity. Is it easy? No, it's not. But a lot of us are trying to plug the gap so that there's end-to-end vaccine manufacturing on the continent. I'll also touch on, you know, not only are we an importer of technology, okay, which we are. So we are an importer of technology, and I'll use BioVac as an example. We work with many other parties where we get the technology from others. Aspen as well has struck deals with Serum Institute of India in terms of getting in tech transfer. I think what is really great is that within the country, we even have 2 entities that are doing tech transfers within the country, that we're not only dependent on external. And I think Afrigen have just demonstrated that with the previous project. So we are a recipient of the technology that Afrigen has developed. So within the country, there is that activity that hopefully can be a lot more. But also, colleagues, I think it's important to recognize that we are also can be an expert. We're not only a consumer of technology. We have the potential to export. Let me say not the potential. We have exported technology, okay? Afrigen has spoken, already exported to 15 countries of the mRNA technology. So already the technology has gone out. BioVac developed Haemophilus influenza B many, many years ago, and we have outright license to 3 companies. And this is not in just poor countries. We have exported to a company in Japan that's starting their Acilla pertussis production quite soon. That HIP technology was actually coming from BioVac. We've done to Thailand, Indonesia, and there's a US company that's also interested in our HIP technology. And this is South African companies that have been able to export. So there is hope. I know I'm running out of time and I'm about to end. This also is, and I'm happy that Shingai, and I hope she's still on the call. And again, I've just made 3 entities. There are many other entities that are working with African vaccine manufacturers. But I've just picked 3, and I've just picked the companies on the continent that we're working with. Gates Foundation, IVI, and CEPI are, as you know, credible global organizations that are working with everybody in the world, okay? They are also working with us as African companies. So what I'm saying is that we are just not sitting and doing some technology of 1965 and nobody's actually supporting us. So we have the best vaccine organizations that are working with many of us, and you'll see the arrows going this way and that way. We have some interaction. I think you got that from Shingai, from Afrigen, and now from myself. I won't go through because I've really run out of time, but just want to, if some of people will see some of the headlines next week from BioVac, I'm very, very excited. I'm very, very excited. And Bev, I don't know how many subjects. The last time I heard was it 9? Was it more now? OCV subjects. Oh, over 20. Okay. I was just checking. Okay, so let me tell you guys what's happening. This is so exciting. You know, whenever I get home, my children say, I don't know what Daddy's excited about. Anyway, so we've been developing an oral cholera vaccine that we got the technology from IVI. Now, in South Africa, people are saying, but why are you doing cholera? Okay. When you go to others, say, oh, yes, please, can we please have a cholera vaccine? Because we know that it's quite endemic in many of our African countries. And there are often outbreaks, you know, if you, I won't go through the cholera stats, but I mean, cholera is actually endemic. Now, South Africa has had a few outbreaks, but the product was actually not necessarily intended for South Africa. Firstly, we need to diversify. We need to go to others. And also, while South Africa may want the vaccine, but it's a lot more needed elsewhere. So we got technology from IVR, and we've been doing the product development in our laboratories. We did our first GMP drug substance approval from SAPRA, which we got, which was a milestone, you know, the first to get. And I'm very happy, which is why I was just checking with Bev. Clinical trial, which is the first that BioVac is embarking on with its own product, started last week. And we now have 20 subjects from what I gathered and going that have been enrolled. Now, why are we excited? Now, if I go to Europe and others say, okay, why are you excited? Guys, this is in an take an African lens, okay, about developing a product, upscaling it, getting GMP drug substance for the first time, getting into clinical trials of a locally developed vaccine. Hopefully everything will go well. And this is the end-to-end that I'm talking about, you know. And whilst we're looking at nice technologies, we're looking at future, we're looking at therapeutics, we're looking at all of this. Can we just get some of these things in line? Can we plug all of these gaps? And so our cholera vaccine is just one of those. And some of the other products that we're doing from a full finish perspective are listed there that I won't go through. Again, the ecosystem that I spoke about, just in terms of South Africa, I think we're very fortunate. I made a mistake. I always talk about it. So I was talking to some of the lenders, trying to charm them when we were in May, we had a hectic due diligence. And I put up a slide like this, and I said, hey, you know what? South Africa does not have much and all of those things, you know. Now, my reference when I was saying that was because, you know, if you look at the US, you look at in terms of the ecosystem. And then this lender, and this is a finance guy, says, no, Mrana, wait, why are you saying South Africa doesn't have much? And I said, no, if you look at what's happening in other hubs, in terms of biotech hubs, you know, we just have a few entities that are trying to do this, from universities to this, to everybody else. He says, well, in an African context, at least you have a lot. You know, so I've now rephrased and said, you know what, in actual fact, it's like that glass half full versus the glass half empty. And I think I'm also rephrasing some of the, you know, my messages that I think we need to embrace that which we have, you know, and make sure that it's a lot more. And I'm hoping that other countries would also do that. Greg, sorry, I probably went way over time, but yeah, happy to take any questions.
Not a problem, Morena. We love your enthusiasm for indigenous vaccine development on the continent. So thanks very much for a wonderful presentation. I like your realistic approach. You know, I always tell people, you know, this concept of manufacturing 60% of Africa's vaccines by the year 2040. I often wonder what were they, when they were sitting on the table, what were they smoking? But I like your approach, you know, at least go for double digits from 1 to 11.
Yeah.
Okay, floor's open to Morena first, and then Shingai, you're still on the line. Shingai, wakey. She's still there.
I'm here, Greg. I'm here.
Okay, all right. But let's just take some questions to Morena first, and then we can open it to you and to Pia as well. So we'll start here.
Hi, thanks for the awesome presentation. I just want to ask you to speak to, you briefly touched on IP sharing and patency and how, you know, between, you've touched on in an African context, but between the global North and Africa and how that process, you know, is started, are there mediators? And then what's the financial implication for all parties involved, and then for the end user or the country that's, you know, procuring that product? I have one more question.
She's got another question. We'll come back to you later on.
Okay. Yeah, so very, very interesting topic. And we try and shy away from, if we can, you know, around IP issues, because firstly, they are so complex and so expensive to navigate. And why, you know, we, as BioVac, we don't get into research. First is because it's expensive. in general in terms of just doing the research, but also you then, get into these IP issues, and whose territory are you in and all of those things. So when it comes to development, and the type of things that we choose, it's often firstly where we have the capability, but also where there are not much IP issues. And so we've been very fortunate with a few projects that we've done that we've never had to. Now we do, IP landscape and all of that, but a lot of the things, like if you look at the cholera, if I just use cholera as an example, the OCV technology, it's been technology that's been there. It's a wholesale vaccine. It needs to be purified. This one is just a simplified. There are no particular IP issues because we want to move the needle that much. That doesn't mean that IP is not important, but I always say that in our experience, know-how. Okay, know-how is far more important. I'm happy to have somebody give me the IP, give me the paper and all of that. But unfortunately, in biologicals in particular, I know it's a general statement for pharmaceuticals. In biologicals in specific, that IP is worth nothing if you don't have the know-how to take the product through. I often say too much. I think we must write a book even about an OCV process which looks like simple. The things that you have to undergo that even the technology transfer partner did not, we got the technology from IVI. IVI only did the product as I think 2.5 liter scale of not more than 5 liter scale. The rest of what BioVac did to take it to 20 liters to 200 liters, we don't know. Nobody gave us that. And you think, okay, it's the same technology, you're just taking it up. Oh my word, I can write about the drama, the real drama that we actually underwent around. And that's know-how. That's know-how. And so if you have to, obviously one can't necessarily pick. I'm not downplaying IP, but I'm just saying let's look at IP in the context also of know-how.
Jason?
Yeah, thank you very much, Maureen. I think very, very inspiring presentation. I have 2 quick questions. One is, you mentioned the Indian manufacturers and the success within a very short time that made them a key player. What are the key lessons or drivers that we can learn from them or that we are using and other vaccine manufacturers in Africa can actually use to accelerate the vaccine manufacturing in the continent? That's one. The second one is the vaccine manufacturing generates a lot of medical waste. How do you deal with the waste containment to ensure that there is no environmental contamination? Thank you.
Okay.
How do we?
Manage the waste,
okay.
the medical waste from...
Just before you respond, Morena, people may not know, Morena alluded to it. Currently India supplies 60% of the world's vaccines, and they export vaccines to over 100 countries. And it's a related question to what Jason was asking you. Are we going to be able to, given the volumes that they're generating, and the new vaccines, pneumococcal is a good example. Poor Pfizer, and we can mention it because Pfizer's lost Prevnar 13 to Gavi. For South Africa alone, it was costing us about 300 Rand or 400 Rand a dose. With the Indian vaccine, it's like 50 to 80 Rand a dose. So how are we going to compete with them? But first, answer Jason's question.
Very pointed question, and I think BioVac can really answer this question confidently because of the pains that we have undergone. I think, and I won't go through BioVac's history at length because then we'll be spending the whole day here. But BioVac really started out of the South African government having a vision to set up vaccine manufacturing in South Africa. If it wasn't for government initiating that, I wouldn't be standing here. I think we need to acknowledge government's role in trying to see that. Now, in that, what they did is that they set up a contract, an exclusive contract, so that we can be able to import vaccines and sell them so that we can generate some revenue. Now, this is just out of importing and selling. You're not going to make much, but you're starting to generate some revenue so that you can go and raise funding independently. Okay. Now, again, I always say it was the blind leading the blind because our contract was short and then we had to extend it and extend it. Anyway, and so there's a book to write about it, but I think we have to acknowledge the role of governments. Now, I know we don't like politics, but we have to acknowledge that, particularly in vaccines which are public health, we can't divorce ourselves from governments in general. So, our African countries really need to, governments really need to come to the party in terms of enabling an environment. And I think we need to take lessons, let's say, from how South Africa did it, which is not all positive. And whenever people come, you know, Kenya, we had a visit from Kenyan colleagues and our Ghanaian colleague. We got a minister from Ghanaian Minister of Health. And I said, we did it this way, but can you do it better? You know, and which is, so I think governments are important. The market instruments that Gavi is coming up is quite important. But I think as African manufacturers, we have to recognize that we only have a finite period. I often say to my colleagues internally and elsewhere that I don't want to come and sit in 2035 and then people say, what have you guys all done when you had an opportunity of these instruments? So we really need to put our best foot forward. And as much as we can criticize some of these instruments that are there, and there's a lot of criticism about them, we have to seize the moments. Those are instruments that we have now. You know, so in general, I think that's it. Well, when it comes to asking about environmental containment within the facility, I think it was your second question related to the controls that normally one has. Okay, we can also write a book about it, which is quite important. You know, running sterile vaccine manufacture is a critical part, and if anything goes wrong within that, as far as environmental monitoring is concerned, you can lose a lot of money. Again, 2021. We were manufacturing, and then we got contamination in that we picked up only after production. Fortunately, the product was not yet released, and we have limited means. Anyway, long story short, we lost 200 million rand in that year. Now, you must remember, we're trying to raise funding, and we're trying to show that our books are okay. On one hand, then you go to funders, say you're looking for money. On another, you say, you know what, just lost 200 million rand. Out of what? Out of contamination. Oh, these Africans don't know what they're doing. Anyway, it's a very important topic that I don't have time to go through, but I just want to say that all the instruments, as I say, there's so many lessons that we can talk about. Just that one topic in itself has its own chapter, but it's a very important one.
You need to take early retirement to write all those books, Verena. There's a question from online.
Yes, but I just wanted to amplify on Jason's question. How do you deal with the wastage? How do you plan or intend to deal with the medical waste, the vials, the needles, the what have yous that will be generated from manufacturing? Thank you.
I mean, around that, why do we have 300 at the moment? Because we lost the PCV contract and all of that. BioVac is actually manufacturing one vaccine as we sit now, which is not a healthy state to be in. But we have 350 employees, okay? 40 of which are in our science and innovation. Everybody else is in the, you can call it manufacturing. Why do we have so many people? A lot of it is to mitigate against risk. The supply chain, obviously making sure that the facilities are okay, making sure that everything is under control. That, as I said earlier, whether you're doing 0 or you're doing 100 million, you still need all of those people. And those people are there to mitigate risk. So on one hand, you need the right people that need to be there. Secondly, automating and digitizing your systems, which in itself is expensive, is absolutely necessary because human errors will be there. You know, so that's another way that you need to mitigate, but it's expensive. We've just got a taste of what AI may potentially help in reducing and being able to have predictive, as far as environmental monitoring is concerned. We had 2 of our colleagues go to Seattle for what was a pilot, and we actually took our data, our environmental data in BioVac that fortunately was already digitized. If it was manual, I think it would have been a problem. It was already digitized, and they put it into an AI model. Now, I'm not saying that, and they put that in terms of predictiveness of all of that. Now, you know, I'm also a bit of a skeptic, in terms of AI, because I just use ChatGPT just to ask stupid questions. But, you know, when my 2 colleagues came back and they really explained it, I said, okay, now this thing can really be powerful. Now, again, it's going to be expensive. Who does it? Who's the validated system and all that? We still have a few years to answer that. But I'm just saying between human, automation, digitalization, and hopefully what would be AI in the future can reduce some of those risks.
Okay, I'm going to ask you to be a little bit briefer because I think there's still quite a few questions. There's some from online.
Yeah. So we have a question from Roger. So he's saying during the COVID-19 pandemic, many new health and vaccine projects emerged. with great ambition and strong rhetoric. So he's saying he's curious, how well are these projects actually performing in terms of reaching their milestones and achieving their stated goals? That's the first question. He also has another question. How many are truly on track to deliver on time? And in practical terms, how can they remain competitive against established giants like the Serum Institute of India? And he's just wondering whether we are seeing a pattern similar to that of some African leaders that give powerful speeches and bold commitments, but have limited delivery on the ground. And he just has a final comment, I think, on environmental sustainability, and I think you answered that with Elizabeth's question.
Okay, to be brief and not five books.
Yeah, so I think some of the, in terms of where are we post COVID? I think commercial COVID-19 vaccine projects are not there. Commercial COVID-19 projects. Now we're still doing R&D and we're still using that SARS-CoV-2, to validate some of the platforms and all of that. So those are still active. But in terms of commercial COVID-19 vaccines, there's none that I know of. The second, I think, the question that I think we must monitor is some of the pictures that myself and Pia showed about where the status of the vaccine manufacturers are. I think that's where the answer would lie. Let's follow Africa CDC, CHI, and others. Hopefully, they will update those slides as we go through as to how quickly or not are we all making our way towards realizing all of these dreams from all the speeches, as was said. But I can only answer it from our lens, but I think some of the pictures that we've shown, like any study that needs to be continuous, I'm hoping that they can update next year and they can tell all of us in terms of where some of our facilities are concerned. The last aspect about environmental, as far as facilities are concerned, is a very important thing. New manufacturing facilities need to be a very environmental friendly. Lenders, everybody's looking at that, and I think it's a responsibility that we all need to have.
Morena has a question, the side, a quick question.
Actually, two. So thank you both for the very interesting presentations. What is being done to make sure that, for example, the AVMA takes into consideration what is being done as part of the mRNA technology transfer program, first? And second, is there anything you are aware of that the African Union, Africa CDC, and Gavi are putting into place to protect the African continent market because of the emerging manufacturing capacity that cannot compete for the moment with the global market? Thank you.
Thank you. Just a quick one. Yeah, so, and unfortunately, I went through the AVMA slides quite quickly, but mRNA as a platform has been recognized through this AVMA instrument. And in actual fact, if you come up with a vaccine candidate using mRNA, your incentive is even higher, compared to the other platforms. So it is recognized already by Gavi and AVMA in terms of assisting us in that regard. I think your second question is quite important. Gavi 6.0, and I hope they spoke about it, has come through, and the budget constraints. Now, we as vaccine manufacturers are concerned because AVMA is not, does this $1 billion, are we at threat of this $1 billion reducing? I know that a lot of current conversations that are happening at Gavi and UNICEF level about not affecting that, yet balancing some of the instruments. So I know all of the role players are having active conversations around your question.
Pia, do you have any comments to that question? No. Okay, we have another
we have one
question
last
online.
Yeah, we have one last question from Abdul. So he's saying he's curious about the regulatory readiness side of all of this. So his question is, our what's going on?
NRA.
National regulatory agencies strong enough to support vaccine production that can meet WHO pre-qualification standards. And he just mentions that right now there's only 9 ML3 compliant agencies, he's saying, including Ethiopia. So he says that's another bottleneck that we need to address head on.
So Shingai, do you want to answer that? Because in your presentation, you briefly alluded to how CEPI is supporting the African regulatory forums.
Yeah, so I think whether it's Afrigen or BioVac or any other manufacturer, whether it's old technology or new technology, you're not going to do anything without the regulators. And so a lot of the times when we talk about building capacity, that capacity is not just of the actual manufacturer of BioVac and its staff. It's about the ecosystem around it. So the regulators and the examples are given by Pierre about growing together with SAPRA is basically what needs to happen. So CEPI is working alongside many of the national regulators, but also the regional regulatory agencies to support the development of regulatory science alongside the development of the vaccinology sciences. If you don't do these things together, you won't be able to bring things forward. And particularly in our case, where we're interested in funding in an emergency, that science is even more complicated, and the regulator must be comfortable, have adequate data to make that decision for emergency use licensure. So yeah, the African Medicines Agency is absolutely critical, and it's very good. It's getting started, and we should be able to see it leading even more from the front in terms of showing what the coordinated, harmonized approach can deliver.
Okay, quick comment from you, Morena. Anything?
Yeah, absolutely relevant question that I didn't touch on in my presentation. We can have all the market instruments, we can have all the lenders coming, we can have Gavi and everybody supporting. But remember when I was talking about this $1 billion that needs to be downloaded, one of the key criteria is you must get pre-qualification. In order to get pre-qualification, you must also have satisfied your own regulatory authority, which should have been ML3 for vaccines. So regulatory is actually going to be a very big topic, or if it isn't already, it can only be a bigger topic because manufacturers will be saying, I need you to help me to get to that point. Developers will be saying, I need you to be able to look at my facilities. I mean, one of the issues that we are battling, I think, between us and Afrigen is not only the big facilities. How do regulators look at pilot scale facilities and apply a lens of a pilot and not apply a lens of a big, large manufacturers? So there's still a lot of understanding and learning that our regulators still need to do, but they also have to learn with real life examples, which is why we need to be there, because all they'll be learning is theoretical examples. So they need, I'd like to believe that they need us as much as we need them, because the only way that they can get better is if they can come and work with us.
Thanks, Marina.
Thank you so much for the two presenters. I'm so excited to know about the OCV vaccine from your side. And I think Greg asked the question, is the vial, the cost? How can you make, ensure affordability for low and middle class African countries while sustaining your manufacturing margin and all these things? And also if you can maybe elaborate on how much you are costing your vial or the cost for per dose, knowing that India maybe have the lowest. Yeah, thank you.
Very important question. Cost has been a big issue that we've also been scrutinizing. Part of our, and I can only speak from a BioVac perspective, part of the reason why we need bigger facilities, not to have shiny new paint and new equipment, is for scale. Our current capability won't give us the scale and the cost competitiveness that we need. We need scale to be able to do that. So, and that's what India has. Now, our facilities will be modest in relation to India, but a lot of the costing that we've done to say, at that scale, can we be competitive? We are marginally competitive. This instrument from Gavi actually is geared towards that. It recognizes that African manufacturers are not going to be competitive. That's the first, that's the basis of this instrument. It's a African manufacturers are not going to be competitive. Let's give them a window of 10 years where we can give them these incentives, where it can get them to a competitive state. But after 2034, African manufacturers, you must be in the real world. And so that's why I'm saying that we need to leverage that, but we are not competitive as you said today. In future, we need to make sure that we get to that, but we can be with scale,
Okay, Shingai, we have to end, but I've got one last question to you. You were talking you were saying in your presentation in the context of the 100 days that one of the critical uh, uh, issues is having those early warning systems, and central to those early warning systems is good surveillance in respect to outbreaks and infectious diseases generally. So what is CEPI doing in terms of developing capacity on the African continent, recognizing that in many countries, surveillance is virtually zero, except for a few sentinel diseases like polio and measles?
Thanks, Greg. So my comment linked to what Morena was saying is that I think the debate or the argument regarding a vaccine manufacturing goes beyond the science and the economics. There's an element of the global health ecosystem and the big debate now about whether that ecosystem is fair. And so I think it's just important to think of that, because there definitely will be a cost to achieve what we want to do. But I think the point is that African countries are saying that they want to do this and they need the investment to be able to achieve this. So just that point to keep in mind. With regards to surveillance, Greg, CEPI can't do what it does without good surveillance. However, CEPI can't invest in everything. So I think the best answer I can give you is there's partnerships with different institutions, particularly with WHO, who has a good machinery that is trying to build up this capacity. In some projects, there are elements of surveillance that are built in as part of what CEPI funds, but broadly, we don't directly fund surveillance as CEPI at the moment. In the whole ecosystem approach is absolutely critical for CEPI's work. Unfortunately, the mandate we have now doesn't give us that ability to fund it directly, but it's absolutely critical and we can't do our work without it. Thanks.
All right, thanks very much, Shingai. I think we need to close the session. Pia and Morena are still here for, hopefully they'll stay for tea. And if anybody in the room would like to tackle them, then please do tackle them. And Shingai, thanks very much for your participation online, and enjoy Tokyo. So let's have some coffee tea and come back at 11. Thanks very much. Are you doing? Well, I think a big do on Thursday. Oh, you don't have to tomorrow. That's tomorrow. We'll come one day. Okay. As a train.
Okay, everyone, can we start moving inside? Okay, everyone, good morning. I'm going to welcome you to start coming back from tea, please, so that we can start the next in our morning sessions, which is a really vital part of this module. I'll just wait for some more people to drift in and check up on them. Okay, I think let's start and hope that folk will keep drifting in. So it gives me great pleasure to welcome you to the next in our morning sessions. I know this is hump day, but I promise this is going to be a fantastic session to help get us over that hump. So first up, we have Professor Tom Scriber from the South African TB Vaccine Initiative. He's the Director of Immunology at SATV, and he's going to start us off trying to link the next session together. Where are we sitting with HIV, TB vaccine progress and initiatives, and how do we actually get this into the stages of getting it to our patients? Thank you and welcome.
Thanks very much. Thanks for the invitation. Always great to be here, and also nice to see so many friendly faces. Yes. Yeah, so I work at the South African TB Vaccine Initiative. We've been engaged in all aspects of the TB vaccine development pipeline. And so I thought today what I'd do is kind of give you a snapshot of why it's important, what's sort of happening, what is some of the science that underlies vaccine development, and talk a little bit about the new vaccines that are coming through. So, yeah. So the first thing I think is, why is it important? And I think everyone should know about this, but TB is a devastating disease in humans. It's been for us for so long that the estimates are that in the course of history, it's probably the biggest killer of humans from a single infectious agent, with more than 1 billion people that may have died from TB to date. And that's because it's been around for thousands of years, but also because it's still killing a lot of people now, and it's obviously a major issue. And it's not only deaths, it's also disability, it's economic hardship, it affects families, and it's certainly a very important issue that we need to tackle, especially in the African continent, where we certainly don't have this infection under control. So that is underlined by the graphic here. This is the WHO estimate of the number of TB cases every year over the last, I guess, 15 years. So the blue line is the estimate that comes out every year, and you can see that more than 10 million cases are diagnosed every year. You can see that it came down a little bit, insufficiently, but it was coming down. But then during COVID, we took a knock, and so it's actually going up, unfortunately, for a number of different reasons. This decrease is actually largely attributable to the line at the bottom, the sort of, I don't know what color that is, the light blue line, which is TB diagnosed in people with HIV. And it's largely because HIV care has substantially improved in the last few decades. And so non-HIV associated TB has actually come down much less than that. And so there's a lot of work needed to get this under control. Of course, this is a vaccinology course, and the question has to be asked, what would a TB vaccine that is effective be able to do? What would the impact be? In fact, I was given the title that specifically speaks to this. So here it is. So there have been a number of recent efforts to try and understand what would the potential impact be. These have been large modeling studies done with very detailed information about multiple countries. And if we had a TB vaccine today that had a 50% efficacy for adolescents and adults in preventing TB disease, then it could prevent 37 million cases and 4.6 million deaths in 25 years. It would be cost effective, so not only cost saving, but cost effective, in fact, cost saving in actually most countries in Africa. And so the economic case for this is a no-brainer. It makes absolute sense that we need a vaccine. We need something to prevent people from getting TB in the first place, while we also find those who have the disease, treat them, follow up people who may have been exposed, provide preventive therapy, and all those other tools. But we really do need to prevent the disease as well. So the case is clear. And so I'm very happy to be working in this on this kind of problem, because there's clearly a good case for it. The history of vaccine development in TB is also a long one. So we've been, as a human population, we've been battling it out with TB for thousands of years. We don't know exactly how long, but it's definitely many thousands of years. There are very old, mummies where there've been evidence that people have had TB. We also know from DNA evidence and evolutionary analysis that the pathogen has been with us for a very long time. But of course, the organism was discovered only in 1882. That's when we were sure that this was actually a bacterium that causes the disease rather than bad air, or there were all kinds of interesting hypotheses that people were throwing around. And actually, pretty soon after that, the development of a vaccine started by culturing Mycobacterium bovis, so the organism that causes TB in cows and in animals, was passaged, and so a live vaccine was developed, which we call BCG. And BCG was actually first given to an infant in 1921 already. There were no trials done in those days. This was before it was a good, people didn't have, there was no kind of structure and no policy, and, it just wasn't done. You would kind of test it out, it was much more, much less rigorous. And in fact, by the time that, by 1924 to 1928, more than 100,000 infants had already been vaccinated with BCG, and there was anecdotal evidence, or pretty reasonable evidence, that it looked like it was making a difference. But the first organized trials of BCG were actually only in the 30s. So after many, many people had already received BCG. And there was a major MRC trial done in the UK in the 50s, which showed very good efficacy, and in fact, very long-lasting efficacy. It's still one of the few studies that has shown extremely long-lasting efficacy of BCG into adulthood. But by 1968, a trial in India, the Chingleput trial, showed that they saw virtually no efficacy at all. And so it raised many questions. And I'll show you in a couple of slides the actual, when we look at this as a sort of an overview, that there are issues. The genome of Mycobacterium tuberculosis was actually only sequenced in 1998. So before that, we didn't actually know the full structure of the genome. We didn't know how many potential proteins or antigens there are. And of course, it's a very complex organism. It's got incredibly powerful evasion strategies, survival strategies, adaptation strategies, which have only really been possible to discover once the genome had been sequenced. And of course, now there are efforts underway to develop new TB vaccines because BCG is not sufficient. And I'll get a bit more into that. So I think we've already heard about COVID. I just wanted to contrast the story, right? COVID-19, it took, we sequenced the organism, or the organism was sequenced in January 2020. And by March of the same year, clinical trials were underway. It's remarkable how quickly that was. So quite a different storyline for these two organisms. And I think we have to acknowledge and recognize that modern medicine does enable us to move much faster. It is just much quicker. But even if we discovered TB today, we would not be able to develop a vaccine as fast as we did for COVID for many reasons, including that it takes up to 6 weeks to grow the organism in the lab. E. coli, you can grow overnight, and you've got colonies, and you can count how many bacteria you had in your inoculum. You can't do that for TB at all. Who are the PhD students who've done their project on TB? You cannot do a quick PhD on TB. Can't. It takes too long to grow the organism. What do we know about natural immunity against TB? So, this concept was asked a lot in the last few years, and under the COVID pandemic, people asked, well, isn't there natural immunity to COVID? Why should I be vaccinated? And so we can ask the same question for TB. Is there evidence that the immune response, or the body mounts an immune response to TB infection that is actually protective against TB? And I think most people will say no. We know that someone who's had TB disease before is more susceptible to get it again, and that is true. However, there's very good evidence that infection with the organism in those in whom it doesn't progress to disease is protective. So I'll show you 2 examples. This first one is from monkeys. Monkeys are a pretty reasonable model for TB. It's difficult to do the experiments, very expensive, etc. But they are quite similar to humans in some ways, and so it's quite a good model. And so what the investigators here did is they infected a group of monkeys with TB by aerosol or bronchoscope, so they put it into the airways. Those monkeys did not develop disease very quickly, and so then they introduced TB again a few months later, a second infection, which was actually the same exact bacterium, the same strain, but they were labeled, so they could tell the difference between the one and the other. And they compared that then to other monkeys that got the infection at the same time as those who had already been infected before. And the result is on the slide. So in blue, these are the colony-forming units, the amount of bacteria that you can find in every lesion, every granuloma. TB causes granulomas or lesions in the lungs that have pathology. And so they could identify those, and they dug them out. They killed the monkeys, dug them out, and counted how many organisms are there in every lesion. And in monkeys that were infected for the first time, there were lots of lesions, and they had enormous numbers of bacteria in them. Thousands, up to 1 million bacteria for every lesion. And then there were a few lesions where they couldn't find anything, sterile lesions in blue over there. But in the monkeys that had already been infected some time ago, when they gave the same inoculum, the same infection, most of the lesions were completely sterile, and there were only a handful, I think 6 lesions or 5, where they even found bacteria from the new infection, and those bacterial counts were super low. So the monkeys were protected against the new infection. So there is an immune response that is protective, and that's exactly what we're trying to do with a vaccine, without the risk of the pathogen causing disease, right? The other piece of evidence for this comes from very old studies and these are epidemiological studies that compared those who already have evidence of prior infection versus those who don't. So for example, skin test positive people who have an immune response to TB, indicating prior infection, compared to those who don't have that. So for example, in the 1950s, nursing students graduate from university and they enter their workplace in a TB hospital. There were no drugs available, so the patients were infectious. The nursing students start to work there and they are heavily exposed to TB. So very high chance that they become infected. Those who had had an infection in the past, they were skin test positive, had a much lower risk of developing TB because of occupational exposure than those who had never seen TB before because they were TST negative. So again, and it was actually 79% protective. So infection comes with risk of developing disease, but remember that most people who become infected do not develop TB. And once they are infected, they have immune responses that are pretty good at doing this. So these are two examples that we can, as humans, mount very, very good protective responses against TB. But in some, it's not good enough, or it's not enough, or it decays, or it gets eroded by other factors, environmental factors, health factors, undernutrition, etc. Anyway, so the point is that there is natural immunity, and that's really what we're trying to recapitulate with a vaccine. Of course, BCG should be doing that. And to some degree, BCG does do that. However, the picture of BCG efficacy or protection against pulmonary TB looks like this. It's highly variable. So in some studies, the red dots represent the efficacy or the estimate in a study. And of course, the error bars represent the confidence interval. And in some studies, BCG has about 70 or 80% protection against pulmonary TB. But in other studies, there's no protection. And in some studies, the error bars are enormous, and so they're either underpowered or they're just highly variable results. And so the question is, why is this? When is BCG protective and when isn't it protective? Why do we even use it if this is the picture? Of course, in most countries, BCG is given at birth or very early on to protect young infants against TB. And the reason that we use BCG is this. It protects much more consistently against disseminated forms and severe forms of TB in young children. And in fact, it's estimated that about 50 to 100,000 lives every year are saved because of BCG vaccination. So we cannot, it's unethical unimaginable that we remove BCG at this stage. BCG is here to stay until there's something that might be better, but it's really important that we give it, and that's because of this. So what is it that interferes with BCG or that causes BCG not to be protective in every setting against pulmonary TB? And it's something that we can debate afterwards or we can discuss. We won't have the time now. But there was a recent analysis, an enormous analysis that included 68,000 people. Mostly from observational studies that sought to try and understand, well, what does BCG protection look like nowadays? Because most of those studies that I showed you are quite a bit older. And we can't actually do randomized placebo-controlled trials for BCG now, because anywhere where people are at risk of TB, they need to get BCG. So we can't have a non-vaccinated group. So it's very hard to estimate these effects. But what the study did is it looked at observational studies where people would have received BCG or not for whatever reason, poor delivery, beliefs, etc., and looked at a number of outcomes, including TB as the outcome, but also mortality. And so I'll just show you two slides. And so what this data suggested is that there was a small protective effect, about 37% protection in children under 5 years of age against TB, and that then essentially after that is no longer significant. So a lot less protective than initially thought. But of course, this comes with caveats of these are observational studies. The number of unvaccinated children in total is actually quite low compared to the number of vaccinated children. And so obviously this wasn't randomized in any way, and there could be many reasons why one would see this. But what was striking was the effect against death. And so you can see here, in all children up to the age of 14, BCG was highly protective against death, which I think is a very important and significant result that we should keep in mind. And so it's good to see that we do have some evidence, even now in modern times, that BCG is an important vaccine to continue giving. So the WHO actually quite recently, came out with preferred product characteristics for TB vaccines. This had not really been summarized and kind of produced until now, which is quite an amazing thing to think about. And so there are two strategic goals. The first one is that we need a safe, effective, and affordable vaccine for adolescents and adults. So a vaccine that works for older ages. And the reason for that is because these are the ages where TB is most infectious. Adolescents and adults develop pulmonary TB that often presents in such a way that people are infectious and transmit the organism. And from a public health perspective, we really need to turn off transmission. We want to stop people from getting infected in the first place. That is where the most gains could be made for everyone as a public health measure. And given that there are limited resources in the world, one should focus on those that are the highest priority. And you can see some of the criteria that they defined there: 50% or greater efficacy. It needs to work in both infected and uninfected people. Many people of these ages are already infected, and because they've been exposed in the past, etc. And the goal too is to get a better vaccine for neonates and infants. So at the same time, we also don't want to forget the young children who are, of course, very vulnerable, where BCG, we may be able to improve on BCG. We might be able to get more gains, or of course, in children who are immunocompromised, for example, BCG being a live vaccine can be problematic and itself can cause disease. So these are both included in the product characteristics. So this is the current TB vaccine pipeline. These are the products that are currently in human trials and clinical trials. The first thing you will notice is that it's not massive, certainly for a disease of this significance. And the other thing is that it is inverted. So usually you have more products in phase 1, and then, a few less in phase 2, and then, some of those make it to phase 3 for testing efficacy. But in fact, it's a little bit the other way around. There's only 4 products in phase 1, and then there are quite a few more that are in phase 2B or 3. So that is a problem. So the problem is that it's small. There's not too much diversity in the type of vaccines that are there, but also that we don't have very many products in phase 1 and 2. And so if the ones in phase 3 don't show efficacy, well, we don't have much to rely on, do we? And so this is highly problematic, and it's a product of underinvestment and difficulties and all kinds of reasons. But at least we're starting to see a little bit more diversity. So, for example, in blue, we have mRNA vaccines that are coming into the picture. The green ones are protein adjuvant vaccines, and I'll tell you a bit more about 1 or 2 of those. And in orange, we have live attenuated vaccines, much like BCG, and you see that BCG is even there. We're still doing trials on BCG. Unfortunately, one of those products, VPM, there are recent results for that from 2 big trials that showed that there wasn't expected efficacy, and so it may be that will entirely fall out. So this whole fear about, are there enough products to pull through if those in phase 3 fail, well, that may actually be happening already. So I thought I'd focus on 2 sort of examples, because these 2 vaccine candidates are in phase 3 trials, so efficacy trials. The first one is called MTBVAC. This is actually a live mycobacterium tuberculosis vaccine. So not bovis, but the organism that infects humans. It's attenuated, so it's got 2 genes that are completely deleted. And those genes are very important for virulence, and they control a number of other very important pathways in TB. And so there's very good confidence that this renders the organism massively attenuated, but of course it can still be grown and produced. And so I guess the most notable trial here is a phase 3 trial that's being performed in infants in Madagascar, Senegal, and South Africa. The trial is about 70% enrolled. The aim is to enroll 7,000 newborns who are then they're randomized to BCG or MTB-VAC. So BCG is the comparator. We can't do placebo, as I already mentioned. And so the aim here is that MTB-VAC has to do better than BCG. That would be the outcome, which is, I think, a difficult thing to achieve. But the vaccine is also in a trial that's being done in people living with HIV at the moment. It's important to test that there's safety in immunocompromised individuals. And of course, they are on antiretroviral therapy, so that's being strictly controlled. That's a smallish trial, safety and immunogenicity only. But then there's also a much more recently a trial that started that is looking at safety and efficacy in adolescents and adults, which will include people that already have evidence of infection, but also those who don't. So in the next few years, we'll hear more about this, and perhaps we will have a new live attenuated vaccine with some efficacy results available soon. I can tell you more if there's interests later on, if that's something you want to hear about. The other one I want to highlight is called M72ASO1E. So this is it's a polyprotein, so 2 proteins that are produced in a polyprotein, and they're kind of mixed up to make sure that the proteins are not functional. And this is formulated in an adjuvant, which is called ASO1E, which is actually also in other vaccines that GSK produces. So GSK developed this in the early years, and recently the protein part they didn't want to develop anymore. And so the Gates Medical Research Institute acquired the rights to develop that further, but it still needs the adjuvant from the ASO1E adjuvant, which GSK retains. And so this adjuvant is actually partly from a natural product. It's made from the soap bark tree, and unfortunately, it's a little bit limited in supply, and so there's competition for this adjuvant because, for example, it's also in the Shingrix vaccine, which is the zoster, for varicella zoster, and that's very lucrative because you can sell this at quite high prices in Western countries, and so that creates a bit of a conundrum that this, TB is never going to be a highly lucrative disease to vaccinate against, and so that presents some problems. But let me show you what the evidence is for this vaccine. So this vaccine is highly immunogenic. So we performed a trial in adolescents here in Cape Town quite a few years ago now. It enrolled both quantiferon positive and quantiferon negative people. Quantiferon is a blood test that we can use to ask, has someone been infected before? Is there evidence of prior infection? And so in purple are those who have evidence of prior infection. And you can see that, so it's given twice on day 0 and day 13. You can see on the left there, already after the first shot, you can see a really striking increase in T cells that recognize these 2 antigens. So it's very immunogenic. And in fact, the second dose is probably not even needed for those individuals because it's already very high, and you get very good memory responses. But even in the green bar, these are quantiferon negative people, after 2 shots, you see pretty good T cell responses. So this is a high amount of T cells, by the way, if you compare it to other types of products. But it also induces very strong antibody responses. On the right, after 2 shots, you see very high IgG levels. So a very immunogenic vaccine, and this is not that surprising. The adjuvant is excellent. We know that from a number of other vaccines. So then this vaccine, based on this kind of evidence, moved forward into a phase 2B, a proof of concept trial. There wasn't enough money or risk-taking behavior to say, let's do a phase 3 trial. We want to do a phase 2B first. And so that trial enrolled only quantiferon positive people, adults, because that's where the bang for the buck is. Quantiferon positive people are more likely to develop TB, so smaller sample size was possible. So 3,500 adults roundabout were enrolled in Zambia, Kenya, and South Africa. They were randomized to receive the M72 vaccine or placebo, and there was a 3-year follow-up period. So 3 years of follow-up for TB, so you can accrue enough cases to do a comparison between vaccine and placebo. It's not going to be possible to do trials where the endpoint is TB vaccine, TB disease in 2 months or in 3 months or in 5 months. It really takes time because TB is a slow disease. And so the exciting result, which was published a few years ago now, is that we saw 50% reduction in TB disease in those who were vaccinated with this vaccine. And so based on that, it took a few years, but we are now in the situation where we have a phase 3 trial, much larger sample size, and I'll tell you a little bit more about that in the next slide. But this kind of evidence was required to get the investment to then fund the phase 3 trial. And so here's a bit of detail about it. It's the same product. Based on the result of the phase 2B trial, the GSK actually said, we no longer want to develop this vaccine, even though there was a 50% efficacy signal. And the Gates Medical Research Institute then had to get the license. And that took a few years. And COVID happened in between, etc. So there was a massive delay. But the phase 3 trial is being done in multiple countries in Africa, as well as Indonesia. The trial has already completed enrollment, 20,000 adults and adolescents. So now the age is from 15 to 44. Really good idea to drop the age because we want to be able to use this in younger people as well. If you only enroll adults, then you're not going to have those younger ages as part of the indication. It included a few people that are IgRA negative, so evidence of no infection. It included a few people that are HIV, living with HIV. The results are, well, it's an endpoint-driven trial, so the endpoints are currently coming in, and so we might actually have results towards the end of next year or the year after. Initially, it was thought to be longer, but enrollment went really fast, and so we hope that we will have some excellent results soon, and I think there will be some efficacy. That's my personal opinion. A couple of issues around this. So that trial is being done in quantiferon positive people, mostly. There will be no efficacy from the small number of quantiferon negative people that were enrolled, because it's just, you'd have to enroll about 80,000 quantiferon negative people to allow enough people to become infected and then to develop disease. And that would just make for such a large trial that it's very difficult to fund that. But we have lots of evidence from many different trials that the immune response is extremely different in quantiferon positive or infected individuals compared to uninfected individuals. Because people mount immune responses to most antigens in the vaccines when they are naturally infected with TB. And we don't quite know what the implication of that is, but it's likely to have an effect on how we understand and plan for this. If trials are only done in quantiferon positive people, and we only get evidence for quantiferon positive people because those are the people that develop TB in the trials, then it's going to be very difficult to know what is the situation in quantiferon negative people. So this is a big issue. And if we want to move even lower in ages, which we really do want to do, we want to protect 10-year-olds or 12-year-olds, those people are much more likely to be uninfected and not to yet have immune responses to TB. And we're not really gaining enough evidence on those individuals. So the question is, how do we achieve greater? So let's say we do see 50% efficacy in this M72 trial, which would be a wonderful result. I've shown you the case that is out there for developing such a vaccine. It's very likely that it will lead to licensure and rollout. But of course, we want to do better than 50%, right? We can't stop if we see that for many reasons. So what are the things that we are asking to try and get high? And so the first one is that we really need to understand correlates of protection. For TB, we still don't know exactly what the immune response is that's protective. It's not like, influenza or COVID or, where we know, oh, we need to induce an antibody response that neutralizes the virus, for example. And so there are many efforts underway, but this is a major obstacle in the TB vaccine field. Selection of antigens may improve protection. I showed you this M72 result where we have a human signal based on 2 antigens. And those antigens were discovered in about more than 20 years ago now. And nowadays we have much better science to identify antigens. So we need to continue doing that, and maybe we can get much better if we have more antigens or better antigens. And then I think the mRNA vaccine platform, which you've heard quite a bit about already, I think provides an opportunity to do things quicker, not necessarily to ultimately to get to a new vaccine with efficacy data, but to ask questions in the relevant host, the human. TB only naturally infects humans and causes disease in humans and transmits in humans. And it may be likely that a lot of animal models are just not quite telling us the right story. In fact, there's lots and lots of evidence for that. And so perhaps mRNA vaccines will allow us to do dedicated experiments. And I know it sounds bad, dedicated studies in humans to understand what does the immune response look like? How do we change the way we formulate these vaccines? Or how do we use different antigens to get different types of immune responses that seem to be good to get better protection? So a little bit about this, just a few more slides. I know I've got 5 minutes. Thank you. Perfectly, I think, perfectly timed. I mentioned the monkey, and the monkey is being used extensively because monkeys are much closer to humans than mice, for example. In immunology, mice are used a lot, but mice are not a good model for TB. And so here is one experiment. This was done in the US, where monkeys were given intravenous BCG vaccination into the bloodstream, high dose, boom. It was incredibly protective. It was quite surprising. So it protected monkeys amazingly, and some monkeys were actually almost completely sterile when they were challenged with MTB, something that had never been seen before. But importantly, what that allowed the investigators to do is to say, oh, wow, high-dose BCG intravenous is protective. Let's give a number of different doses where we titrate it down, and then we get a sort of spectrum of protection in the monkeys, and then we can ask what immune responses correlate with the level of protection. That's what you need to understand correlates of protection, immune responses that correlate or induce protection. And that's what they did. And there were many, many features that correlated with protection. The 4 most important ones were all in the lung, not in the blood. For human studies, this is bad news. In humans, it's really difficult to measure immune responses in the lung. It's highly invasive, it's expensive, and actually in some people, it's not quite safe. You cannot do it on a massive scale in a large clinical trial. But anyway, the most important ones were all in the lung. And there were Th1 cells were much higher in protected. Yellow are protected animals, red are unprotected animals. Th1, Th17 cells, again, highly protective or much higher in those who are protected than not. Natural killer cells, we don't have to go into immunology. I know some people get the hives when we talk about deep immunology, but natural killer cells are a very interesting result because these are not antigen-specific cells. These are cells that probably collaborate with T cells to achieve or help to protect. And interestingly, IgA antibodies. So mucosal antibodies, IgA levels, were slightly protected or slightly associated with protection. And there were many others, but these are kind of the most important results. So important results for us to think about in humans. There are now massive efforts underway that collected samples during this M72 trial that showed some efficacy. We collected the blood samples, not from the lung, so that we could try and see if any of these look like they might be protective in humans. There's an enormous effort underway, and we actually hopefully going to get to results very soon. And a few people in the room are also involved in that. So it's very exciting. Just to show you that there are candidates that are in preclinical development. So the Stop TB Partnership Working Group for TB Vaccines collects this information all the time. Here are a number of them. Some of these, one actually in the top right corner is an mRNA vaccine. We've got some live attenuated vaccines. We've got some viral vectored vaccines, and we've got a few protein subunit vaccines. So there is some diversity, but we have no idea whether these will be protective in humans. I'm not going to speak too much about the mRNA platform because Pia spoke very well to this already and this morning. There are a lot of reasons why we should take the opportunity and try and make vaccines with mRNA, because it's quick, it's highly adaptable. We have incredible amounts of data on the safety and effectiveness of mRNA vaccines. We've never had so much safety data. I know that people are really worried about the safety and it's caused a lot of hesitance, but actually we have enormous amounts of safety information about them. And we know a lot about the efficacy. And so we really need to see if we can use this. It may happen that it won't work, but we'll find out enough and it'll advance the field. And so we can't afford not to do it. And so we've, as a group, as a South African consortium, jumped onto this a little bit. So what we did is in the human studies identified that there were immune responses to certain antigens that we could tell in humans are elevated in those who are protected against TB compared to those who are not protected. So people who developed TB disease had lower levels of immune responses to these antigens than the people who maintained infection without developing disease. And we called that the Titan vaccine, and this identified 4 antigens that are now in an mRNA vaccine that Pia mentioned is involved in terms of the manufacturing processes. We've done a lot of preclinical data. I'm not going to show any of those data, but as Pia mentioned, we do see that the vaccine is immunogenic in mice and that it can protect mice against challenge with TB. And hopefully we can move that forward and do some clinical trials on that in the short term. Just to mention that it does take a large team of people to do that. And so that's kind of what's represented on this slide with lots and lots of input from many different people. So it takes an ecosystem. And so that's all I really wanted to speak about, just to mention that, of course, these kind of trials and these studies take an enormous amount of collaboration and partners and expertise on many different levels. And of course, also a lot of funding. And so that's what's acknowledged on this slide.
Thanks so much, Tom. That was fantastic. We're going to start off with a question to you from Tony.
Thanks so much, Tom. So I used to give this lecture about 20 years ago, and it's great to see the progress, but it's also depressing to see the lack of progress in some areas. I wanted just to, I mean, there's so much to say, but I just wanted to zone in on one thing. And there used to be a slide we show where we had the drugs, the diagnostics, and the vaccines, and we showed that they were all kind of invented in the days of my great-great-grandfather. Now, drugs have moved on a bit, and you've shown us about vaccines, but my sense is that diagnostics are still lagging. And as Gary Martins once said to me, if your endpoint stinks, then your trial stinks. So particularly in the vulnerable populations like kids and HIV-positive adults, has there been any light coming down the tunnel there? We're still really bad at diagnosing TB.
Yeah. And in the context of clinical trials, you really want a foolproof endpoint. So the endpoints in clinical trials are still microbiologically confirmed disease as the primary or most robust endpoint. I mean, in kids, that's, as you know, extremely difficult because most children who develop TB are not confirmed. And we can't do trials only with that as the endpoint. We'd never be able to kind of get to the end of it. So we have more loose endpoints as well, like unconfirmed TB. There have not been major steps there. And just, I mean, of course, in the public health sector, that's not the definition that we need to treat a child. And so we as a public health community, on the side of caution, and probably many are overtreated. Many people who don't have TB get treated, but at the same time, we still miss many cases. And so it's just a difficult situation. But in clinical trials, I can't see us getting away from this at this stage, because we really need, and I mean, for example, careful analysis of that M72 trial where we did see efficacy shows that using different endpoints can be fraught with difficulty. And it is really, I mean, I think there's still very good evidence that we have to remain very rigorous in the way we ascertain cases to make sure that the evidence remains interpretable and strong.
Hey, thank you for the presentation. I wanted to ask, what do you think are the actual realities of rolling out the vaccine in Africa? If let's talk about M72, if it's going to be efficient, it's going to do well in the clinical trials if we think about the pharmaceutical side. So if the adjuvant has a patent on GSK, and we talked previously earlier today about how important local manufacturing is. So if the patent is not going to be open or the adjuvant won't be shared, how the M72 can be then rolled out?
Yeah, that's a key question. And it's a very concerning issue. As we stand, I'm not in the boardrooms where those conversations all happen, but my understanding is that there isn't yet a structure that's been worked out for that. There are many people that are advocating very strongly, and there are negotiations that are happening. The reason the Gates Medical Research Institute bought the rights or has the license for the protein is for this exact reason, that it doesn't go haywire. But I don't know what it'll look like with the adjuvant. I really don't. And of course, there are people who are working on alternatives to AS01E. You know, nowadays, adjuvant science has moved along quite a bit, and there are synthetic adjuvants that look really promising and quite similar. But the question is, how do you bridge those? So if let's say you had to do a bridging study between AS01E and this new adjuvant, what would that look like? Does it have to include, efficacy for TB? We don't have a correlative protection. We can't just measure neutralizing antibody levels in a smallish trial. It's a very tricky issue. And so the most likely scenario might be that the vaccine will be rolled out in certain areas of the globe first, to kind of generate more data, and then at some point expand up. But I don't know, and it's another major issue in the TB field.
So we have a question online, and it kind of builds on Julia's question. So Alan is asking do you know whether there are companies in Africa at the moment that are currently working on making the adjuvants?
My understanding is that that's not happening in Africa, but I don't know exactly.
Okay. And then Cornelius just wanted to know that in one sentence, what can you say is the main challenge when it comes to making TB vaccines?
Can't say that in one sentence. There are many. Maybe that we don't have enough people working on the problem. I think if there was a massive effort from many different, from industry, from academia, from public-private partnerships, etc., then we would be able to get there quicker. But I mean, there are many other reasons, and some of them I've mentioned in the talk. Right, it takes a long time. It doesn't incentivize people to work on TB if it takes 3 years from enrollment to the end of the trial to get enough cases to show something.
Thanks, Tom. Our next question, and then maybe one more question thereafter.
Thank you very much for that. I think M72, it's potentially quite exciting what it could do. I'm just wondering if it was to be to show efficacy once it gets rolled out, would that mean that you'd have to do IGRA testing before you give the vaccine, or would you only roll it out in places where there's a high burden? And secondly, because it's only because the theory is that it works for those who have infection, and it's 50% efficacy in the phase 2 in the previous trials. And the goal is that we want 50% efficacy for both those with and without the infection. I mean, how much would it, I mean, I'm sure it will be still very impactful, but I'm wondering how would it practically look?
Yeah, that is built into the modeling. So if you really want to know, you can look that up. But the idea is that no, we wouldn't have to test. We would, because that just becomes a crazy way of implementing a vaccine, and that's why IGRA negatives are included in the trial, so that we generate enough safety data, but there won't be efficacy data for IGRA negative people. But it'll be given. I mean, the idea is that we would give it to everyone, and then generate kind of post-licensure data on those, and I guess we'll find out.
And then our last question from Marsha.
I just wanted to ask, so the 50% efficacy, how's that established? Is that an arbitrary cut of 50% efficacy? And then like, how do we establish that's sufficient, like for efficacy? And then the second question is the implication of rollout for our HIV positive patients, because there was only a small number of people in the participants in the trial. And then the other thing is vaccine hesitancy and uptake. Do we prime our patients ahead of time to try and kind of instill that confidence now already to say this is coming, these are where we, so that it's not like just thrust on them and then said this is available and then they don't know which way
That's a really important. Can I take that one first? A really important point. There are efforts underway to start to do that, and there are teams of people working on it. It's very likely that that the regions where there are trial sites will have first access anyway. And so there are programs underway to work on that, but it hasn't started. How was the 50% arrived at? Yeah, it was slightly retrospective. So that was, so the phase 2, until we had an efficacy signal in that phase 2B trial, there were many in the field who didn't really believe that it would even work. And so a lot of stuff didn't start until then. So so what then was done is that because we saw it was 49.3% efficacy or something like that, the modelers started working with that number and said, well, if we do see 50% efficacy, what would the model look like? And so they have very large epidemiological models that have, they've been used for decades and improved all the time about, if you bring in TPT, what would be the effect? Or if you changed screening for TB, what would be and so you can also plug in this model where you have certain ages, you assume how long the vaccine would be efficacious, and based on that, you can then estimate what the possible effect would be, and if it costs a certain amount, how much would that save, what would the DALYs be, etc. So those are the kind of calculations that come into it. But the 50% was basically informed by this. Of course, you can model, when you say 50%, you can model 30%, and you can model 70% and ask, well, what does that look like? And so 50% was deemed acceptable because it was already seen. Going much lower than that obviously makes things worse, and going higher than that makes things better. I can't remember your second question now. Oh, yes. So, I mean, the only thing I can say is that we already have results from HIV-infected people who are on antiretroviral therapy and have good control, so are immunocompetent. And if you vaccinate people who are immunocompetent, the immune responses are very similar, actually. So as long as it's done in combination with a good ART program, which of course would be needed, then I don't think that there would be, I mean, maybe in very large studies, you would start to see small differences. But based on current evidence, it does look like the vaccine is immunogenic. There are no more safety issues than there would be in uninfected individuals. And so I think that it would just be part of that rollout.
Thanks so much, Tom. And thanks for all the questions, everyone. Great engagement. So next up, our speaker is Erica Anderson-Nissen, who runs the immunology lab at HVTN based in Cape Town, and also forms part of the network doing studies on this. And this actually leads us beautifully from Tom's talk, Erica, about those people where we actually need extra help because things like BCG wanes and, you know, protection from previous infection wanes. So over to you. Thank you.
Thank you. Can everyone hear me okay? All right, well, thanks very much. This is actually the first time that I've been able to give this talk in the same session as Tom, and it was actually nice to see your talk, Tom, and some of the parallels to mine. So hopefully he set me up well for this talk. So I was also given a very provocative or helpful title, HIV vaccines 40 years on, where are we now? So what I'm going to take you through today is, first of all, just talking a little bit about why do we still need a vaccine for HIV. So I think most people are quite aware of pre-exposure prophylaxis methods, and there have been some breakthroughs recently in with the lenacapavir results. And so I want to really address that up front, and we can talk more about that in the discussions. I then want to take you through the history of developing a protective HIV vaccine and just the incredibly important role that the continent of Africa has played in this so far, and I'm sure will continue to play in this. And then lastly, a section of the talk which I continually have to update, which is sort of new approaches to HIV vaccines that are coming down the pipeline that give us hope. So I think it's obligatory to start with something about the global HIV numbers. This comes from the UN AIDS fact sheet from 2024, highlighting that 1.3 million people became newly infected with HIV in 2023. And of the 1.3 million new cases, almost a third of those were in East and Southern Africa. So this is a region of the world where we really need to push ahead and focus and try to get an effective HIV vaccine. So what is the news about pre-exposure prophylaxis? Well, in 2024, the Purpose 1 trial was published. And this was a phase 3 trial that evaluated the superiority of subcutaneous lenacapavir, which is an HIV capsid inhibitor, that was given subcutaneously every 6 months to individuals in the trial versus daily oral Descovy or Truvada. And it enrolled girls and women ages 16 to 25 in South Africa and Uganda. And there were actually 0 HIV infections in the more than 2,000 person lenacapavir group. So 100% protection was observed in this trial. So this is fantastic results. It's almost close to that level of protection was achieved in a second trial that was published this year. And so where is this amazing advances in PrEP fit in with our concept of an HIV vaccine, and why do we still need one? Well, I would point you to this article that was published in April of this year. I'm really talking about what are the amazing ramifications of the lenacapavir findings, and then why do we still need a vaccine? So just to summarize that here, well, it's safe and highly efficacious in preventing HIV. The efficacy observed in the Purpose trials is really similar or even better to some of that in many conventionally available vaccines. And it's thermostable, which is huge, as you know, in our field. It can be shipped and stored at room temperature. But it's not going to solve all of our problems. When the results first came out, the biggest issue is cost, right? And accessibility to people who can't pay what Gilead was charging for it. But it also requires injections every 6 months. You have to self-identify at high risk for HIV acquisition and really have access to a stable healthcare system if you're going to go in for injections every 6 months for whatever period of time you think that you're at high risk. It's a drug, so there's potential for drug-drug interactions. And you can imagine if you get an injection every 6 months, the drug wanes over time. So at times when you might have low levels of lenacapavir in your system, if you're exposed to HIV, you have the potential of developing more resistant HIV. And we'll talk more about diversity in HIV being a huge barrier. And there's barriers to cost adherence and implementation. So it's not the answer. It is one amazing tool in our toolkit for prevention of HIV, but it's not going to fix the problem. So where are we with modeling studies about the impact of an HIV vaccine? And I actually asked around, and there isn't a new modeling study that's been published yet since the knowledge about lenacapavir. So I refer you back to this 2016 modeling study that was published, which here's the graph showing basically the number of new infections with HIV in the millions over time. And the modeling study that was done modeled HIV vaccine being introduced in 2027, and assuming that this vaccine would have 70% efficacy, that it required 3 doses, and that you boosted after 5 years. And the different colors just signify the different assumptions made in this modeling study. So green is sort of the current trends at the time. This was back in 2016. In orange, and then in purple, you have scale up of what's called the UN AIDS Investment Framework Enhanced. And there are probably people in this room who know more about this than I do, but it basically involves all the other sort of prevention methods, scale up of PrEP, treatment as prevention, other methods, etc. And you can see in the solid lines, it's modeling if you didn't have a vaccine. And in the dashed lines, it's showing you how many infections you could avert if you actually did have a vaccine of this caliber. And so the bottom line is we could prevent a lot of infections if we had an HIV vaccine, and used in conjunction with other prevention methods, we could have a huge impact on the pandemic. So time to develop a vaccine. How does HIV compare? This is, I just wanted to point you also to this AVAC website. It has amazing resources showing beautiful infographics about HIV vaccines, HIV prevention, a lot of statistics made very clear and accessible. So I encourage you to visit that website and use their resources. But you can see that in comparison to other diseases, as my title said, you know, we're 40 plus years on since the development of the microbiological cause of AIDS, and still developing a vaccine for HIV. So why is HIV so difficult to target in a vaccination setting? Well, like other pathogens that you're hearing about this week and discussing this week, it infects at mucosal surfaces. So these are really complex environments where you have a lot of barriers, immune factors at play. Which ones do you really target to prevent infection at a mucosal surface? And so when you're thinking about the development of an HIV vaccine, and I think we always have this dichotomy of the vaccinologists and the immunologists, and I think especially in HIV, those two have come together a lot because we've learned so much about the immune system through the work done trying different vaccines and trying really to get very high tech about how we design these vaccines. So you'll see that, I think, there's a thread that continues through the talk. But the HIV vaccines that we've developed have tried to target sort of broad classes of immune responses to stop or control infection. And so broadly neutralizing antibodies, you'll see the approaches to induce broadly neutralizing antibodies have been tried. Approaches to induce non-neutralizing or binding antibodies that then work through other mechanisms of the immune system have also been tried. And then another one is cytotoxic T cells to really kill virally infected cells. Why has HIV vaccine development been so challenging? Well, one of the major reasons is the genetic diversity of the virus, and we'll get into that more in the next slide. Other challenges being no natural immunity to HIV infection and no good human challenge model, imperfect animal models, limited return on investment to develop vaccines. Again, the problems that all of us face looking at diseases that affect populations that can't pay a lot. And vaccine development is slow. And at the end of my talk, I'll talk more about some approaches that the field is taking to try to accelerate clinical trials for new vaccines. So HIV diversity, here are some phylogenetic trees that I think just show you very visually how diverse HIV is and make you understand really what a challenge it can be. So on the left-hand side of this slide, I have a phylogenetic tree showing HIV diversity in the Democratic Republic of the Congo in 1996, so one country. And then you see the similar tree on the same scale for global flu in the same year. So you can see the scale is incredibly different. Then when you go over, sorry, it might be hard to see, on the right-hand side of this slide, it shows phylogenetic tree for HIV diversity in Brazil in 2010. And then this little tiny dot that you can't even see is the SARS-CoV-2 diversity in the first year of the pandemic. And then it's blown up on this side here. So the scale of diversity for HIV is just incredible and makes it really challenging for us. And where is a lot of that diversity focused? Well, here's the structure of HIV on the surface. You have this envelope protein. And these purple parts, the trimers, are highly variable. And so the HIV can easily avoid immune responses by changing the sequence of its envelope. It also has this glycan shield, so it puts sugars on the surface of it, and that makes it also more challenging for antibodies to recognize. So we've got a lot of challenge with HIV. So what is the history of developing a protective HIV vaccine? And my hope is that this will give you sort of a concise history, some of the joys and a lot of the disappointments in this field over the last 40 years. And I just wanted to reiterate that African countries have made major contributions to HIV prevention trials. This is another nice infographic from the AVAC website. And just showing in the year 2018, there were more than 500,000 participants from Africa enrolled in HIV prevention trials. So this includes vaccines, microbicides, other treatment trials, et cetera. But just the enormous commitment that we have on this continent towards HIV prevention. And as I mentioned at the start, when you look at those mucosal surfaces, you think about how HIV can infect, there's been a lot of thought that's gone into engineering vaccines to induce different types of immune responses that would hopefully then be protective. And so on the left-hand side, you see a schematic where if you imagine that on the y-axis is the HIV RNA, and then on the x-axis is time, showing exposure and then over time. And your ideal scenario, right, was you have a vaccine that can completely prevent acquisition. But we also might be able to develop a vaccine that prevents prolonged systemic infection. So someone might get infected, but your immune system can actually get it under control and eliminate the virus. Or on the right-hand side is sort of the least desirable, but also could have a huge impact on the epidemic and prevent disease, where you get exposed, you get infected, but the virus remains at quite a low level, and so therefore you don't transmit it to other individuals. And so there are various immune mechanisms that I'll highlight as we talk through the different vaccine strategies that have been tried that could help with these different goals. So the first would be the induction of broadly neutralizing antibodies. They'd be sitting at your mucosal surfaces, and the virus doesn't even get in and infect. All the way over to the right-hand side of the slide, where you have induction of antiviral T cells that are going around and killing infected cells, but keeping things under control. So the first approaches that were tried were really to hit this high bar. Can we induce broadly neutralizing antibodies? Broadly just means that it would be effective against lots of different strains of HIV, because we have this issue that there's huge diversity worldwide, and you really want one vaccine that could work in many parts of the world. And so that was aimed to really hit the diversity question and, overcome that. And so in 2003, the VACCInology trial was the first phase 3 study in high-risk volunteers in the US and Thailand. And unfortunately, this trial found no efficacy. So the field had at least gotten to the stage of an efficacy study, tried it, and then, unfortunately failed. So then the field really swung toward, could we induce antiviral T cells? There were a lot of studies done in various models as well as in humans to show that the early appearance of HIV-specific T cells correlated with initial control of viremia. So those T cells were cytotoxic, were doing something. CD8 T cell responses correlated with long-term control of replication and slower progression to disease, to AIDS. And then they identified conserved T cell epitopes. So I told you this problem of the envelope of the virus being super variable, but there were also genes in HIV that are a lot less variable. So for instance, GAG. And so if we could find parts of HIV that were less variable, maybe those could be targeted by the vaccine, and you could develop one that would be very cross-reactive across different clades. And so sort of T-cell strategies were put into practice. And the first results from a T-cell focused vaccine came out in 2007. And this was the Merck Ad5 vaccine. And that was tested in the US in the STEP study and in South Africa in the Pambili study. And these were companion phase 2B studies in high-risk volunteers. And unfortunately, this trial, we used an adenoviral vectored vaccine, which induced great T-cell response, but there was no efficacy observed. And in fact, there was this finding that actually there was a possible increased level of HIV acquisition in certain subgroups in this trial. So this was basically the worst possible outcome you could have for an HIV vaccine trial. It didn't work, but there were also certain subgroups of men who were uncircumcised and had neutralizing antibodies to adenoviral vectors actually seemed to show higher levels of acquisition. So this was a crisis. Everything was stopped. People were unblinded. All of the risks were described, etc. Now, meanwhile, in the background, there was also a companion, another trial called the HVTN 505 that was also coming along, and again used an adenoviral vector vaccine to try to get good T-cell responses. And unfortunately, this trial also showed no efficacy. So the field really hit this roadblock that, oof, maybe these T-cell inducing vaccines actually weren't doing what they were supposed to, and pivoted quite a bit. Now, some of this timeline is a bit jumbled because things, these trials start and they take varying lengths of time. But I wanted to then talk about, in 2009, the RV144 or Thai trial results were published. And this was a phase 3 study in low-risk volunteers in Thailand. And I'll just go into that one in a little bit of detail because that was the first ray of hope that we could actually make an HIV vaccine. So here's the way the trial was set up. It was using the ALVAC, which is a canary pox vector, including HIV genes inside. And then it also had 2 boosts, which using the AIDS-VAC protein boost. And this used the alum adjuvant. And this was tested at that time, it seemed like an enormous trial, 16,000 volunteers who were low risk, and in Thailand, it was a phase 3 study. And what did we find? Well, the study found, if you look at the probability of HIV infection over time, and you see the placebo recipients in blue and the vaccines in green, it was actually an estimated vaccine efficacy of 31%. And this was, you sort of blew everyone's minds in the field because there are actually a lot of naysayers that said this trial was a complete waste of money and that they shouldn't even go ahead. And then it went and showed 31% efficacy. You can see the p-value is 0.04, so it was modest, and a modest efficacy of 31% with not great statistical significance, but it was the first indication that maybe we could actually make an HIV vaccine. And Tom talked about this a little bit, but one of the important things then is if you have some efficacy signal is doing some immune response analyses to try to figure out if you can figure out what the immune response was that was associated with protection so that when you develop further iterations of this vaccine, you might be able to improve upon it. And so there's immune correlates analysis that was conducted. And the significant findings really were that IgG antibody responses that were targeted to this hypervariable region of envelope were actually associated with a decreased risk of infection. So if I were in the trial and I had really nice high titer IgG induced, then I was at lower risk of infection. But conversely, IgA, which is an antibody that's known for being present at mucosal surfaces, actually was associated with an increased risk of infection. So there's a lot of work done in the field to try to look at how these antibody responses might be working together or not working together to try to understand why this vaccine showed 31% efficacy. And basically the bottom line, this is just a schematic showing that envelope trimer on the surface of HIV. And what they found was that the IgG antibodies could bind, but if you had IgA antibodies, they actually kind of blocked the binding of the IgGs. And the IgGs were really important for Fc receptor or other cells coming and attaching to those antibodies and having effector functions. So I think it just illustrates the complexity of the problem. It's all very great to induce antibodies, but what type are you inducing and how do they interact with the other antibodies or other immune responses that the vaccine is inducing? So after 31% efficacy signal was seen. There was great enthusiasm, and that's actually why my laboratory, which is part of the HIV Vaccine Trials Network, was built here in Cape Town, was to support trials that then went forward primarily in South Africa, following up on this vaccine. And so the vaccine was tweaked to include products that match the clade C circulating strains of the virus here in South Africa. The adjuvant was changed to one that was known to induce better immune responses, MF59. And unfortunately, that trial fell flat. So the vaccinations were actually stopped in 2020 due to futility. So though we'd seen this great signal in Thailand, we thought we'd made the vaccine even better, tested in South African participants, it doesn't work. We can talk a little bit more about the reasons for that, perhaps in the questions of this, sections of this talk. So simultaneously, there was this PrEP-VAC trial that has been running, and this was also designed to sort of induce binding antibodies similar. The hope was that similar to what was seen in the RV144 study, although this one used an MVA, another poxviral vector vaccine, conducted in Uganda, Tanzania, and South Africa. And unfortunately, this trial also stopped vaccinations in December 2023 due to futility. So we had this great finding in Thailand. We found that these binding antibodies might be important and associated with protection. Then you do follow-up studies and you can't find the same result at all. So then the focus was also going back then to T cells again. So, okay, we had this initial T cell finding with the adenoviral vectors, but they didn't show what we hoped. In fact, they looked bad. But simultaneously, Johnson & Johnson was developing a mosaic vaccine approach. So I talked about the T cells being important and looking at the more conserved regions of the virus. They developed a vaccine that looked at the viral diversity in sequences across the world and took pieces of those sequences from all the different clades and put them in one vaccine and called this their mosaic approach. You'll see some familiar vectors there, adenoviral, but serotype 26 this time to avoid the issues with Ad5, as well as an MVA vaccine. And they had a wonderful, huge vaccine trials, program basically to go from phase 1 all the way up to efficacy studies to test Alvac versus their Ad26 vaccines in using these mosaic approaches with boosting with the clade C proteins. And unfortunately, there was, so if you look at the summary in the middle of this slide, there's Imbokoro study that was conducted here, as well as the Mosaico study that was conducted in the Americas. And these studies concluded in 2021 and 2023. And the vaccine efficacy in the Imbokoro study was only 14.1%. And you can see that's a huge confidence interval. So there was really no vaccine efficacy observed. And the Mosaico study was even worse. So unfortunately, this T cell mosaic approach didn't work. So it's been a journey, that's for sure. And many people have worked very hard, and there's been great funding and support to develop HIV vaccines. But you can see that you can put all of this work in, and yet you can arrive at these huge efficacy studies and not get the desired result. So I wanted to then sort of back to the future, new approaches. So now we're back to where this field has swung back to broadly neutralizing antibodies. And it's not because we ran out of ideas, et cetera, but it's actually because our knowledge of protein engineering of envelope of HIV has really increased over the years since those early 2003 trial. And we can actually take new approaches. And I think it's really an exciting time in the field because there are some just really new approaches in biology that are being taken that will hopefully move this forward, this vaccine forward. So one of the things that I just will show you one slide about in the broadly neutralizing antibody field is it's all very well, yes, if we could get great titers of broadly neutralizing antibodies, we could stop and prevent HIV infection, but how much do we actually need? And so there are a set of studies that were done a few years ago called the AMP, or antibody-mediated prevention trials, which were not vaccine studies, but in fact, instead what was done was to recruit volunteers to be infused with broadly neutralizing antibodies. intravenously, and took high-risk volunteers who would be willing to be infused with the antibody, and then see whether this antibody could protect. And there was a lot of planning done for this trial as to how much antibody need to be infused, when you needed to measure, because obviously infuse it and then it falls off over time. But to try to figure out statistically, could we figure out from these trials how much antibody you actually needed to prevent infection? And so these were the HVTN 703, HPTN 081, and 704-085 trials that were done both in sub-Saharan Africa as well as in the Americas and in Europe. And what they did was to infuse individuals with the VRC01. And what they found was, in fact, that these infusion of these monoclonal antibodies did not significantly reduce overall risk of HIV acquisition. But if you looked at the strains of HIV for people in this trial, the people who did become infected with HIV, in fact, were infected with strains that were kind of matched to the antibody that's infused. So again, it goes back to this problem of viral diversity, right? I have a broadly neutralizing antibody in my system, but if I'm infected with a virus that doesn't really, that antibody isn't that effective against, I'm going to get infected. But what they found when they did viral sequencing of the people in these trials, and Carolyn Williamson's lab at UCT has led these efforts, is that in fact, people who did acquire HIV actually had viruses that were less sensitive to the antibody that had been infused. And they were also able to calculate the level of broadly neutralizing antibody that people would need in their serum to be protective. So even though they didn't show massive protection, and it's probably because they didn't actually start with enough antibody, they were able to figure out if we had a vaccine that induced a neutralizing antibody, this is the level that we would need. And so it really gave the vaccine field for broadly neutralizing antibodies a target to aim for. So the field has worked a lot recently on how to get better broadly neutralizing antibodies. You can isolate them from people who have been living with HIV for many years, but they take a long time to develop. And Tandile in the audience, who is in my lab, used to work a lot on that. So if you have more questions, you can chat to her. But what the approach that's being used now is to do what's called a germline targeting approach. So there's been huge advances in how to make envelope proteins, how to stabilize them, how to engineer them and give them to people in vaccine scenarios. And so the approaches that are being used now are do a variety of sequential immunizations to kind of imitate what would happen if you were living with HIV and you were developing antibodies in your body over time to eventually get to the broadly neutralizing antibodies, but do this in a vaccine scenario. So what they do is they take germline targeting immunogen, so an HIV protein, they vaccinate people, they see which naive B cells are being triggered. Then they slightly tweak that immunogen to kind of diversify those B cells to try to increase their ability to neutralize the different viruses. And there's all these sequential vaccine regimens being tested right now to see how do you get to the endpoint of getting a broadly neutralizing antibody, and how do you reduce the number of vaccines and which immunogens are the best ones to do that. So it's a really exciting field looking at protein structure, protein engineering, as well as looking at different adjuvants. How do you give these proteins? Do you give them all in one dose? Do you give fractionated dosing? There's some indication that if you do fractionated, you actually get better B cell maturation and B cell responses. So there are really some exciting developments happening in the field right now. And I'll just refer you to some trials that have been conducted in Africa. The IAVI, or International AIDS Vaccine Initiative, is leading a lot of these trials. And there's both sort of a protein adjuvant approaches that are being tried, as well as mRNA-based vaccines. And my organization has more than 20 trials planned in the next few years to test different immunogens. This is just a reference to show you some of those trials. So alongside with the broadly neutralizing antibody approaches, there's also been a lot of developments in how do you target T cell responses, and thinking about how do you address this issue of the diversity of HIV. And, ultimately, we have a very complex immune system. You're actually probably going to need all of the arms of the immune system. There's a reason why it's so complex, and we have all these different cell types and mechanisms for defense against pathogens. You're going to need B cells and T cells and other mechanisms in the innate and other parts of the immune system to really prevent HIV. So simultaneously, there's been a lot of work in thinking about, we had this mosaic approach to look at different sequences and put them in to get a T cell response. But how else could we look at designing a better T cell vaccine for HIV? And here's one example of a trial that's actually happening right now. And this is amazing work that's been done at the Reagan Institute in Boston by Gaurav Gaya and colleagues. And what they did was to actually look at the three-dimensional protein structure of different proteins in HIV and identify which amino acids in those structures were highly conserved and highly networked. So how did they function in the three-dimensional space? And if you mutated one of those, it would mess up the structure of the protein. So they've been really focused on developing new ways to get a better immunogen for T cells that would be more conserved against clades of HIV and really critical for the virus. And so there's a new a trial that just started July 28th. The first immunization was conducted in Zimbabwe. And so that's using a gorilla adenoviral vectored vaccine. So you get the theme that adenoviral vectors make really good T cell responses, but the trick is to figure out which one you want to use. So this is trying a gorilla vaccine, adenoviral based vaccine. So we'll wait and see what the results of that are. So I think, one of the critical questions faced by all of us is how do we speed up clinical trial testing of different vaccines? In the HIV space, there are a lot of new ones coming down the pike, and there's been a lot of thought and effort into how do we actually speed up testing so we can really rapidly eliminate candidates that we don't think are going to work and move much faster into higher phase trials. And so that's where the sort of the advent of the discovery medicine trials have come up. And I won't go through all of the details on this slide, but just to say that the idea is to do much smaller phase studies that can actually give you scientific information that's needed to improve the vaccine regimen. And so the idea is that you would have, these would be small trials where you have, these are small trials where you have, often fewer than 30 people in the trial, and they're not even always placebo controlled, but just really quick trials to allow you to do a lot of immune response measurements in a small number of individuals and get some indication of whether you think this vaccine candidate should actually move into larger scale studies. And this slide just shows sort of a mind-bogglingly long list of some of the discovery medicine trials that are ongoing or just starting now to really target different parts of the HIV envelope trimer. So it's an exciting time. And we, are really looking forward to the results of these different trials. Again, trying different adjuvants, different dosing schedules, different engineering of the protein to try to figure out what might get us faster to a broadly neutralizing antibody. So to summarize, an efficacious HIV vaccine still eludes us. I walked you through the results of 4 efficacy trials that have really failed. The RV144 trial was, the first beacon of hope, but we were not able to recapitulate those results in the sub-Saharan African trials or in the Europe or America trials. The mosaic vaccine regimen has not gone well, and this PrEP-VAC trial has also failed to show efficacy. But in data that I haven't shown you, there's been a lot of work. Even in a trial that doesn't work, you can look at subgroups and you can try to get signals of immune responses that might be important for efficacy. And I haven't had time to go through those with you today, but there are some nice studies that have been done that actually show, we really do probably need antibody and T cell responses to work in concert to get a better vaccine. I told you about other promising HIV vaccine approaches that are actually being tested right now. But of course, as probably has come up or will be talked about more in this course this week, there are huge concerns over funding. And you can tell by my accent that I'm from the United States and, deeply disappointed in where my country sits right now in terms of funding for vaccines in general. Embarrassed, all of those words. But, I think we just need to continue to move ahead. We've done so much work. And hopefully the lessons, the HIV vaccine field, has been incredibly well funded compared to many other vaccines. And I think we just need to keep moving forward with what we have. And I just wanted to end the talk saying that, that the work that's been done and the money and the effort that's been put into HIV vaccines really did lay a framework towards this hugely rapid development of a SARS-CoV-2 vaccine. And is hopefully the lessons that we're learning in the HIV vaccine space are being translated into other fields. And so I think collectively we're going to move forward and we will eventually get there. So I'd just like to thank the members of my lab. Again, Tandile is here. She's happy to chat to you more about what we do here in Cape Town, and the many people who have supported the work that I've shown to you today. Thanks so much. I'll take any questions.
Thank you so much for that insightful talk. And just reminding us again, we might have medication progress, but if we don't have vaccine progress, does it really make a difference? So I'd just like to also remind the audience, please ask questions, but if there are any that might straddle better into the next talk, which is about implementation and capacity building, if you could hold some of those over. Thanks.
First, I would thank you for that insightful presentation. I am Mariam Adel from Egypt. I have worked in my master's degree on HIV and people living with HIV in Egypt. And this technology that you are that you are talking about in presentation is very promising. I want to ask about what about CRISPR-Cas9 technology approach? Did you use for developing HIV cure or developing HIV vaccination? And also, this is the first question. Second question, I think that the most problem facing people living with HIV is the stigma and discrimination. We have several solutions for that and I want to talk about it. For example, we use mobile application to reach these people, to link it to adherence to care and to link them to care. Did you use it? Did you face this problem? And did you use this solution or this technology? And thank you very much.
Thanks. I'll take the second question first. So I think there's huge stigmatization, and there have been, especially over the course of these many vaccine trials, huge opportunities to look at issues of stigmatization, look at behavioral issues, what prevents people from accessing care, what prevents people from some of these trials, with PrEP coming along, you do an HIV vaccine trial, you must offer pre-exposure prophylaxis to people in this trial. But yet you offer it and so many people actually refuse to even take it, even though they are maybe high risk, etc. So there's been a lot of opportunities for some of those more behavioral studies and to address the questions of stigma. For instance, in an adolescent trial where people are very, the adolescents are hyper sensitive to how they're viewed, and they don't want to carry, pills that rattle around in their backpacks or something to school. Everything from that to uptake of PrEP, et cetera, et cetera. I'm not specifically aware of some of the mobile applications that have been used in the studies, but I'm sure there have been some. Yeah. So I mean, those things are absolutely critical, and you can develop the best medicines, prevention, vaccines out there, but if you can't get people to come when you need them to get really good immune responses, then it's not going to work. The HIV cure field is coming along enormously. There's this issue of once you acquire HIV, it integrates into your genome. And so how do you reach those reservoirs? How do you reach like every last cell in your body that has HIV in it? And many of those technologies are being tried. And we're trying to learn between the vaccine and the cure fields, how can we talk to each other, as well as the cancer field. So there's a lot of progress in cancers in terms of how to target cells that are apparently expressing certain things. And so how can we learn across fields and really advance all of us? Yeah.
Oh, thank you so much for the presentation. My question, maybe it's a little too early for this discussion, but is the idea to vaccinate everybody? Because, for instance, if we partners of people living with HIV are at risk, but also identifying them would have to be through the person living with HIV, and in many cases the status is confidential and people don't want to share their status with their partners. So is the idea to make this a mass vaccination thing or just target risk populations? But I know it's a little early to discuss this.
No, it's a good question. And I didn't even talk about efforts to look at HIV vaccines in babies, right? Because ideally, you would be able to vaccinate people much earlier on before they're sexually active, or even to protect babies against mother-to-child transmission, et cetera. So I think, the ideal would be everybody, but, depending on what we find, and there's some early data actually that if you vaccinate babies, they can actually develop better B cell responses in some cases. And so there's a lot of work going on to look at sort of across the age spectrum, across different scenarios. Ideally you'd have one, you give it to everybody, but that may not be where we end up, at least initially. Questions about
Thank you very much for the very insightful presentation. I'm a medical doctor and public health professional from Tunisia, and I started my public health work on HIV. What my question is focusing on HIV too. It's always the neglected epidemic of HIV, but there is some data or some literature showing that there is a cross immunity between HIV-1 and HIV-2, whereby people infected by HIV-2 tend to be less likely to be HIV-1 infected or develop some sort of a milder infection by HIV-1. Is there efforts on vaccine development that look into the model between HIV-1 and HIV-2 cross immune response and the potential to use HIV-2 as a genetically more stable than HIV-1 to immune people or immunize people against HIV-1? Thank you.
That's a great question. And to be honest, no one has brought up HIV-2 in any meetings I've been to recently. So I actually would need to go back and look and see what's happened since those studies. Yeah, because there is some really interesting sort of cross-reactivity data on them. And I'm not sure actually where that's gonna I would have to go look up.
Thank you. I believe we've got a question online.
Yep. Khadija is saying thank you so much for the presentation. And they're asking what are some of the efforts put together to make the HIV vaccine available to all countries in Africa?
That's a great question. I think, in the aftermath of the RV144 Thai trial, and when there was huge enthusiasm and hope that the vaccine that was tested here in South Africa would actually show an efficacy signal, there was a lot of momentum towards preparing how do we roll it out, et cetera, et cetera. To my knowledge, a lot of that has stalled because in some ways the field has gone back to the earlier phase trials. So there's a bit more time till we actually get to somewhere. But there have been huge efforts looking at, vaccine systems, looking at, in conjunction with some of the COVID vaccine work also about how do you get vaccines out to people, and what are the preferred characteristics of the vaccines that we need in order to be able to distribute them widely. Yeah.
Thank you for the presentation. It's quite complex ideas, actually. I'm just wondering about the BNABs. It looks like they require boosting. And even with the trial, with the RV144 trial, the efficacy that was seen, was it sustained or did it decrease?
No, it's a good question. And as you pointed out, and I haven't even showed you some of the most complicated trial designs, but at this point, we need a lot of immunizations, at specific timed intervals. And in a lot of cases, we're not even assessing durability of the response more than sort of a year out. So that the durability is going to be the next hurdle. If we can find something that's giving good immune good immunity, nice memory profiles, then yeah, at what point are you going to need booster vaccinations, et cetera? They're very complex designs, and it raises huge questions about, on a global scale, if we find one that works, but it requires 6 immunizations, how practical is that actually?
Okay, any last questions? Okay, if not, then I'm going to just close this talk. Thank you so much. Okay, but we're also seeing, I think, just looking at where the disproportionate burdens of disease lie, that none of this actually of us even being in this room is meant to be seen in isolation. So even with progress in development and labs, we really do need capacity building and vaccinology training. Corinne Deshay, formerly from NESI, which is the Network for Education and Support in Immunization. And she's now at the Institute of Tropical Medicine in Antwerp, Belgium. And she's here to talk to us exactly about education, interacting with policymakers, and integrating all of this now. Thank you so much. Please.
Yeah, thank you for the introduction, and thank you for the invitation to be part again of the vaccinology course. I've been here some years back, then a few years I missed, but thank you very much for the invitation again. And I hope my voice will keep going for the time of the presentation. Yeah, okay, thank you. So I got the title, Intersections Between Clinical Trials, Regulation and Evidence-Based Practice in Advancing Immunization Policies. When I got the title, I thought, okay, how much time do I get? But then I took a step back and I thought, let me look for some recent publications, typing in the different topics in the title. And then, of course, AI gave me this. The intersection of clinical trials, regulation, and evidence-based practice is crucial for advancing immunization policies by using data from clinical trials to inform regulatory approval and guide the implementation of policy based on robust evidence. Clinical trials generate the safety and efficacy data needed by regulatory bodies to authorize vaccines, while public health experts use this data alongside real-world evidence to develop and refine policies to maximize vaccine impact and address barriers to uptake. I thought, yeah, that was a very nice summary. Can I stop now? Do we have people here from a national regulatory authority? No? Okay. Okay, let's, so what is a regulatory authority? What is their mission? And here's some, I listed from different countries or agencies, their definition. So a national regulatory agency, they are responsible for ensuring that products released for public distribution are evaluated properly and meet international standards of quality, safety, and efficacy. And these 3 words you will see come back in the different agencies. The mission of the European Medicine Agency to foster scientific excellence in the evaluation and supervision of medicines for the benefit of public and animal health in the European Union. So some regulatory agencies will only focus on for human medicines, but others also animal health. FDA, we relied very much on them, so let's hope for the future how it will continue to promote and protect the public health by helping safe and effective projects reach the market in a timely way and monitor products for continued safety after they are in use. I have here from India, and then since we are in South Africa, from SAPRA, which is the national regulatory body responsible for overseeing the safety, efficacy, and quality of health products in South Africa. So These regulatory bodies, there are several functions that they have to commit to across the project life cycle for medicines, vaccines, medical devices, and including in vitro diagnostics and blood products. And in the graph, you can see that there are common regulatory functions for all types, and then there are some specific for vaccines and blood products. And I've listed here that is better to read. So the the mission of regulatory authorities that they have to look at the national regulatory system, registration and marketing authorization, vigilance, market surveillance and control, licensing establishments, regulatory inspection, laboratory testing, clinical trial oversight, and NRA lot release. So for this presentation, we are more interested in the clinical trial oversights, vigilance, and marketing authorization. Now, these 9 steps of the regulatory authority can be assessed by the WHO Global Benchmarking Tool for the evaluation of national regulatory authorities. And there are 4 levels. Level 1, there are some elements of regulatory exist, but it's the basic level. Level 2, maturity level 2, is an evolving national regulatory system that partially performs essential regulatory functions. We heard earlier this morning that maturity level 3, and I will come back to that later, stable and well-functioning and an integrated regulatory system, and continuing improvement, maturity level 4. So all these functions here can be benchmarked against this. Then here you can see, we have seen it earlier, the development of the vaccine, pharmaceutical quality to non-clinical, moving to clinical trials, evaluation and decision to being made, manufacturing and safety monitoring. So the points that I mentioned below, the main role, and for this presentation, support vaccine development by defining requirements. The National Regulatory Authority also approves clinical trial and ensure compliance with manufacturing standards. Then they assess the data generated and make benefits risk balances for each of the medicines. And then after the licensing, conduct pharmacovigilance activities and continuously monitor the benefit risk balance over time. So, and I briefly go in each of these 4 steps now. So the first, support vaccine development by defining requirements. The authorities establish and enforce regulatory guidelines and standards for vaccine development, manufacturing, and testing. So these guidelines provide a framework for vaccine developments so that they adhere to rigorous scientific and ethical principles, and that they meet the international standards and provide maximum benefits with minimal risks. And these guidelines will cover from preclinical, clinical trial design, good manufacturing practices, and quality control measures. So before a clinical trial is even started, manufacturers can contact the regulatory agency and ask for scientific advice how best to implement the clinical trial. So that is the first step. So then the regulatory authority has to approve the clinical trial application. And the clinical trial, when you submit it for approval, it needs approval from the regulatory body, scientifically and technically, but you also need approval from a research ethics committee. So these both steps are very important. So this clinical trial application usually includes the data from the preclinical data from the laboratory animal studies ensuring safety and potential efficacy of the drug. Clinical trial protocol, so how will you conduct the clinical trial, needs to be included. Informed consent forms need to be included in your clinical trial application, and information on your investigational medical product. So the details about the medicine or the vaccine that is being tested, including the manufacturing and quality details. And then the Research Ethics Committee, as just mentioned, they give approval regarding the ethics components there. And you need the approval from both the regulatory authority and from the ethics committee before you can start the clinical trial. And yeah, this was just for completion, so that needs in your preclinical evaluation. So pharmaceutical quality studies, vaccine components and their purity will need to be included, biological activity, data on each step of manufacturing, data on the controls used to ensure that each batch of the vaccine is consistently of good quality, conditions for storing the vaccine, so these data are required for your clinical trial application, and also if there are any laboratory studies, in vitro or in vivo studies, and etc. This all needs to be included in your clinical trial application. So here you can see you need all the data on the preclinical assessment. Then you submit your file to the National Regulatory Authority. They assess the file, and if positive, you can start with your clinical trials. And that is both for each phase. You submit again your file to the regulatory authority before you can move to the next step in your clinical trial studies. And just for completion, but I think I can skip that here, I made a brief summary on the phase 1 trials, phase 2 and phase 3, but I think for this group I can skip it here, but it is in the slides available. So the regulator really wants the data then from the phase 3 trial, and we are really interested in the safety, efficacy, and immunogenicity. And regulators are very strict in all these data. When these data are submitted from the phase 3 trial, then the National Regulatory Authority will look if the data are complete, safety, efficacy, then they can go to a marketing authorization application. So the company submits all the data from the clinical trials to the regulatory authority. So they need the clinical trial results showing the efficacy, safety, the overall benefit risk profile, the pharmaceutical data, information on the drugs formulation, production methods, quality control process, the labeling and packaging. Proposed labeling information should be included, including the dosage instruction, indications, side effects, and other relevant details. And they need to have a pharmacovigilance plan. After the vaccine has been introduced in a wider population, we need to continue following the safety and effectiveness of the vaccine in the broader population. So these pharmacovigilance plan and risk management plans are very important at that stage, that they are agreed how it will be continued once the vaccine is on the market. So when that file is submitted, it's not immediately approved. There can be questions going back and forth for additional information or clarification before the marketing authorization is granted. And in the EU, the market authorization is submitted to EMA or to a national regulatory authority if the drug will be marketed only in one specific country. But if in the EU we want to market it for several countries, definitely has to go to EMA. So their final decision definitely is based on the benefit risk, that the benefits in protecting the people outweigh the potential side effects. So benefit risk. So as said, the regulatory authority would like to see the data of efficacy study, and the gold standard is your randomized controlled trial in phase 3. So absolute protective efficacy of vaccines by comparing the reduction in the incidence of the infectious disease in question versus the incidence in a group that received placebo. If there is an authorized vaccine, the trial may be designed to estimate the relative efficacy of the candidate versus the licensed vaccines with a non-inferiority design. And then case definitions need to be used for the primary analysis. So ideally, we want efficacy data, but sometimes that is not possible. And then we heard it earlier this morning, many times correlates of protection. And here I just, as a simplified decision tree, you can look at it. If large field trials, phase 3, are not possible, you need to look at how else the vaccine can be approved and regulated. So regulators are often looking at different ways of approval, and so the immune correlates of protection are important. And then I found this recent publication, serological threshold of risk reduction. And currently there were phase 2 trials on the GBS vaccine for maternal immunization in lower middle income countries, but it seems that phase 3 trials are not feasible or would take more than 10 years to have the sufficient data. So they are looking at other ways of approving the vaccine through this mechanism. So in the EU, so when we have all the data and it is approved in the EU, there can be full approval based on efficacy, as I just mentioned, or immunological data. There can also be conditional marketing approval, and that was the case for COVID-19 vaccines during the pandemic. Can be approval under exceptional circumstances, and then the EU also looks at, it was previously called Article 58, scientific opinion. Now it's EU for all to use vaccines for outside the EU, so like the malaria vaccine. So the EMA evaluates these data from the clinical trials from the malaria vaccine as well, although it's not used in Europe, but for other countries. Yeah, that is just briefly for information. Also data from EMA. So normally for each step you completed, preclinical, you give your data to the regulator, it will be evaluated, you move to phase 1, that is completed, you give your data to the regulator, they evaluate it, plus your protocol for phase 2. So it is really a stepwise approach in normal vaccine development timeline. Now, in the course of COVID, and we heard it this morning earlier, we see that the timeline is much shorter due to the rolling review of the data. So instead of really waiting of all the data being complete before you can move to the next step, as data were coming in, the regulator was reviewing it. And if that the first step was already promising, they could move to the next step, while of course still continuing having to provide all the data in the end. And this rolling review that was is normally based for conditional marketing approval, and then the final approval can be given when really all the data are complete. So then just staying briefly with COVID-19 vaccines, this was a recommendation also from EMA, and we heard about the platform for mRNA. So the new COVID-19 vaccines for this season in Europe with the new variant included. This approval is based on manufacturing quality and non-clinical data, given that data with the prior vaccines of a different composition supports the predictability of clinical immunogenicity and reactogenicity. So the platform for mRNA vaccines is approved, and now it's a new variant that is included. So based on previous data and on the quality, these new call it seasonal or yearly update from the COVID vaccine is based on this principle. Okay, so now the vaccine, the market authorization is there. The vaccine is being used broadly in the population, but still the work of the NRA is not completed yet. It continues throughout the entire vaccine life cycle, including pharmacovigilance, changing in the project, manufacturing or extension of indication of use, and any new evidence that impacts on the benefit risk. And so you can see the post-marketing surveillance and especially safety is very important. That will give a feedback loop, and the national regulatory authority needs to evaluate if new safety data are coming in, make a new benefit risk analysis, make new recommendations if needed, and take corrective actions if needed as well. And yeah, here a little bit more in detail. So why we need this, especially the safety data, because during the clinical trials, we will not pick up the very rare events or combination with other vaccines, co-administration. In clinical trials, not all subpopulations have been tested, so new groups can be included. So very important to continue to look at the estimate rates of occurrence of your adverse events in the local population compared with trial and international data. You really want to know that. And that can also say something on the quality of the vaccine is you suddenly expect a certain number of adverse events and you suddenly see much more. What could be the reason? You need to evaluate that, identify rare adverse events, detect, correct, and prevent program errors. Is there a problem with the vaccine launch or brand? That's what I said. If that is the case, then the NRA need to take corrective actions and remove the product from the market. And very important, good communication is there. If there is an increased AFI related to the vaccine or coincidental events, good communication is very important to the public to maintain confidence in your program and do not lose trust in your project, in the product, in the regulatory authority, in the Ministry of Health. And then also during post-marketing surveillance, you will also see, can there be maybe new aspects of vaccines to be evaluated, the route of administration, could it change, etc. So very important post-marketing surveillance. So the work of the National Regulatory Authority actually never stops, even if the vaccine is on the market, continuously evaluating safety, efficacy, and other things. So once the vaccine is on the market, yes, the regulatory authority needs to continue to follow up. Okay, so we have our data from the clinical trials. They informed national regulatory authorities. The vaccine is granted marketing authorization and can be used. How do we make the immunization policies? And here I briefly have the immunization policy advisory framework. We all have heard that at the global level, we have SAGE, a strategic advisory group of experts who give the global policy recommendations and strategies, and they support regional and national challenges. You have the regional technical advisory group on immunization, and then country, you have the NITEX, the National Technical Advisory Group on Immunization. Just briefly touching on this, I'm not going to say too much because I think Benjamin is giving a detailed talk on the NITEX, but they are important in the system here for developing the policy. So if you look, what is the role of the NRA and the NITEC? Regulatory approval by the NRA, and they look at quality, safety, and efficacy. That is what they are interesting. That is their data. And from there, the vaccine label or the package insert is being made. Now, the NITEC, they give a public health recommendation. So they also look at quality, safety, and efficacy data. But they will also look how can the vaccine be used for maximum public health benefit. So they also look at other aspects. So they also look at the local disease burden, the vaccine effectiveness, the impact, program feasibility. Is it easily to be integrated in the system? Cost effectiveness, public acceptance, equity, very important. And they use the guidance from the SAGE that I just said, can use the guidance from SAGE to inform these decisions. And we see that various recommendations are across countries due to variations in local factors such as disease epidemiology, healthcare system capacity, cost, public health priorities. As the vaccine might be licensed, but not all countries have maybe introduced PCV vaccine or rotavirus vaccine. They can make a prioritization in the country which is more needed than another vaccine. And then we see when we look at the schedule, some start a schedule at 6 weeks, other countries can start a schedule at 2 months. So this is country dependent, and that is the role from the NITEC very important in. So in 2009, there was a consultation looking at the interaction between the NRAs and the NITECs, and it seemed that the relation at that time was not very satisfactory, not sufficient exchange of information. So there should be a very good interaction between the NRA and the NITEC, and maybe a representative from the NRA can be ex officio member in the NITEC group, but I'm sure Benjamin will go more into details there. And I mentioned about the post-marketing safety surveillance. I think NITECs are very well placed to give input on how to develop certain studies on that. And if the NRA receives feedback on adverse events, and there is a change in benefit risk, immediately feedback needs to be given to the NITEC, to the immunization program. So communication between them, very important. And here, what I needed to inform the immunization policy, they look at, as said, at the burden of disease. Is it a public health problem? Are there other interventions that are being used in the country or not? So they're looking at economic and financial criteria. Of course, that's said, looking at safety, efficacy, and quality, that definitely, but they look at the broader spectrum. They look at the characteristics of the vaccine presentation. Programmatic, is it at the same time as maybe your 6, 10, 14-week schedule, or is it a completely new schedule? And availability of the supply. And then this was a recent publication from 2024, where they give guidance to the NITECs, so all the things that they should consider when developing the immunization policy. There were 7 categories. I just copied 2 here. So definitely vaccine characteristics. We've gone through that: safety, efficacy, and effectiveness. They will also look herd immunity protection. That's also something that they will take into their consideration. And the emergence of non-vaccine serotype, all these things they need to take into account. And then another one that I copied here, but you can look into the publication for the 7 categories. The accessibility of the target population, the availability of the vaccine, cold chain, is everything fine? Information management, and can you really manage to monitor your disease and your AEFIs? So these things, so the NITEC looks at a broader spectrum to inform their immunization policy. Then, having said that, I'm sure we all came across what off-label public health use of a vaccine. So that's using the vaccine outside the conditions approved in the NRA approved label. And I'm sure we all know examples. The HPV vaccine, currently many countries give it in a 1-dose schedule instead of the still regulatory approved 2-dose schedule. Yellow fever vaccine in a fractional dose during shortages. Influenza or COVID vaccine used in pregnant individuals. The 3+1 pneumococcal PCV vaccine, sometimes countries use a 2+1 or also 3 and no 3+0 then. So these are, off-label use of the vaccine. Why? The clinical trials, as we said, does not include all the target groups. And from the post-marketing studies, you will get more or new information on, for example, the vaccine schedule, the route of administration, or the target groups. Now, that does not necessarily lead to the label change, because then these data need to be submitted to the NRA, who review the data again, and then maybe the label can be changed. But these data can be used by WHO and SAGE to make off-label public health recommendations to use the vaccine. So we are not doing this quietly. There's very much evidence supported. So the post-marketing studies that assess the real-world effectiveness and safety that can be conducted by the manufacturers, and that can then lead to the label updates if submitted to the NRA, or it can be done by public health or academic institutions, and that, as just said, can then support the SAGE and the NITEC recommendations. So WHO and SAGE, they will only recommend the off-label use when there is a strong evidence of the benefit in case of supply shortages, and if simplified schedules can improve the coverage and the feasibility. So, and the WHO guidances will clearly note when the recommendations are off-label. Now, there are then some considerations for the countries, of course. What would be the liability of off-label use of a vaccine? Some countries restrict off-label recommendations. You cannot issue such recommendations. Other will protect if there is will protect the providers who follow the NITEC guidance. So if there is an off-label recommendation by the NITEC, that is fine for the country. So again, regular communication between the NITEC and NRA would reduce these off-label situations. And if new data are becoming available, it would be good that manufacturers ask to update the label as new information becomes available. And then again, communication again, if there is an off-label public health use of a vaccine, you clearly need to communicate why this is to the different population to really maintain the public confidence. Because if you suddenly issue a new recommendation, your communication needs to be there or you will lose trust in the whole program. Then briefly focusing on for Africa. So the data are found that less than 3% of clinical trials are carried out on the African continent. So that's a serious underrepresentation. Implications also go beyond Africa in terms of global health security. So, and I think it's very important to have the clinical trials here in Africa. I think we remember when rotavirus vaccines were first licensed based on data in high-income country, high efficacy. Then finally, when the vaccine was introduced here with delay, we saw that the efficacy was much lower. So you need to look at the local setting, etc. So very important to have the clinical trials here. And this slide, we heard it this morning already from a few other speakers on the right side. You see the type of the bigger the circle, the more funding is there for clinical trials in Africa. So you see that the majority is malaria, HIV/AIDS, COVID, TB. And the colored box there is actually a high burden of disease in Africa, but you see they're only very much underfunded to do the clinical trials here in Africa. Now, the other slide, this is from a publication from 2025, so very, very recently. You can see first as the first one is the funding for research, and then the next one on top is where the clinical trials in Africa take place, and you see the majority, South Africa, Kenya, Tanzania are most of them, and then the one down are the maturity levels. Are the maturity levels that I mentioned briefly earlier, done by the global benchmarking tool, and the ML3, 9 countries now in Africa. They are here in dark green mentioned. Now, especially what we heard earlier this morning, we want to develop more vaccines in Africa. So definitely your maturity level 3 is needed. But even all countries should have a good regulatory functioning, because pharmacovigilance, you introduce vaccines. So your pharmacovigilance is important. If you would do clinical trials or clinical trial oversight is important. So these 9 steps that I showed earlier for the NRA, certain are definitely for all the countries available. And of course, if you are a vaccine producing company, then you need all of them. So, but you see there is still a need to strengthen these regulatory systems, and especially around pharmacovigilance. There are several activities or initiatives going on in Africa, and clinical trial oversight as well. So then I briefly want to say a word on the Pan-African Clinical Trials Registry, PACTR, that is managed here at the South African Medical Research Council. So that's a regional register of all clinical trials conducted in Africa. And I think it's very important that it is registered there, that you can see which countries are doing what clinical trials, where is the expertise for certain topics. So it's an open access platform where clinical trials can be registered free of charge. And it's also part of the WHO International Clinical Trials Registry platform, the ICTRP, so that you can check different platforms are linked there, and PACTOR is there as well. So very important to register the clinical trials in Africa. Then I want to say a word on AVAREF. AVAREF was founded in 2006 by WHO, the African Vaccine Regulatory Forum. It's a network of African national regulatory authorities and ethics committees that uses harmonization and reliance as pillar for capacity building. And you can see here the different activities that they do. And what is very important here, they look both on the regulatory side, but also the ethics committee at the same time. So the approval process for your clinical trial can move faster ahead. So the joint reviews, ethics committees and regulators of participating countries can exchange and work together to speed up the review. And then finally, as we know, AMA has been established in Rwanda. You see again their functions, the same as what I mentioned earlier. They will also coordinate joint reviews of clinical trials, do inspections, and will also have a main role in harmonizing over the African region the norms and standards. So as key messages, clinical trials are the foundations. The regulatory approves the clinical trial, then look at all the data, granting the marketing authorization. The NITEC or public health institutes use this evidence and the real-world surveillance at the post-marketing to inform policy decisions. We need a good collaboration between NRA and NITEC, clinical trials in Africa, and the need for strengthening regulatory capacity. And that's the end. Thank you. And my voice.
Thank you so much for that talk, Dr. Deshay. And your voice did really well. Beautiful.
Yeah, I think it's collecting now.
Any questions in the room thus far? We don't have any online yet.
Thank you for the great presentation. My question is there another supranational agency that makes sure the regulatory agency is doing a good job?
Yeah. Yeah, it's on that or?
I have a question, but are you going to answer?
Well, there is the international regulatory harmonization. I don't know the exact, but they issue the standards and norms, WHO as well, and ideally that all need to be adhered. And as we heard earlier, if you really want to go for vaccine production and your national regulatory authority needs to fulfill all these criteria at the level 3 and 4, you also need to have your pre-qualification. And that, I think, is your quality and standards need to be good. Otherwise, you will not get your pre-qualification from WHO. Yeah.
Hi, I'm Nicholas from BioVac. Thank you for this. It was great. My question relates to a certificate of pharmaceutical product and how that applies to vaccines. I've worked with the FDA and EMA before, and for instance, if you can provide a certificate of pharmaceutical product, meaning it's gone through the EMA or the FDA, the MHRA, the TGA, it reduces review timelines, for instance, in Saudi Arabia from, I think, 13 months to 3 months, the review process. Do you ever see that playing a role in African countries? And is there an uptake on this certificate CPPs in Africa?
Good question. I actually don't know, but maybe from the colleagues here can answer that. But indeed, that would be a great help to move that forward. Yeah, that they and that EMA and AMA collaborate together. I think that is the aim. AMA is just starting up, so we will see, will they follow more or less how it works in EMA, the clinical trial registration and everything. But I think it's very important that they collaborate together, and information that is available, that it is shared among the different regulatory to move indeed forward. So yeah, thank you.
We have a comment online, Dr. Deshay.
Yeah.
Thank you. So this is a comment from Abdul. He had a question later on the role of national regulatory authorities. So his comment now is just we can keep talking about Africa's vaccine sovereignty, but without taking regulatory strength or talking regulatory strength, we are 1,000 miles away. And he just talks about the fact that if regulatory systems remain low and fragmented, investors lose confidence, and that there's a need for strong regulatory bodies to create predictability, trust, and quality assurance. So he thinks this is where AMA will come transform the regulatory space.
Yes, indeed, that's a very good comment. And as I said, only 9 countries are at maturity level 3. So that means that the majority of the countries, it's only level 1 and 2, and we need to move forward with that. Absolutely. So yeah, thank you for that comment. And hopefully AMA can play a huge role in that. Yeah, yeah.
Thank you. Our next question is from Ben.
Thanks, Karine. Excellent presentation, and hope your voice gets back to its usual. So, Karine, I think you possibly familiar with the case in Rwanda where they used Marburg, I think, vaccine against Marburg, which had not even gone to phase 3 clinical trial. And I wonder whether that is an example where maybe such collaboration between the regulatory authority and the clinical trialist and the policymakers. And so I'm wondering, how do you take politics out of that and bring all these expertise together and make a decision? It's a vaccine that hasn't been tested in phase 3, it's in phase 2, but we have a problem, we have an emergency, no other alternatives are available, let's use it. Do you see this kind of intersection between those 3 key players as very crucial in this kind of scenario?
Yeah, thank you, Ben. Yes, I think so indeed. For Marburg in Rwanda, they actually installed a phase 2 trial with the type of vaccine that was not being approved. I think there needs to be commitment from the government to do that, from the politics. And I think for everything that I have been talking about here, I think collaboration is so important. We are not alone. Often you might maybe think the regulators are very strict and say, oh yeah, now the company wants maybe a shortcut or this or that, but we have to listen why we are all there. And definitely in emergency, I think the regulatory needs to look at alternative pathways to, in this case, to advance it forward. So definitely, hopefully that is a good example for, well, we don't want future outbreaks, of course, but if it happens, that we can learn from these things, also from the rolling reviews during COVID, etc., to see how we can adapt certain regulatory pathways in certain situations to move ahead faster. And definitely political commitment is there also needed. Yeah, yeah. Thank you.
Okay, thank you for this very insightful presentation. I have one question. Actually, what is actually the difference between full approval and EUL? That, as we know, that billions of doses have been administered COVID under EUL, and does it be a concern for the community which is providing this dose, which is actually under EUL? So what's the real difference? Does it matter on any issues relating to like any issue? Yeah.
Thank you. So full approval, then the regulatory authority has all the data from all the clinical trials for safety, efficacy. And then if these data and the benefit risk is positive, they get a full approval. And I think in the EU, it's for 5 years. So that is fully licensed. In the case of COVID vaccine, it was more a rolling review. So not all the data were available, but we needed to use the vaccine because of the emergency. So that was valid for 1 year. And then if more data could come in, then it can move to a full license. That is the difference.
Thanks very much. This is Erisa from Uganda. I'm a member of NITAG.
Good.
And of course, at the core of the NITAG is evidence-based, because this is what we really rely on to advise government. However, we have poor quality local data, and you find that yes, at times we recommend that vaccines are introduced in the country, but then post-marketing safety surveillance is also poor. Case in point, during COVID, there was a lot of misinformation, and post-market, I mean the post-rollout surveillance was poor, and actually this fueled, vaccine hesitancy in my country. And of course, many of our Ministry of Health, they are more interested in the therapeutic effects of these vaccines, and they do not really pay a lot of attention to the other, what I'll call collaterals. Now, from your own experience at the global stage, what advice would you give some of these countries like mine? And I believe other African countries, some of them might also have the same problem. Thank you.
Thank you very much. Indeed, I think we need to keep the safety surveillance very much going on. And I think indeed some of the diseases where we are vaccinating already many, many years, the disease is sort of disappearing, but you get the vaccine and you get to see the side effects. So the balance of risk is actually a little bit changing there. So I think it is very, very important. Sure, you have to be open and trusted to communicate about it. You can say, yes, there might be small side effects, but if you do not take the vaccine, and maybe you are also some small side effects for 1 or 2 days, if you don't take the vaccine and you get infected, you might have lifelong disabilities or whatever. So I think that is very much important, I think, to remind them of the disease. What can the disease cause as well? Yes, and vaccine hesitancy, and definitely with social media now, it spreads so fast, and it is specific for certain vaccines. Other vaccines are accepted, but I think we need to bring the positive messages. What is the benefits of the vaccine while being transparent? There might be small side effects, but that is closely followed up. And please think what would happen if you do not take the vaccine and you get ill. I think that's maybe the message that we forget to give these days.
Thank you. This is more based on your ethics and regulations during your clinical trials. So I believe that most of the people who would enroll for clinical trials are those from disadvantaged backgrounds, mostly maybe for the benefits like the allowances that they get when they participate in these clinical trials. How do we maintain the balance on controlling that so that people just don't enroll in clinical trials just for the benefits besides informed consent? Or maybe you can delve deeper on the informed consent side. Thank you.
Yeah, indeed. That is definitely an effect if they get paid to participate in a clinical trial, that might be the main reason. But I think informed consent is very important here. And I think the ethical approvals for a clinical trial are very strict these days. And I think the informed consent needs to outline clearly what it is, what will the benefits and the risks. So, and they have to sign it. And I think you cannot fully exclude it that people might participate for the wrong reasons. But I think informed consent and the role of the ethics committees is very important here to safeguard it as much as possible.
Thank you. We have one more question.
Shall I go? Okay. Thank you, Karina, for your presentation. And I really appreciate how you have highlighted the NITAG function. I'm a NITAG member from Namibia. I do have I think it's one question for you. I'm a strong believer of community-led solution. And there's always something in my place where I come from that do we have some of the scientists who really encourage community indigenous knowledge? And if they so, can you perhaps give me if there's any other vaccine which was led with indigenous sort of like background and it went through some clinical trial, especially for Africa, because I reserve some of my comment while I'm raising this, but I just want to hear.
Good question, but I think I cannot answer
Okay.
that one.
Or maybe
If anyone else from the colleagues has.
Maybe just to clarify, do you mean progression of something that's being developed based on indigenous knowledge? Okay. So the formulation is taking indigenous knowledge, incorporating it, and has it then taken it into clinical trials, is the question.
Bodies, do we have a space where this indigenous knowledge can be, accepted, regulated, and getting into the development of the vaccine?
I think you want to comment on that? Yeah.
Currently at Africa Health Research Institute, Professor Neba Kaleni had invented or came up with the herb that he had dreamt about. And it went through the lab work. Currently, it has passed all the SAPRA requirements, and it's now in the phase of encapsulating it so that it can be tested in clinical trials.
Hi, sorry. For the regulators, this has always been simple. To register something, it must go through certain threshold of clinical trial. Where the knowledge came from is irrelevant. And so that means whether it came from, pretty much lots of stuff. If you've got salicylate, for example, it was like the willow bark that someone chewed long ago, right? And then realized that the pain, and we do have a number of compounds like that would have had their origin in indigenous. For it to be registered through with a regulatory stamp of approval, it has the same threshold of being able to enter it. So you can have any knowledge, indigenous knowledge system, but you have to take it through a clinical trial because then it has passed muster in terms of safety and efficacy and all other things in order to protect your population. So that's just how it is. Sorry? No, well, the search is the same. That means it doesn't matter where you got your knowledge from, whether from your grandmother, but if you take it through a clinical trial, you can register it. That's how the regulators have always worked. If not, people have managed to get them to be sold as supplements and other things that, like the way vitamins and other supplements go through, but they're not taken through the same regulatory system. For you to be a prescription that is regulated, the threshold of how to get it done it has to go through a trial that then prove efficacy and safety with very clear guidelines of how that came about. Yeah. Sorry.
No, spot on. Many examples of development based on prior knowledge. So thank you. It seems like you don't actually want to go to lunch. You want to just keep talking. We can do that also. Okay, just kidding. But thank you so much, Dr. Doshay. Thank you. What a fantastic session. And thanks to you all also for your engagement and questions. Enjoy lunch.
We're on. All right. Good afternoon, everybody. All right. Good night, everybody. Okay, because when I said good afternoon, I didn't get any response. So thank you. Oh, that's more like it. Thanks, Bev. All right. Good afternoon once again, everybody. And I'm just trying to check if our speaker is online. Rose, can you hear me?
Yes, I can hear you loud and clear.
Oh, great. So it's good to have you here with us. Can you just give me additional 10 seconds? I need to do some mass mobilization and ensure that everybody that should be in is in. Or maybe I could get a volunteer or two to help mobilize the masses. Thank you. Thanks, Julia. No, I can always count on you. I tell you, this is the fun session, the session after lunch. We are all heavy, drowsy. All right. So once again, good afternoon. And my name is Elizabeth. I'll be chairing this session. And I'm glad that our next speaker is already online, all the way from Kenya. So you had better pay good attention. Otherwise, there will be no Kenyan tea after the session. So Rose, without wasting time, I'll hand over to you. You could take the first minute or two to do a proper introduction of yourself. Thank you. Rose?
Yes, I'm here.
All right. The floor is yours.
Yeah, and thank you for the kind introduction. So I'll put.
Yeah. Excellent.
Yeah, I'll put my video so that everyone can see me, but I'll switch it off for the sake of presentation. Yeah, so my name is Rose Jalango. I work for the Ministry of Health. I'm the immunization program manager and also an alumni of the program, Africa Vaccinology course. I think I did it in 2018. So again, it's a pleasure coming back as a speaker, just to share insights about malaria vaccine introduction, having worked closely during the pilot implementation, from the time of implementation, especially in Kenya, and working across with other countries. So I hope that from this, we can learn lessons about the malaria vaccine. For the countries who've not introduced, they'll have something to take away. And for the ones who've introduced, we can share ideas on how best to strengthen the uptake. So will you be able to project the slides from your end?
No, Rose. How about you share your screen?
Okay, let me do that.
Thank you.
Nancy.
So we are equally working on it from this end. Salim, the slides are here. Okay. So while we wait for the IT to sort out the slides, for a virtual chocolate, who can tell me what Rose will be telling us, Saba? Malaria. Great. One virtual chocolate. All right. And like she said in our introduction, for a high five, that's not virtual. I can actually deliver on that. What is our hope for the countries that are yet to introduce the malaria vaccine? To introduce. High five. Now, for a hug to the ladies. All right. The slides are up. Rose, are you still with us? Rose.
Perfect, I'm here. Thank you.
All right. Over to you. The floor is yours.
All right, thank you. So I'll be taking us through. Okay, so I'll put it as sort of a journey from how it started. And I have to acknowledge even before I start that I've had to borrow heavily from WHO. We work together in terms of the slides. So I'll be giving a presentation overall about Africa, but again, giving specific examples about Kenya concerning the malaria vaccine introduction. So next slide, please. So in terms of the outline, we'll discuss a little bit about the malaria burden, the pilot implementation, the vaccine rollout, and the lessons that we did learn from the sub-national rollouts across the countries that did the pilot and also the subsequent countries. Next slide. So I know all of us have heard about malaria, and again, it remains a primary cause of childhood morbidity and mortality, particularly in Africa, with a lot of deaths occurring under the age of 5. Yes, so we see that it's still a problem or a challenge in many African countries. Next slide. So particularly in Kenya, it's a significant public health problem. With more than 70% of the population at risk of malaria, and this is especially children and the pregnant mothers. So to just highlight how much of a concern it is, it accounts for more than 50% of outpatient visits, especially in areas that are very endemic. And then the transmission level or geographical coverage of malaria is different in Kenya. So I'm sure also in every other country, they have different zones when it comes to the distribution of malaria. So in Kenya, we have the 5 epidemiological zones. We have the endemic zones, the seasonal transmission zones, the epidemic prone, and also the low risk zone. So particularly, we have an interest in the endemic zone, which is the western part of the country that has been highlighted in red, where the prevalence is more than 10% in terms of the risk of getting malaria. And then it's something that is always throughout the year. Next slide. So even as we discuss how malaria is a challenge, is a public health challenge in most of the African countries, there has been some work, especially behind the scene, and this work has spanned for more than 30 years in terms of looking for a vaccine for malaria disease. So it's a disease that has been quite here with us, but we had not found, we didn't have an intervention in terms of a vaccine, but we had other interventions that were in place. So for example, spraying, sleeping under insecticide-treated mosquito nets. But these interventions could only control the disease up to a certain level. And from that level, the fight against malaria actually plateaued. And that is why there was need to get other additional tools to be able to fight malaria. So on this slide, we just show the amount of work that has been put in place to be able to come up with the first malaria vaccine, which was the RTSS S001, as it is known. And so the final stage before we started implementing it is that we had the phase 3 clinical trials that showed some promising data concerning the efficacy and the data of malaria vaccine. So what I do know is Kenya was among 11 countries that participated in the clinical trials. And after those clinical trials, there was a WHO recommendation that countries should do pilot implementation of this vaccine. Next slide. So it was in 2015 where we have these global experts of both immunization and also malaria program that came together to recommend a pilot implementation of this vaccine in real world setting. So the vaccine, the reason we had the pilot implementation was one, to find out whether it's feasible to reach children with 4 doses of malaria vaccine, and then 2, to evaluate safety of the malaria vaccine, again with emphasis on 2, on meningitis and cerebral malaria, and then 3, to see the impact on mortality and especially on severe malaria. So 3 countries did apply for this pilot implementation, and that was Kenya, Ghana, and Malawi. And that is why in most of the slides we'll keep a reference to them, because those were the countries that did have the pilot implementation implemented. Next slide. So because it was a malaria pilot implementation, it basically meant that the vaccine was introduced at a subnational level, targeting areas with high endemic for malaria disease. But again, there were additional evaluations that had to be done by other partners. So we had a consortium of different partners working with the 3 countries to be able to evaluate the malaria vaccine in actual setting. So we had the GSK providing the malaria vaccine. We had the ministries of health of both Ghana, Kenya, Malawi rolling out the vaccine. And of course, we had funding from various funders and technical support from both WHO, PAC, UNICEF, and all the other technical partners. So that is why it was called the Malaria Vaccine Implementation Program, because it was set up for a specific cause. Next slide. So most of the pilot implementation was introduced in 2019 and ran through to 2023. So what happened in both Malawi, Ghana, and Kenya is that we looked for areas which are endemic for the malaria disease. So for example, in Kenya, we had the western part of the country participating in the pilot implementation. So in this pilot implementation, so I'll give you an example, the whole western part of the country, which consists of 8 counties or 8 districts, were found to be endemic of the malaria disease. So we took some of the sub-counties to act as the counties to initially introduce the vaccine, whereas the other sub-counties were acting as a comparator. So we had some regions introducing the malaria vaccine, whereas others were acting as the comparator, and this was across all the 3 countries. We had this so that all the data that was coming from these countries would be comparable at the end of the pilot implementation program. Next slide. So I know when we were talking about the comparator, the first some counties introducing initially and then the subsequent countries introducing later, to most people or some of the communities thought that it was a trial, or it was a trial or a clinical trial, but it was a malaria implementation flagship program because it was being conducted in a setup, in a population setup, and not in a controlled setting as with the clinical trials, how they are conducted. Next slide. So in terms of the introduction, So we had the 3 countries introduce the vaccine. So of course, the malaria vaccine was introduced through the immunization programs because they coordinate vaccination services, but they worked closely with the national malaria control programs. Because remember, when you're introducing this vaccine, it was not supposed to be a magic bullet or work in isolation, but it was supposed to work in the context of other available tools that are already in place to fight malaria. So it was integrated into the routine schedule. It was integrated at the service delivery, in that the nurses who gave other routine antigens were the same nurses who are giving the malaria vaccine. It was integrated into the recording and reporting into the KH and the DHIS. And when it comes to community engagement, all the communities were engaged before the implementation of this vaccine. And then in terms of storage and logistics at the lower levels, it was stored with other vaccines in the schedule. So how we were working with the National Malaria Control Programs is that we had integrated messaging that as much as those children who are receiving malaria vaccines, they still needed to continue with the other interventions. So in case they had fever, they had to go to the hospital, they had to continue sleeping under treated insecticide mosquito net. And then the areas that needed spraying, they continued with the same intervention. Because remember, these interventions were supposed to be very complementary. Next slide. So specifically for Kenya, we did launch the malaria vaccine in September 2013. And what we saw about this is that there was political goodwill in terms of the pilot implementation. This was launched by our cabinet secretary then, and then it was supported by WHO, UNICEF, and then it was also a community sort of launch. So that at least the community could be integrated into the whole concept. Next slide. So I'll quickly go over this. So this is how malaria vaccine was introduced in Kenya. We had said that it was introduced in the western part of the country, 8 burden counties, and then it was in selected sub-counties, which are purple in the map. And then the other pinkish color were acting as a comparator. So we rolled this out in the immunizing facilities so that it could be integrated with the other vaccines. Next slide. So in terms of the schedule, so again, remember this was supposed to fit within the various immunization schedules. And we had guidance from WHO on how to schedule the vaccine. So the vaccine, the first dose was supposed to be given as early as 5 months. And then the interval between the first 3 doses was 4 months. And then the last dose was supposed to be given 15 to 18 months after the last schedule. So again, different countries worked within their context to be able to come with that schedule because there's a lot of flexibility. So in Kenya, you can see the first dose was given at 6 months, the second dose at 7 months, the third dose at 9 months, and then the fourth dose was scheduled at 24 months. So again, this was supposed to coincide with other childhood interventions. So for example, nutrition, administration of vitamin A, weighing, or even co-administration with other vaccines. So that's why you see different countries actually adopted different sort of schedules to be able to roll it out. Next slide. So we are privileged during the pilot implementation to have additional support to be able to generate evidence-based high-quality communication materials, to communicate to the different stakeholders, to communicate to the community. And these were the posters and IEC materials that were found across different setups. Next slide. So again, materials were translated into local dialect. So depending on the country, so in Kenya, we had different dialects in terms of from the radio stations to the posters. And this is just to connect to the community. So any intervention will actually require that you connect with the community in terms of the materials and also the communication that goes out to them. Next slide. So we had our first child who received the malaria vaccine in 2019, and actually we've kept track with the parent for the last 6 years. And the child is actually now in grade, it's actually doing well in grade 1. So what we reported from the mother reported is that the child did not have episodes of malaria as a child, and also has never been admitted or had any blood transfusions as a result of the malaria vaccine. So this again is coinciding with what was gotten from the other 3 countries. Next slide. So you remember we had the malaria vaccine implementation. So that was from 2019 across the 3 countries. And then it was running from 2019 to 2023 in Kenya, Ghana, and Malawi. So the data from these 3 countries, and again, this is a summary of it, is that 1, there was the vaccine was confirmed to be safe. Remember, one of the things we were evaluating was on the vaccine safety issues. So the vaccine was confirmed to be safe across the 3 countries. And then the vaccine had high uptake and high impact. And the reason for this is that the community was aware of malaria. The community was aware of the effects of malaria. They'd either seen a child die out of malaria, admitted, transfused. And so when we did introduce the malaria vaccine, it was something that the caregivers were actually willing to take up. So in terms of vaccine attributable reduction in all-cause mortality was at 13%. And then in terms of severity, reduction in hospitalized severe malaria was at 22%. So we did not see a reduction because you see we had the different evaluations going on at the same time. So there was no reduction in other control measures for malaria, that's insecticide use or care-seeking behavior or uptake of other vaccines. So again, we found that where the other preventive tools had not reached, we had some places where the malaria vaccine was actually reached far more people compared to the other traditional tools that are used for malaria vaccine. So it is based on this data from the 3 countries that there was a recommendation from WHO to be able to scale up the malaria vaccine in the same similar settings. Next slide. So again, one of the things that was to get highest impact from the malaria vaccine, it has to be used in a context of the other malaria control tools. So as we can see from this slide, the highest impact is actually achieved when a mix of malaria interventions are used together, including the malaria vaccine. Next slide. So of course, we had quite a number of lessons from the 3 pilot countries. But I'll just go through some of the key lessons that we did learn, is that number 1, for the introduction of malaria vaccine, use of the existing systems to be able to roll out the malaria vaccine was seen as a success, instead of creating parallel programs to be able to roll out the vaccine. So we had so that means integration with service delivery, integration of the data collection tools, integration with the logistics information management systems. That also was really critical. And then number 2, we had some sort of health system strengthening benefits to the countries that did introduce malaria vaccine. And that came in the form of either purchase of new cold chain equipment that led to as a sort of system strengthening in a way, because new cold chain equipment purchased means that more capacity to store not only the malaria vaccine, but other vaccines. So the third thing that we did learn is that for rollout of this vaccine, countries need to work with the national malaria control programs, because remember, these interventions are supposed to be complementary and none is supposed to substitute the other one. And then the fourth thing that I do remember is that community engagement. In any intervention, community must be at the heart of the engagement, because once you get it wrong with the community, it means there'll be no uptake from the community. So we did engage a lot of the community, got feedback from the community, and also integrated it in terms of our policies. So again, this information on the lessons learned from the pilot implementation is available and has actually been published across all platforms. Next slide. So based on this, based on the feedback or based on the findings from the 3 countries, that is when in October 2021, so WHO recommended malaria vaccine for prevention of malaria, falciparum malaria in children across African countries. So it was sort of the base from and the findings from these 3 countries that opened the leeway and to introduce malaria vaccine across the African countries, especially in areas with high endemic burden. Next slide. So by the time of, so these are the 3 countries, these are the 3 children, and then these are the numbers in terms of the children who have received the malaria vaccine. So initially remember that when it was a malaria pilot implementation or a flagship program, we did get, the 3 countries were supplied with vaccines from GSK, but after the pilot implementation in 2023, what happened is that Gavi, the Vaccine Alliance, was able to now finance the countries to be able to introduce the vaccines. So both Kenya, Ghana, and Malawi were able to continue the vaccination program, again, through support from Gavi. So it was again after the findings from these 3 countries that the Gavi board approved funding to be able to support other African countries to be able to implement the malaria vaccine. Next slide. So as we speak, and again, based on those results, we do have 24 countries that have introduced the malaria vaccine. So the introduction of malaria vaccine is actually subnational. Again, countries are working with their malaria control programs to be able to see the areas with the highest burden of the disease for them to introduce the malaria vaccine, because it has been shown with those specific areas, it has the highest impact. So again, what I do remember is that at the time when this was being made publicly announcement, many countries, African countries, expressed interest in terms of introducing the malaria vaccine. And one of the challenges that was there was actually an issue with the supply. So the countries that introduced initially, WHO had a cap in terms of the number or the number of doses that every country was supposed to get, because the demand for the malaria vaccine vis-a-vis the supply was a mismatch. Next slide. So again, the good news is that there were other candidates that were being worked on for the malaria vaccine, and that's why we had an additional malaria vaccine, the R21, which was recommended in October 2023. So this has come to bridge the gap that was there initially as a result of the demand challenge that was created initially. So right now we have in the market the RTSS and also the R21 malaria vaccine. And again, WHO has given guidance on how countries can go about it in terms of which one to use based on how they support each specific country. Next slide. I think it won't be fair for me not to talk about the uptake in terms of what were some of the challenges and some of the data that we had from the pilot implementation. So remember, this was a 4-dose vaccine schedule, injectable. And initially we had concerns about the uptake from the community. How we wondered whether it was feasible to give a 4-dose injectable and the community to accept it and also take it up. But the good news is that what we found that it was feasible to give the 4-dose schedule for the malaria vaccine. So as you can see, we used to approximate the uptake of the first dose of malaria vaccine, and then you compare it with a 3rd dose of pentavalent given at 14 weeks. And then we also compare the second and the third dose with the antigens given at 9 months. And what we did find again across the country is that they were the level of performance or uptake was quite high even for the malaria vaccine. So the challenge that was there was the fourth dose that was given in the second year of platform of life. And again, the challenge is not unique only to malaria vaccine, but you've seen other antigens, for example, measles, rubella, given in the second year platform of life, the uptake is low. And again, that just points out to the caregivers or the health system in terms of strengthening the uptake of antigens or interventions given in the second year platform of life. So what malaria vaccine has done is that it has strengthened the second year platform of life in terms of uptake of vaccines. So it has opened a leeway in terms of giving other interventions in the second year platform of life. Next slide. So for the countries launching the malaria vaccine, remember we as per November, it's 24 countries that have so far introduced the malaria vaccine. It's still, again, an emphasis for them to work with the National Malaria Control Program and again to use vaccine as part of the package control measures. So you'll find that there's a lot of integration with the malaria control program, especially during community engagement and also when it comes to key messaging to the community. Next slide. So in terms of the schedule, you'll find that different countries have different schedules because WHO guidance on how malaria vaccine should be introduced allows a lot of flexibility in vaccination schedules. And again, this is to allow countries to align with their programmatic needs and also to really adapt it to their local context. You'll find that other countries would give the second dose at 15 months, 18 months, 24 months. And again, this is based on the NITAG recommendation and also programmatic considerations that are very specific to the countries. So you'll find that with this flexibility, you'll find that there's a lot of leeway or leverage for the countries to be able to leverage on. Next slide. Yeah, so I know the first three countries have a lot of experience when it comes to the new vaccines introductions, that Kenya, Ghana, and Malawi. But again, there are a lot of opportunities that have been created across the board for countries who are yet to introduce, for countries who've just introduced, for them to learn and share, to learn from other countries and also share experiences. So there are different workshops, webinar platforms, and regular coordination meetings that have been put in place to be able to support these countries as much as possible. So any country that is introducing the malaria vaccine or any other vaccine, they can leverage from the experience of these countries to be able to move this ahead. Next slide. So I'll summarize it by just concluding on where we are as a country. So I know we are still in the western part of the country in terms of the administration of malaria vaccine. But initially, when we had started, not the whole part of the western counties were introducing the malaria vaccine. And this is because they had not reached the threshold of more than 11% to be able to introduce the malaria vaccine. So what we've done is that we've scaled up the malaria vaccine to the whole western part of the country block. And what we do plan to do is that currently we are currently administering RTSS, but we do plan to switch to R21. And again, these vaccines are comparable in terms of their performance effectiveness. But again, there are different things that countries look at before they introduce or switch vaccines. So at the point where we are at, we are looking at the issue of cost, and that's why we are going to be switching to R21 come Q1 of 2026. So I think I'll stop there, and I'm happy to receive any questions, comments, clarifications from the presentation. Thank you.
Hello. Thank you so much, Rose. Thank you. That was quite informative. Any questions for Rose?
One on this side.
Yes. And another one here.
Thank you very much for.
I go.
Okay. You can go ahead.
Thank you, Ravon. Thank you very much for the presentation. I have two questions. The first one is about measuring the impact of the malaria vaccine with regard to the reduction of morbidity and mortality. How did you go around this? Because we have this challenge in Sudan, where with weak health information system, we are unable to really capture the impact. Maybe we thought about doing some cohort studies, case control, or things like this. So if you can share some of your experiences. And the second one is about the forced dose. As we have seen from the graphs you presented, we see a very low uptake of the forced dose, but I could see that you succeeded actually almost in the 3 countries to go around this. So what are the solutions you did in order to increase the coverage of the forced dose? Thank you.
Thank you. Rose, you want to answer that quickly?
Thank you for that really good question. So in terms of impact, remember, we can do it in 2 ways. So initially, we had the 3 countries that were supported even to do the impact. There were additional support from the evaluators to be able to measure the impact of introducing the malaria vaccine. But routinely, what again the country has done is that we work with the malaria control program, and they do their surveys every 3 to 5 years. So from the survey that was done before introduction and after introduction, there's a clear reduction in terms of the burden of malaria disease in those specific countries. So the surveys being done by the malaria control program or even the routine surveys from KDHS, demographic health system survey, that could be able to give an indicative of the impact that from the vaccine. But again, there's also, we work with our researchers. We have KEMRI in the country that we do work together to be able to demonstrate impact of any intervention that has been introduced. So on the second question about the second dose, yes, it was a challenge. We started quite low. But again, with the working together with the community, we were able to see some of the gaps that are there in terms of one, our healthcare workers were not really emphatic on telling the mothers that beyond 1 year, we still have to have those health visits to the health facility. So we had to strengthen the interpersonal communication skills, had a lot of webinars with the healthcare workers, and also created packages for health education at the health facilities. Secondly, we leveraged on the multiple activities in the immunization program to be able to reach out to these children. So for instance, we carried out several campaigns, either polio or measles rubella. And during these campaigns, we also leveraged in terms of reaching out to these children who had not received any intervention in the second year platform of life. The third thing that we did is that we also strengthened the periodic intensification of routine immunization activities. We leverage on those activities to be able to reach out to this particular age group. But also at the service delivery level, intentional outreaches with a view to just integrate and look at it from a broader perspective that children, pregnant mothers, teenage girls can be able to come to be able to access the package that has been given during an outreach. So those are some of the ways that we did. But again, the increase is actually very gradual. So over the last 6 years, for example, in Kenya, this is when we've been able to reach the 50% mark. So it's something that's quite worth trying.
Thank you so much, Rose. Subsequent questions, let's keep them short, like the malaria shot itself, so that there can be more uptake of questions and answers. Thank you.
All right, thank you very much, Doctor, for that wonderful presentation. I saw the malaria vaccines is given a series of 4, and I see a serious variation regarding the various countries. Knowing vaccines are normally given in series, and then why that they having that differences, and if that difference is there, been there, what could have been the impact of ensuring or achieving a sustained immunity over time. So I want you to make, that's what my understanding is, specific times will be given so that you maintain that sustained immunity over time than given a different time point based on the country context. Thank you.
Thank you for that question. Rose, please.
All right, thank you for that question. So in terms of the scheduling, so WHO gives some guidance, again, based on some background work that has been done before they release the scheduling. So the scheduling, there is no much difference. So they tell you that dose one should be given anytime from 5 months. So actually, basically, the first 3 doses should be given within the first 1 year, between 5 months and 12 months. And then the 4th dose is supposed to be given in the 2nd year platform of life. So again, based on the studies they've done, they've shown that in terms of effectiveness or even the impact is actually similar, despite the variation despite the variation across countries. So there's a lot of flexibility in the scheduling, and that is based on scientific advisory given by WHO.
Okay, thank you so much, Rose, and congratulations for the 3 countries. This is Fatima from South Sudan. We have introduced malaria vaccines last year, and we are doing scaling up. When we started, the acceptance rate was 73%, and the uptake for the first dose was very high. But then subsequent doses have been very low, especially the 4th dose. The compliance was not there. When we also, you mentioned communication with different languages, but what was your communication strategy? Because when the vaccine was introduced, people were thinking that no malaria at all. And this was the thinking, because when you say no, you are giving polio vaccine, that means no polio. But when they realize that they have still to use the other intervention for malaria, like sleeping under mosquito net, and steer clear for mosquito around the houses for the mosquito breeding areas, the acceptance level went down, plumbing, and we are facing some challenges in communication. I think this is an issue for us now, how to communicate around this, especially, and I want to know your experience in Kenya in this area. Thank you so much.
Thank you, Ruth. Is there a possibility of sharing the communication materials that seems to have worked so effectively in Kenya, probably for translation into South Sudan's local language?
Yes, we can. We can share as much as possible, but maybe just to highlight how again we went about it when we started the introduction. So I do remember, especially at the inception time when we were doing the communication materials, the IEC materials, what we did initially was to design materials at the national level and then take them to the community to be able to communicate to them. But one of the feedback, as much as it sounded simple, is that the people at the community will tell you that in this poster that you put here, this is not how our homes look like. So can you adjust this home? Can you put a chicken here? Can you put a goat here for it to speak to them? So that's one of the things and the feedback we did incorporate. The second thing that we did is that at the health facility level, we created materials to be able to, there's health education sessions by the healthcare workers. So repetition, repetition, repetition. So anytime they come for an intervention, we had a way of outpatient clinics repeating or communicating messages of other health interventions, and including malaria vaccine that was just being introduced. So in terms of the dropout, yes, I do acknowledge that even in the routine antigens, initially, especially, let me give an example of pentavalent 1. The first uptake is quite high, but again, subsequently, you do have dropouts of children along the way. But again, this is because of different reasons. Either mothers have moved from one facility to another, or they delivered at their aunt's place, now they're going back home. But again, this is supposed to be less than 10%. What I know is that the difference in terms of dropout between the 3rd dose and the 4th dose is actually quite high. And the reasons we got from the community again is by the second year platform of life, the mother is either pregnant or is involved in other economic activities, has forgotten that they're supposed to be taking their children to the health facilities. So it requires a lot of repetition and also continuous creating of awareness to be able to reach out to these children. So I know when we introduce a vaccine, there's a lot of pimp and pump in terms of communication. But again, later we just let it be in terms of let it slide and go with the other communication, or they'll just figure it out. But what we've learned is that it requires constant reminders, constant communication, either through TV, radio stations, or the local channels that we do have. So the other thing we learned is that Kenya is very vibrant when it comes to the social media space. So we leverage on the social media space to be able to do that communication. And also we leverage on the community health structures. So using the community health volunteers and the community health promoters. So in Kenya, the community health volunteers move from household to household. So they have the vaccination schedule. So if a child has not completed their schedule, they are able to refer to the health facility to be able to get the vaccine. So what I'm alluding is that there is no one way of making sure that we create awareness. So we use a mix of tools or a mix of available ways in our countries to be able to reach to these children. So congratulations to South Sudan, you've done a good job. Remember Kenya, before we reached the 50%, it took us 6 years. So again, doing the work, being patient, whatever the rise may not be very drastic, but for sure with that consistent application of those various tools, you'll be able to see a gradual increase of the second dose, of the fourth dose of the malaria vaccine. But again, we'll be able to share any materials that you need through the organizers to be able to share with the team.
Thank you so much, Rose. Please, we have just 3 minutes left or less. So make your question short and sharp.
Thank you so much, Rose. Mine is actually on the fourth dose of the vaccine schedule. We actually introduced a malaria vaccine last week. I just wanted to learn from you from the time there's this cohort that we're leaving out every time we're introducing. I can see from the schedule you shared, you introduced the first cohort was the multi-age from 5 to 7. Then the fourth dose was at 22 months. I just want to learn if there's anything that you saw where the age group that we're leaving out, like in your case, from 8 months to 22 months, if they maintained without receiving this vaccine? And also, how did you monitor the first cohort multi-age to ensure that they've actually received the fourth dose? Our worry is the mix-up. Maybe the healthcare worker would be thinking, because this child is even after 10 months, they can receive the dose. Meanwhile, we have this cohort that we need to monitor and ensure they receive all the 4 doses. Thank you.
Rose, please, before you respond to that, Seppi, I understand there are quite a number of questions online so that Rose can take them all together. Thank you.
Okay, yeah. Thanks, Rose. There was someone who wanted to ask a question, so you can just unmute yourself and ask if you still have a question online.
Okay, thank you, Rose. Mine is just with regards to the switching you talked about in the last point of the presentation. I understand the R21 has some advantages aside just the cost, I think even in terms of efficacy from what we know in the literature. My question is, how do you intend to manage the dual product deployment? I understand you still have some of the RTSS in stock without fragmenting the schedule. Or how do you intend to conduct the switch? Thank you.
Ask another question? No?
Rose, please, could you in 10 seconds respond to both questions? Our next speaker is waiting.
All right, let me try my best within the 10 seconds. Excellent questions. So number one, in terms of switch, not only for malaria vaccine, but any switch within the immunization space, what you do is that if you have a date for switch, you make sure that the product, for example, the RTSS, is fully utilized within the country before you switch to another product. Again, this is just to avoid having two products in the country that can bring confusion to the healthcare worker. So we do a lot of monitoring of stocks and management before now we switch to the next product. It applies to all the switches within the immunization program. So the other question is on the scheduling. So for the scheduling, I know Kenya took 5 months for dose one, 6 months for the first dose, second dose 7 months, and third dose at 9 months. And the reason for this is that we wanted to create an opportunity for children who have not received the malaria vaccine between 6 months and 9 months. When you first encounter them at 9 months, they are eligible. So again, the schedule is not fixed. And again, we work as the other antigens. So for example, you say, so for Kenya specifically, if any child under 5 who has not received malaria vaccine comes to the clinic, we have a policy on how the children can be caught up. So this is a whatever we call the big catch up, so that it does not only apply to malaria vaccine, but it applies to pentavalent also and other antigens, so that you can find a child who comes for the first time at 6 months. You do not deny them the vaccine because they did not come at 6 months, but you start them the vaccination and catch up, schedule them for the other subsequent doses for them to be able to complete the schedule, similar to malaria vaccine. So if you have a child who comes for the first time at 12 months, you start the first dose and then you schedule the others according to how they're supposed to be administered. So thank you very much.
10 seconds.
I know.
Thank you
But
so
thank
much,
you.
Rose. But I understand that there are still more questions on the online platform and that you do have access to it. So
Okay.
while the second speaker is speaking, could you please respond to those questions? And if possible, hold the line if you are available after the second speaker, then the questions, if there are more questions for you, we'll direct them to you. Let's jam our hands together for Rose. Thank you, Rose, for that excellent presentation. And then we go quickly to the next speaker. Is a very wonderful gentleman, also from Kenya, by the name Kenneth. Please, do you mind? Sorry, we borrowed some seconds from your time, but where is Kenneth?
It's not him.
All right, one clap, just one. You're welcome.
Thank you so much. I think there was a slow reaction because it's Lawrence, so maybe that's why. But I'm going to ask for your attention. You know, when you come mid-afternoon on day 3 of a course like this, you might have some what we call half moon eyes. So I'm gonna take some 2 seconds for us to do a quick exercise. If I say A, we put up our left hand. If I say B, we put up our right hand. If I say X, we put up our left. And if I say Y, we put up our right. You see, the eyes, exactly, the eyes have started widening. So I'll repeat myself. If I say A, we put up our left hand. If I say B, we put up our right hand. If I say X, we put up our left. And if I say Y, we put up our right hand. One last repetition, because I can see eyes getting wider again. If I say A, we put up our left hand. If I say B, we put up our right hand. If I say X, we put up our left hand. And if I say Y, we put up our right hand. Are we good? This can be a small RCT to see those who are attentive and not. So A, C, Very good. B, X, Y, X, B, and Z. Fantastic. Thank you so much. Thank you so much. It's also a strategy for us to let go of our laptops and our phones and concentrate on the next presentation. So thank you for closing your laptops. So as we start off, those were 4 serogroups of the 6 most virulent serogroups in meningococcal disease. So as we start off, this is quite a highly fatal disease and quite serious in our part of the world, and quite timely in terms of the audience that we have in this meeting. The content of our presentation will be first just introduction of invasive meningococcal disease, an overview of the current vaccines that are present, invasive meningococcal disease in Africa, the progress in meningococcal vaccination in Africa, challenges in meningococcal vaccination, future opportunities for vaccines, and conclusion of our discussion. We'll just start off with the disease itself. The main pathogen in invasive meningococcal disease is Neisseria meningitidis. This is an aerobic gram-negative oxidase-positive Diplococcus bacterium. It's exclusively a human pathogen. It's an obligate commensal organism found in the nasopharynx. It can be capsulated or non-capsulated. There are 12 known capsular serogroups. In some literature, there are actually 13 known capsular serogroups, and most of the meningococcal disease, the mortality and morbidity, is caused by 6 serogroups. That's serogroup A, serogroup B, serogroup C, serogroup W, serogroup X, and serogroup Y, 4 of which, of course, were in our exercise earlier. We have 7 other serogroups. That's serogroup E, H, I, K, L, M, and Z, which cause less of the disease, but these 6 out of the 13 cause a majority of the morbidity and mortality. We're going to discuss about the nature of this disease and also its epidemiology. You find that 4.5 to 25% of the general population are asymptomatic nasopharyngeal carriers. I'm going to stop there. That's around 5 to 25%. That means every 5th person in each row may be an asymptomatic carrier. That's what that means. And in this room that we are in, if somebody's coughing, sneezing, well, there you go. Only a small proportion, less than 1% to 5% of the carriers progress into invasive meningococcal disease. So a small percentage will progress into invasive meningococcal disease, but the tragedy of it is that 50% of those who get that bacteremia will then develop meningococcal disease. Transmission is through respiratory droplets, and that's also through the inhalation. And so you can find that that carrier status is quite key in the transmission. When we look at the epidemiology further, we find that it's low in children, around that 5%. It peaks in adolescents at around 23.7% and declines with age to around 7.8% after the age of 50. A lot of that activity and carrier status is in the adolescent group around 18 and 19, just purely because of their behavioral aspects. So they are in college, they're moving into hostels, they're in sports, they're hanging out together. So you find if there are carriers, then transmission can take place a lot during this age group in adolescence. When we look at the organism itself, we find that it does have the capsule and surface proteins which help it evade the immune system of the host. So you find that the capsule structure is how we classify the serogroups. So you find the various structure of the capsule is how we now give it its nomenclature in terms of serogroup A, serogroup B, serogroup C, serogroup W, serogroup X, serogroup Y, etc. And then we also have surface proteins. So you find that you have some pili that help in the attachment, some other surface proteins that also help in the adherence, causing pores into the whole cell, getting this pathogen into the cell, and of course, causing destruction. As we've discussed, of course, different factors that help in immune evasion, but also key from the capsule is how we get to develop the vaccine, because the structure of the capsule gives us the serogroup, and therefore from that capsular polysaccharide, we can then develop the vaccines from there for that to enable to train our immune systems to recognize these capsules and destroy the pathogen. In terms of the pathophysiology, as we've discussed, 5 to 25% asymptomatic carriers. In less than 1% to 5%, it does then go into our bloodstream and cause bacteremia. After causing that bacteremia, you find that in 50% or more, we can develop meningitis once it crosses the BBB, that's the blood-brain barrier. In another 20%, it can cause septicemia and meningococcemia. And then in 15%, pneumonia, and then also in other areas can cause disease. Why is this important? This is because we have a disease that has low incidence rates but high fatality. Even when the disease is diagnosed early and adequate treatment is started, 10 to 15% of those with invasive meningococcal disease will perish. Invasive meningococcal disease is fatal in 50% of cases if left untreated. 1 out of every 2. The case fatality ratio of invasive meningococcal disease among patients with meningococcemia is up to 40%. The elderly have the highest case fatality rates among the age groups. And when we look at the various serogroups, you find serogroup W is associated with the highest case fatality rate at 12.8%, serogroup C at 12%, serogroup Y at 10.8%, and serogroup B at 6.9%. These are all numbers, but for the person who gets this disease, it's 100% in terms of when they perish. So the effect is real. Severe physical sequelae are experienced by 10% to 20% of invasive meningococcal disease survivors. Permanent and potentially devastating sequelae among invasive meningococcal disease survivors include neurological damage and hearing loss. Invasive meningococcal disease is also associated with significant behavioral problems and psychological sequelae among the survivors. This means post-engagement with this disease, you may never be the same again, even if you survive. In terms of diagnosis, gold standard is isolation, culture, and gram staining of Neisseria meningitidis. As we said earlier, it's an aerobic gram-negative Staphylococcus. Neisseria meningitis may also be identified and serogrouped through molecular genetic methods, for example, the PCR. In terms of treatment, we have ceftriaxone or cefotaxime, chemoprophylaxis for close contacts, giving rifampin, ciprofloxacin, ceftriaxone to prevent secondary infections, and then supportive treatment. That's for the patient who has the disease. That's fluid resuscitation, management of shock, and then vaccination for prevention. So when we look at the symptoms of invasive meningococcal disease, we find that it does start with non-specific symptoms. So fever, headache, malaise, then progresses to photophobia, neck stiffness, and finally to altered mental state. So throughout the progress of this disease, you find that not very easy to diagnose, but very quick in terms of causing morbidity and mortality. One can actually lose their life within 24 to 48 hours. So it's quite the mix of characteristics for this disease, whereby you have these non-specific symptoms, but also high morbidity and mortality, and then quick in terms of 24 to 48 hours, one can lose their life. So you find that it starts to show that prevention is quite key for this disease, because by the time diagnosis is made, you only have a window of 24 to 48 hours. When we look at this graph, we can see the case fatality rate versus incidence, and we can see that it has low incidence rates but high fatality rates. Up there in red, you can see the meningococcal disease. So fewer cases but high in fatality. When we look at meningococcal vaccines, two main types: polysaccharide vaccines and conjugate polysaccharide vaccines. The conjugation is the addition of a protein that helps us with some of the characteristics of the vaccine. For the polysaccharide vaccine, this was introduced in the 1960s, helped with prevention, but had short-lived immunity, poor immunogenicity in infants, did not offer a great booster response, T-cell independent response, did not substantially reduce courage, therefore not very effective in herd immunity, and also can induce immunologic hyperresponsiveness. Polysaccharide vaccines are still used, especially in outbreak response in many of our countries, and quite still effective. Then came the technology of conjugation, and conjugate vaccines came into the scene, where we had T-cell dependent antigens, longer lasting immunological memory, and also reduction in nasopharyngeal carriage, therefore conferring that herd immunity because we reduce the carriage rate, therefore also affect the transmission. Numerous of companies have different products, especially in conjugation. So you find monovalent conjugated vaccines, bivalent conjugated vaccines, quadrivalent conjugated vaccines, and our recent discovery in this space is now the pentavalent vaccines in meningococcal vaccines. Important to mention, meningococcal vaccines for the B serogroup. Group B capsular polysaccharide is poorly immunogenic and has risk of autoantibody production due to its similarity to human neural cell adhesion molecules, and has made polysaccharide-based vaccines ineffective for Group B. Therefore, protein-based vaccines have been developed to overcome the challenges with polysaccharide capsule for Group B. And when we look at Group B, also the strain variability for Group B strains is highly diverse with variability in key surface proteins. And this renders it effective in preventing disease, but has been having limited efficacy in dealing with the nasopharyngeal carriage. So Group B, quite a clever serogroup, been not easy to create vaccines for it, but of course, we do have some vaccines that have not been very effective in dealing with carriage, but have been effective in preventing the disease. We are in South Africa and always must have a mention for the host country. Of course, meningococcal vaccines have not yet been included in the South African EPI schedule. Quadrivalent vaccines are available but still underutilized in both public and private sectors in terms of the South African landscape. Meningococcal vaccines against serogroup B, which is currently the leading cause of invasive meningococcal disease in South Africa, yet Men B vaccines are still not yet available in South Africa. Maybe this information has been updated, but as I checked last, they are yet to be available in South Africa. When we look at Africa as a whole, so the different serogroups are different epidemiology across Africa. Africa quite a large continent. Serogroup A, historically prominent in the meningitis belt of sub-Saharan Africa. The African meningitis belt is a belt as it stays in the middle of Africa. 26 African countries from Senegal all the way to Central Africa to Ethiopia in the east. So you find highest epidemiology and burden of disease for meningitis. Serogroup B and C, common in Europe, North America, Australia, and South Africa. Serogroup W and Y increasing in many regions, including South America and Europe. African meningitis belt, as you can see on your screens, those are the 26 countries from Senegal to Ethiopia. And you find that inner ring of red, that's where the highest burden of disease is. High endemic rates in these countries and annual seasonal outbreaks, but also explosive epidemics every 5 to 12 years, where you find it's actually a national issue, but annual pockets of outbreaks in these countries when it comes to meningitis. The region is home to approximately 430 million people, and peak epidemic incidence of the disease can actually exceed 1,000 per 100,000 population. So you can see a disease that has high fatality can actually exceed 1,000 per 100,000 population in incidence. This graph is one of the core graphs that shows vaccination works. So we can see the epidemiology of Neisseria meningitis type A, quite high in terms of its prevalence before introduction of the monovalent serogroup A conjugate vaccine around 2010, 2011, 2013, where there was a drastic drop in that incidence and prevalence of the disease. And we can see that since that incorporation of Menafrivac into EPI schedules and into countries in the meningitis belt, we have had that drastic reduction. So immunization does work. And by 2021, over 350 million people across 24 countries in the African meningitis belt had been immunized. 350 million. That's not a small number, but we can also see in the epidemiology the disease dropping. Of course, this was a monovalent vaccine, and subsequently a dramatic decline of one serogroup led to increase in serogroups WX and serogroup C in these countries, and therefore also the discussion of having not only monovalent vaccines but quadrivalent and even pentavalent vaccines because of the different serogroups that still cause morbidity and mortality. So this is the epidemiology in South Africa. Between 2004 and 2016, we find that Neisseria meningitis serogroup W was the main serogroup causing morbidity and mortality. And between 2016 and 2021, we find that men B are causing high morbidity and mortality. What is this telling us? And also the previous graph, that we cannot only rely on monovalent meningococcal vaccines, but also we need actually, quadrivalent or even pentavalent vaccines, because whichever serogroup would be having the highest epidemiology, then people have protection against all the serogroups that are available. And this is where the unpredictability of meningococcal epidemiology comes in, where at any one time and in any one outbreak, there could be a leading serogroup from the 6 that we've discussed earlier. Progress, meningococcal vaccination in Africa, where are we? Prior to 2011, meningococcal A accounted for approximately 90% of the meningitis cases, but introduction of a low-cost, temperature-stable, monovalent meningococcal A conjugate vaccine marked pivotal shift in the disease control strategy, and we've seen a decline in the epidemiology of men A, and we're now dealing with the other serogroups across the continent. World first, Nigeria introduced the new 5-in-1 vaccine against meningitis. And do we have any Nigerians in the room? Fantastic. Should be proud of this. Quite a great feat. Having the pentavalent vaccine deployed. And also in that rollout, they managed to vaccinate at least 1 million people with this new vaccine, which meaning people are being protected against all the 5 serogroups in that country where we have high endemicity for the disease. So there is progress from 2010, 2011, where we had that's the monovalent conjugate vaccine, to 2024, where we're now at pentavalent. Hopefully, of course, get to hexavalent and beyond. But of course, just showing that even on the continent, the war against disease is ongoing and prevention is taking center stage, especially in this highly fatal disease. What are the challenges in meningococcal vaccination? So just like any other vaccines, vaccine hesitancy is real. So historical medical exploitation, depending on the engagement between the people and the health programs. Misinformation campaigns, conspiracy theories. We're in the era of information, TikTok, WhatsApp, and algorithms. You click on 2 hesitant videos, the algorithm understands what you want to see. You click on 2 conspiracy theories, the algorithm knows exactly what you want to see, and they'll take you down that rabbit hole. So we have to engage, we have to be in those spaces. We cannot stay away, because these are gadgets that are in our pockets. So people are clicking on the on this information, and we have to be in the forefront, giving the real information. Misconception about vaccine targeting. So we are hearing subnational deployment of vaccines, outbreak response, we're in political eras. So you can imagine if a vaccine is not being deployed nationally and only in a certain region, the politics of the day and whatnot. These are real issues and how people can spin those stories. So we have to be aware of what's happening in the environment. We have to go ahead of this and discuss because our continent is highly political and of course, impact of unverified information. We have to be there to be proactive. Most times we are reactive with the information. We need to go ahead of it and ensure that we are ahead of this. And remember, those spinning these stories are more on the emotional aspect. So we can't also be very rigid. We also must go into making our information in a certain way that is palatable to the communities. Funding and infrastructure, another thing that's quite a crutch on our continent, funding and infrastructure. Conflict and political instability. Of course, you can have a great program, but if there is a war, a coup d'etat, etc., then that ceases. Antimicrobial resistance, which was extensively discussed earlier. Opportunities for future vaccines. Of course, there's been that development, of course, from monovalent vaccines all the way to pentavalent. Hopefully, the technology continues to improve. Defeating meningitis by 2030, a global roadmap. So 194 World Health Organization member states have pledged to defeat meningitis by 2030. 3 visionary goals. 1, eliminate bacterial meningitis epidemics. 2 is reduce cases and deaths. So a 50% reduction in cases of vaccine preventable bacterial meningitis by 2030 and 70% reduction in deaths globally by 2030. And then improve the quality of life for survivors. So some of the survivors have neurological damage, some have hearing loss, etc. Some become amputees. They've been amputated. So how are we preparing our health systems to deal with survivors? They've gone through a vicious war with the disease. Are we now going to leave them by themselves after they've won the war? 5 strategic pillars: prevention and epidemic control. So that's the place of vaccines. How are we preventing this disease? How are we ensuring, especially the most endemic areas, people are vaccinated? Diagnosis and treatment. Where are we in terms of our diagnostics? Because from diagnostics is where we get disease surveillance, and from disease surveillance is where we get this data that we're always asking for. So the data that we need comes from the labs. Where are our diagnostics? Then disease surveillance. That disease surveillance starts to give us a story. And when it gives us that story, it enables decision makers to make the right decision. Support and care for people affected by meningitis. So these are for the survivors. And then also advocacy and engagement. If we need resources, we must have share of voice. We must put up our hand and we must tell the leaders, look here, we need this here. Ongoing risk in the meningitis belt, of course, epidemics showing up. For example, Men C epidemic in Benin that showed up, Men X in Ghana, Men W in Congo, Men C in Togo. So these are cyclical and they occur every 5 to 12 years. So are we preparing for the next epidemic? In conclusion, there's been a lot of progress in terms of protection against invasive meningococcal disease. We have ongoing challenges as we've discussed, and it's the people in this room that will have the solutions. Thank you so much. And in this juncture, I'll stop there.
Please jam your hands together properly. Again, first I owe you an apology. I don't know why I keep calling you Kenneth. You are obviously Lawrence. All right. Any questions for Lawrence? Okay, there are also questions online. So let's take the first two in the room and then we go to the online audience. Please put up.
Thank you very much. It's a well-detailed presentation. I appreciate. Just a quick one regarding AMRs. It's a conversation we had yesterday, and you have presented it as a challenge. And you also said that you vaccinated more than 350 million. Do you have any data in terms of AMR in comparison to the vaccine uptake, whether it's increasing or decreasing?
Okay, it's great. So currently, no, and I think that also came up in the previous discussion on AMR, that this is a new space in which we are all going into and seeing the value and that correlation of this, the quantification of the value of, the AMR and the quantification of it in terms of course, monetary aspects, and also the ROI of the vaccination and that. So a study in terms of relating the two, in terms of investment in meningococcal disease, I've not seen such a study. But yes, AMR is a big issue. And if we were to get that challenge with meningococcal disease, you see, we only have that cefotaxime and ceftriaxone. It'd be a big mess in this space.
Well, thank you for this presentation. You have addressed the gap about the pneumococcal meningitis vaccine in South Africa, but we need to have a collaboration idea to address the opportunity, also to find a solution. I think the solution begin from integration of this pneumococcal vaccine into national immunization program. As is, I think it is highly cost. So integrated in the national immunization program will be very beneficial. Also targeted, also collaborate to make active surveillance to target the high risk group. We can begin for vaccination of high risk group. So we will prevent the spread of this outbreak or prevent the spread of epidemics. Also, I encourage the education campaign and increase awareness by the importance of meningococcal vaccine. And I encourage you also if there is any commendation from, thank you.
Fantastic. Thank you so much. But also in the same breath, of course, as she said, high risk groups. So we have Hajj travelers, and we know during Hajj, there's a lot of, crowding and staying close with each other. And we've seen the carriage rates. So what are we doing to ensure everybody who leaves the country is vaccinated so that when they come back, of course, then they're already protected, because they're not going to be, on the other side forever. They're going to come back home. So how are we doing in protecting those who are here? Because when those people come back, then they're going to meet their people, but also for them themselves as they go and meet all these people from all over the globe in one space. Yeah.
Thank you, Lawrence. There is a question from Colette. So she's saying, is the pentavalent meningococcal vaccine included in the Gavi portfolio?
We're very lucky to have Gavi team in the room. So I think maybe we can.
We have HPV.
Yeah, great.
HPV Gavi. I mean, the Nigerian launch was supported by Gavi, so definitely it is. Thank you.
Fantastic. Yes. Part of the program.
And also tomorrow we'll have about targeted vaccination for travelers with LB for the Hajj and the risk groups. Going for next question.
Thank you very much for that presentation. In terms of South Africa, there are these studies now looking at MenB, Bexero, for preventing gonorrhea. I know it might not be in adolescents now, I'm not sure, but maybe that could contribute to the MenB in South Africa if those studies are successful.
I guess we'll wait for the study to see to see what the study tells us. But yeah, we might have to wait for the results of the study. Yeah.
While the mic is making its way to, I think you wanted to ask a question. Oh, okay, then may I ask my question? Thank you. Just looking in terms of the meningococcal belt that you showed. Now, I'm Nigerian born, and I see that the belt cuts across my beloved country. Now, from the lens of equity and to balance, what will happen, for example, I'm a Southerner, and the South is very much excluded from meningococcal vaccination. So supposing I lead a demand that the Southerners must also be vaccinated, and then they come with this pentavalent vaccine, will it lead to the introduction of serotypes that were not prevalent in the area that was initially excluded? That's the one question. Then how do I, as a vaccine advocate, make the government understand that, look, there is a risk of inequity, and how do I make my fellow populace understand that, look, if you don't have a problem, don't ask for it?
Very, very, very multi-layered question. Thank you so much. Nigeria also, of course, has many dynamics between the North and the South. So vaccine equity is a real discussion in terms of Nigeria, both through the politics, the religious divide, the various ethnic groups that are there, and also, of course, resource mobilization. So all these factors, like we said, in terms of advocacy, it's not just about, this is the data and that's what it is, but also in contextualizing vaccination in where we are. So first of all, it's an inactivated vaccine. So of course, it's a kill vaccine. Whatever is going to be administered is not going to yield any vaccine-related disease. But secondly, also, there is resource mobilization. There are those who are at risk, and do we have enough money for vaccination for all? So this is why now stakeholder engagement is necessary. That stakeholders from the North and the South are all on the table. We all look at the local data that's there, and then we all engage as all stakeholders, both in the North and in the South, so that then information is what dispels conspiracy. So what is the data? Where are we? We have NITAGs. Can we all discuss? Can we all adopt this? And then can we all speak with one voice? So I don't know if that helps in addressing the issues here.
Okay, sorry, before Bronte, let me just quickly ask a follow-up question on that. I still, back to that map of the meningococcal belt, the Southwest, where quite a number of people go for Hajj, was excluded. So how do we look at that? Because like you rightly said, they won't stay on the other side forever. They will come back home.
Yes, and that's why I was saying all stakeholders on the table, so that then the decision is localized and it's owned by all. That, look, this is what we need to do. These are the risk groups. This is where there's high endemicity, and these are the vaccines that are there. How do we make the best decision for Nigeria? Because the decision will not come from without Nigeria. It has to come from Nigerians for Nigeria, because Nigerians are the ones who understand the dynamics in country. And the solution in Nigeria will not work in Kenya. Kenyans will have to make their decision, similar to South Africa, Senegalese, and all of the 26 countries in their meningitis belt. We must localize our decision making for our epidemiology to solve our problems. Yep.
Thanks, Lawrence. Bronte?
Thank you. Thank you so much, Lawrence. So from my understanding, South Africa's in the private sector just made Men A available this year. I stand to be corrected from my colleagues, but it is 2,000 Rand a dose. And knowing that that makes up a large portion of the diseased serotype in the country, how, and I'm posing this to you, but I guess also to the group as a whole, off the back of Prof. Radzani's presentation yesterday in terms of access, all that's doing is widening the gap. So I guess I'm posing to you, it's a question about access in terms of cost and reaching, really vulnerable populations. I know it's a tough one. Thanks.
I think that's an ecosystem question for the whole ecosystem in terms of vaccines. So cost drivers, of course, access, you have your payers as well. You have your government, in terms of also your regulators. So it's not, one answer question. So why are we at this cost? What is the main cost driver? that has to be addressed. And then from there, of course, access, equity, those are big discussions. So I may not have a one sentence answer for that. But like I said, it's a vaccine ecosystem question in terms of why does it cost so much for such a necessary intervention. Yep.
Thank you. Thank you very much. So I'm not officially here, but maybe this is a nice opportunity to just prime you to think a little bit about the talk on travel vaccines tomorrow, because it becomes more complicated. What Madam Speaker has just mentioned about the North and the South in Nigeria. So if the South of Nigeria is asking the question, why are we not vaccinated? That's part one. Part two is, now I'm going to vaccinate my South African travelers who are going to Nigeria. They're not necessarily going to the north, they may be going to the south. If we are saying that for the population, the risk is so low, not high enough to vaccinate them, why am I vaccinating my travelers? And in part, that question arises because 30 years ago, when I started travel medicine, you're going to the meningitis belt, you're going to get a vaccine. The vaccine was around. I think the vaccine was already around. But now, because of the success we've had with the meningitis belt, is it still justified, also from a cost point of view, to say, not only do you need a yellow fever vaccine to go to Nigeria, but you should really also get a meningitis vaccine. So it becomes very complex. And I think it's easy in a way for people who work with public health money and where you're talking large numbers and you've got nice statistics to say, what is the risk for this part of the population? What's the risk for that part of the population? But now when we talk about individual travelers, it becomes a lot more difficult to say, when is the risk acceptable and when is the risk not acceptable, and when should we be using the vaccine to protect an individual traveler? And then at the same time, every individual traveler that is being vaccinated, you can either say that we're widening the inequity gap, or you can say every drop helps. So I'm not giving answers, I'm just leaving them out there. If you're wondering what to do tonight, you can think about it.
Well, yeah. A lovely lady has told me must respond. Yes. Yes. Very, very good that you bring that out. And the science is the science, the data is the data, and the epidemiology is there. If it's endemic and people are going somewhere, let them be protected against disease, because it would be sad that they could have been protected against a vaccine preventable disease and they didn't get protected. So the science is the science, the data is there. Other factors come in place, of course, population in Nigeria and the cost of everybody and whatnot. But for those who can be vaccinated, let us not be the barriers. Let us ensure that they're protected so that then we deal with disease. So for those who can get the vaccine, let them be vaccinated.
We have one last question online. So Anthony wants to know whether there's any progress on vaccines for non-bacterial meningitis.
That's quite a large pathogenic group.
Yeah, I guess like
Yeah.
cryptococcal meningitis.
Yeah. Viral meningitis and whatnot. It's quite wide. So yeah. Of course, technology every day getting better in terms of vaccines. But of course, there's also prioritization in terms of which is highest morbidity and mortality, and then where do we save lives? Earliest, fastest. So quite varied. I mean, in terms of how many pathogens we can discuss here.
Yeah, so I have a question building on Anthony's question. Do you know if there's any vaccine work that's ongoing when it comes to cryptococcal meningitis? So cryptococcal meningitis was my PhD baby, so I'm just curious now. Yeah.
Well, numerous companies, of course, numerous ideas. I'm only here keeping brief for Sanofi, but I may not speak for all organizations in terms of the work that they're doing, including also in crypto. So let me probably leave it at that. But if anything pops up, of course, then I do have your email. I will shoot it across.
Just to follow up on Sepi's question, and I'm glad, Sepi, we do have some things in common, because I also started with cryptococcus neoformans, and I know it could be a nuisance, particularly in the immunocompromised population. So, and it's a fungus, it's a yeast, but it's a fungus if you trace up the phylogenetic tree. And those guys are, if you think protozoans are difficult to deal with, I mean, let's not go into granular details, but anybody who has had to deal with candidiasis will know that dealing with fungus or yeast, they are terrible. So I think you've saved yourself by not speaking for other companies, but seeing that it is a challenge, and that in South Africa, we do have a large immunocompromised population. Maybe when next we see Morena, we'll pose that question to him to see if BioNTech has anything, in the offing along those lines. Thank you. Any other question for Lawrence that I call Kenneth? No other question. Thank you so much, Lawrence. And we have 4 minutes more. Lawrence has saved us time. So I don't know if there is also any other question for Rose. Rose, are you still online? She's not. All right. So we're saving 4 minutes. Nella? Bronte. All right. Jam your hands together for my beautiful colleague, Bronte, as she takes us through the daily assessment. Thank you. And please clap for me.
Thank you, Madam Chair. Afternoon, everyone. It's that time of day when you hear my voice, you're going to know what's coming for the rest of the week. It's no more, no more listening, but we do need your feedback. So just like we've done yesterday and the day before, we'll be putting there you go, a QR code on the screen. And I think you know what to do. Please use your smartphone